Buenos dias, everybody. Welcome to day 3 of the National Plant Biochemistry and Molecular Biology Congress. We will get started, but first, an announcement. If you have been attending this meeting and you love it, and you want to be an organizer for the next meeting, please stop by if you can to our business meeting, which is from 5 to 7 p.m. Mexico City time tonight, using this exact link that you have here. We would love for you to help organize the next meeting, and hopefully without a pandemic, it will be in some beautiful place in Mexico. So please stop by if you would like. Without any further ado, let's get started. Our first speaker is Hai-Ling Jin. She's from University of California, Riverside, and she'll be talking on a very interesting topic today: cross-kingdom RNAi and extracellular vesicle-mediated small RNA trafficking between plants and fungal pathogens. We're happy to have you here. Take it away, Hai-Ling.
Thank you so much. First, I would like to thank you for the invitation, and I also want to say that it's a great meeting, and the organizers did a great job to share the meeting with the public, so allow the graduate students and postdocs to have free access. That's a great job. All the students and postdocs enjoyed the meeting very much. So today I would like to take the opportunity to introduce our work on cross-kingdom RNAi and small RNA trafficking between plant and fungal pathogens. My lab is working on bacteria and fungal pathogens and their interaction with host plants. Today I will only focusing on fungal pathogens. As everybody knows that fungal pathogens pose a great threat to global food security. It can cost billions of dollars crop loss every year. The fungal pathogen I chose to work on is called Botrytis cinerea. I'm sure everybody have seen this because it can cause gray mold disease on almost all the fruits, all the vegetables, and many flowers. And it was ranked the #2 most harmful plant pathogens by Sarah Ger and her colleagues. And the Botrytis has its own RNAi machinery. It encodes dicer proteins and organelle proteins, so it can produce a population of small RNAs, can manipulate its interaction with the host. So a few years ago, like small RNAs are short non-coding RNA molecules that can cause induced gene silencing. They are processed by dicer or dicer-like proteins into small RNA duplexes. And then these duplexes are incorporated into another class of proteins called organelle proteins. So the organelle proteins are the effector proteins of RNAi, can take the RNA and find their target RNA with complementary or partial complementary sequences to induce silencing at post-transcriptional level by inducing mRNA degradation or translational inhibition, or at the transcriptional level by inducing DNA methylation or histone modification. So a few years ago, a talented postdoc, Anna Weiberg, and a very good graduate student, Ming Wang, they discovered that when Botrytis cinerea infect plants, it can trigger immune responses just like any pathogen infection. But Botrytis evolved a novel mechanisms that can deliver its small RNAs into the host and hijack host organelle protein to silence host immune responsive genes, including MAP kinase cascade components, cell wall associated kinases, and other immune responsive genes to achieve very aggressive infection. So this cross-kingdom RNAi was adapted as an advanced virulence mechanisms by those aggressive fungal pathogens like Botrytis cinerea. So we were very happy to see that a year later, similar findings was also reported in animal parasite interaction systems. So Amy Burke's lab from University of Edinburgh, they discovered that gutworm nematode parasite can also transfer its small RNAs into mammalian cells and modulate immune responses. And even not only this mechanism is conserved, but even the small RNA targets pathways are also conserved. So what she found is the parasite small RNA, one of the parasite small RNAs, it can also target a regulator of MAP kinase signaling pathway. As you remember, we talked about the fungal small RNAs can target host plants, MAP kinase cascade components, right? So over the last few years, more and more reports from both the plant and animal systems to show that plant fungal pathogens like Verticillium dialy or Paxinia strophomis, they all can deliver its small RNAs into their host, either Arabidopsis or wheat, and suppress the host immune responsive genes and then achieve aggressive infection. And even rhizobia, which is a bacteria, cannot produce, it doesn't encode, its genome doesn't encode RNAi machinery, but its tRNA can also generate small RNA-like RNAs, and those small RNA-like RNAs can also get transferred into host cell and then hijack host organelle 1 to regulate modulation in ligands. We were also very happy to see that a couple of years ago, animal fungal pathogen, which is a mosquito fungal pathogen that can also deliver small RNAs and then get into the mosquito cells and hijack mosquito organelle 1 protein to silence mosquito immunity gene, this TOR receptor ligand SPEZO4 gene. So here you can see that not only cross-kingdom RNAi is broadly present in the host pathogen parasite interaction settings in both plants and animals, but also even the molecular mechanism is conserved. They can use host organelle protein to silence host immune responsive genes. So then after we see that a fungal pathogen can deliver small RNAs into the host and silence host immune responsive genes, one of the key questions we asked is this cross-kingdom RNAi bidirectional? Because during that time, people already observed that if you ectopically express some small RNAs or double-strand RNAs, target pathogen genes, this silencing can work. We call it host-induced gene silencing. So is there any host endogenous small RNA can also be delivered into the interacting organisms? And in order to answer that question, one of the most straightforward way to address this is to isolate purified fungal cells from the infected tissue to see whether they carry host small RNAs. But this has been very challenging because it's almost impossible to isolate pure fungal cells from infected leaves. Many of my colleagues told me that, Hailin, you forget about it. But I was very lucky that I got another very talented postdoc, Chang Cai, and then he came to the lab and very briefly take on this project. And then a new method was developed. We took the advantage of the differences between the plant cell wall and fungal cell wall. Plant cell wall is made of cellulose, hemicellulose, and pectin, and the fungal cell wall is made of chitin and glucans. So in that case, we can basically separate these two different types of cells. So what Chang collected a lot of infected tissue and first used the cellulase and the microenzymes to dissolve all the plant cell wall, and then use Triton X 100 or any detergent to break all the plant protoplast. So all the plant cells were released after you dissolve the cell wall, and then break them with detergent. Then you centrifuge and collect the leftover cells, and those cells are mostly fungal cells. But as you can see, these fungal cells are still pretty dirty because they have a lot of plant cell debris. So in order to purify these fungal cells, we did the second step protoplast prep, which to remove all the fungal cell walls by digesting with the lysine enzyme from trichoderma. And then after several rounds of wash and purification, centrifugation, we purified a pretty clean fraction of fungal protoplast. And then I asked Chang to do another step of quality control to make sure that the purified protoplast of fungal cells are not containing abundant Arabidopsis host, abundant RNA, such as actin. Sometimes he can still detect the actin by RT-PCR. So in that case, we have to abandon that prep, that sample, because if you can detect something by RT-PCR, even very trace amount, you can see a lot in the deep sequencing. So he finally managed to get several preps with this high quality of purified botrytis protoplast. Then he made a small RNA libraries and then subject to deep sequencing. So even we use 40 reads per million as a cutoff, which is pretty high cutoff, high stringent cutoff, he still find about like several dozens of host small RNAs present in the fungal cells, including microRNA and the siRNAs, such as microRNA 166. So when I see microRNA 166, I feel like, microRNA 166 is pretty abundant. So if they move into fungal cells, is it because they are very abundant? So it's just a simple diffusion, concentration-dependent diffusion. So in order to answer that question, we look into the host small RNA list more carefully. Then we feel like it's not the case because we found several pairs of small RNAs, such as TACI2, siRNA 453, and the 7110. These two small RNAs, small interfering RNAs, they are generated from the same non-coding mRNA precursor TACI2. Both are accumulated to a very similar level in the total RNA fraction, but only one got transferred. These two small RNAs are also generated from the same precursor TACI1C, and both accumulated to the similar level, only one got transferred. These two are another pair of examples that they are generated from the intergenic region, but only one got transferred. So based on this, we are pretty confident that those host small RNAs are transferred to the fungal cells, not only by simple concentration-dependent diffusion, but rather by selective transport. So this is rather selective. Only one got transferred, not the other one, even though they accumulated to the similar level and they generated from the same precursor. So now we know that plants can also deliver their small RNAs into the fungal cells rather selectively. But then the next question is that how do plant cells deliver small RNAs into fungal cells? Although cross-kingdom small RNA delivery has not been reported during that time, but extracellular vesicles has been shown to transport small RNAs between cells within organisms like in the mammalian systems, like nerve systems, like tumor systems, or body fluid, like blood or other body fluid within our organisms. So we hypothesize that probably plant also use extracellular vesicles to transport small RNA, although at that time plant extracellular vesicles has not been well studied. So in that case, we adapted the method from mammalian systems to isolate the plant extracellular vesicles from apoplastic fluid. So apoplastic fluid are the extracellular fluid in the leaves, so that contains a lot of secrete molecules in potentially contains secrete vesicles. So we use the mammalian system method by using ultracentrifugation and purification. Using 100,000 G, we purified the extracellular fraction. Recently, we published a paper in JIPB, Journal of Integrative Plant Biology, specified the best method for extracellular vesicle purification from plant leaves. So if you are interested, you can check this paper out. We lay out a clear protocol for EV extraction. And we use the same quality control after the EV extraction. We make sure that the abundant mRNA are not, the host RNA are not detectable by RT-PCR, and we use this quality extracellular vesicle fraction for small RNA profiles. Again, we did 3 biological repeats, just like what we did with a fungal protoplast, and then only chose those small RNAs that are present in every library of the 3 biological repeats to at least 40 reads per million. And then we found a couple of hundred of host small RNAs in this extracellular vesicle fraction. And then furthermore, we found that the small RNA patterns are very similar to what we found in fungal cells. So more than 80% of the host small RNAs we found in fungal cells, fungal protoplast, are all present in the vesicle, extracellular vesicle fraction. So this tells us that those host small RNAs are probably use extracellular vesicles for delivery. So extracellular vesicle is one of the major pathways for small RNA transport from the host to the fungal pathogens. We further confirmed that these fungal small RNAs are inside the vesicles because we were using micrococcal nucleus treatment, and those small RNAs are still protected by the EV envelope, unless we rupture the vesicles using detergent like Triton X100. And then some people worry that even we use a micrococcal nucleus, maybe it still cannot remove all the RNAs outside of the vesicles because they might be protected by proteins. So in order to address that, we even use the protease, like protease K, together with micrococcal nucleus. So even if the RNA is protected by protein, we remove the protein by protease K and use a micrococcal nucleus to remove all the RNA. We can still detect those host small RNA. That gives us the confidence that indeed those host small RNAs are present inside extracellular vesicles. So then, although extracellular vesicles were not well studied in plant systems, but in mammalian systems, they have been extensively studied. They can be divided into several different classes based on their biogenesis pathway and their biomarkers, protein biomarkers. So, and the exosomes, which is a small extracellular vesicle that have touch spanning as a biomarker and derived from multivesicular bodies. So this class of extracellular vesicles has been shown to play a pretty important role in small RNA transport in mammalian systems. So in order to determine whether plant also has different classes of extracellular vesicles, if plant also have exosomes, we start to look at the tetraspanin family. Although by protein homology analysis, we didn't find any close homologs in Arabidopsis that have amino acid similarity to mammalian tetraspanin family genes. But based on the protein structures, this one class of proteins, now we call them tetraspanin-like proteins in Arabidopsis, they share very similar topology to the tetraspanin in mammalian systems. But there are 17 members in this gene family. So in order to narrow down which tetraspanin genes in Arabidopsis we should focusing on, we cannot work on 17 genes at the same time. We first check their expression level, and we found only two of them are induced by botrytis infection, which suggests that they play probably a more important role in plant responses to botrytis fungal infection. So we mainly focus on the tetraspanin 8 and tetraspanin 9. So Sheila McCormick's lab was working on this class of genes and look at their function in pollen development. So they developed the marker lines and the native promoter. So she was very kind to provide us these mark lines. And then we found that tetraspanin 8 GFP transgenic plants, after botrytis infection, you can see a lot of vesicle-like structures are concentrated at the botrytis infection site. So this is the botrytis cell, and these are the fungal, the plant cell. And then you can see there are a lot of vesicles or multivesicular body-like structures around the infection site. But here we don't really know if those vesicles are secreted or not. So we isolated the extracellular fluid from the infected tissue, and in this fluid they don't have plant cell anymore. These are extracellular fluid, and then we can clearly see the vesicle, the tetraspanin 8 GFP labeled vesicles. So furthermore, we show that tetraspanin 8 is also colocalized with the plant MVB, the multivesicular body marker, ARA6 inside the cell. So this data tells us that plants also have exosomes, subclass of extracellular vesicles, because they have this tetraspanin as the biomarker, and that they are derived from the MVB, just like in the mammalian systems. So plants indeed have tetraspanin, exosomes, tetraspanin positive exosomes. Furthermore, we use electron microscope to observe those extracellular vesicles at the interface of the fungal infection site and a plant interaction site. And this EM picture catches the right moment when the outer membrane of the MVB fused with the plasma membrane and release these interluminal vesicles. So those are considered to be exosomes, a subclass of plant extracellular vesicles. So then, since plant may have multiple class of different extracellular vesicles, so one of the most advanced way to purify a subclass of specific extracellular vesicles is to use immunocapture. In order to have a successful immunocapture to purify a subclass of extracellular vesicles, you need to have a good antibody. But the tagline cannot work because both the N-terminus and the C-terminus of the tetraspanin proteins are both inside of the extracellular vesicles. So you won't be able to use the end tagline for pull down. So in that case, we generated a native antibody that can recognize the extravesicular domain, the EC2 domain of the TET spanning 8. And then luckily, we got a very good TET spanning 8 specific antibody, native antibody. It can only detect one band in the EV fraction, and then this band disappeared in the TET spanning 8 single mutant background. So that means this antibody is very clean and also is very specific to TET spanning 8. So we use this good antibody to purify TET spanning 8 positive EVs or exosomes. We pull down those the exosomes and then purified it and then detected, still can detect the host small RNAs in those in this purified TET spanning 8 positive exosomes. So this experiment provided the definite answer that plant small RNAs, including microRNAs and siRNAs, they can be transported by TET spanning positive exosomes in the plants. So now we know plant can secrete small RNAs using exosomes, the subclass of EV. And then the next question is, can those EVs get into the fungal cells? They can't get out from the plant cell. Can they get into fungal cells? So in order to answer that question, I asked Chang to use the extracellular fluid, which only contain the vesicles. And we use these vesicles, we use the TET spanning 8 GFP transgenic line to isolate this apoplastic fluid, purify the extracellular vesicles so you can see those vesicles very clearly. Then we just put some fungal cells into the vesicles. And then Chang was very excited to show me that. He said, well, within 2 hours, all the vesicles got into the fungal cells because after the Triton X detergent treatment, after 2 hours, all these TET spanning labeled vesicles are still there inside the fungal cells, cannot be ruptured. At the 0 hours, all these vesicles can be ruptured, nothing gets in. So this blew me away. It is very clear that those plant extracellular vesicles, these exosomes, can be literally taken up by fungal cells. And then in these fungal cells, we can easily detect host siRNAs and microRNAs. So this tells us that plant can not only secrete those small RNAs using extracellular vesicles, and then those extracellular vesicles can also get into the fungal cells very efficiently, and then those small RNAs can translocate from the plants to the fungal pathogens. So then the next question is, once these plant small RNAs get into the fungal cells, are they functional in the fungal cells? So in order to answer that question, we took the genetics approach. We isolated TET spanning 8 and TET spanning 9 single and the double mutants. The TET spanning 9 mutant in the stock center is not correct, so we have to generate the CRISPR-Cas9 of the TET spanning 9 single mutant. We also generated the double mutant in the TET spanning 8 knockout mutant background, silenced TET spanning 9. So this is a knockout, knockdown double mutant line, not a completely knockout. It's a TET spanning 9 is the knockout down line. And then in those single and double mutant, we can see that botrytis protoplast isolated from the fungal cells, they contains much less host small RNAs. And then this double single mutant shows much more susceptible phenotype compared to the wild type. This tells us that this, when you knock down the transport pathway, the vesicle pathways, the plants become more susceptible. That tells us that those transport pathway, those extracellular vesicles contain molecules or or proteins or RNAs that can contribute to plant defense. So if you knock down this transport pathway, plants become more susceptible. Since we already know that those extracellular vesicles contain host small RNAs, so we hypothesize that probably those host small RNAs can have targets in the fungal cells and silence fungal virulence-related genes. So we predicted bioinformatically the potential targets of those host small RNAs in fungal genes, and we found indeed that there are a set of fungal genes can be targeted by the host small RNAs. Strikingly, more than 20% of those fungal target genes, they are involved in vesicle transport in fungi. So this actually makes a lot of sense because fungus needed to secrete like a cell wall degradation enzymes and toxins and many other molecules to attack plants during infection. So they need to have a vesicle transport pathway to do the work. So plant is pretty clever, try to target those the transport pathway to inhibit the delivery of fungal virulence-related molecules. So to confirm our bioinformatics prediction, we examined the expression level of the 3 candidate genes that are involved in the fungal vesicle transport. These 3 are the potential target genes of the host small RNAs. And we found that after infection, the expression of these target genes are indeed downregulated. So then we generated the deletion mutant in the botrytis, which delete all these 3 genes. So we have these 3 different mutated botrytis strains, and these mutated botrytis strains become much less susceptible, much less virulence on Arabidopsis. That tells us that this host small RNA target gene in botrytis really contribute to pathogen virulence. So we were very happy to see that one of the candidate genes we discovered in botrytis, VPS51, which was also determined to play a very important role in virulence in a human fungal pathogen, Candida albicans. So some of those novel virulence-related factors in fungus are also conserved between plant pathogens and human fungal pathogens. And this actually is a pretty good way to isolate or identify novel or new virulence-related genes by looking at the host small RNA potential target genes in fungus. So in summary, what we found is that during fungal-plant interactions, fungal pathogens like botrytis, they can deliver small RNAs into the host and utilize host organoid proteins to silence host immune-responsive genes. But during this evolutionary arms race, plant also evolved similar mechanisms. They can also deliver its small RNAs using these extracellular vesicles, especially the multivesicular body derived exosomes, deliver small RNAs, use these exosomes into fungal cells, and then these vesicles can get into the fungal cells and suppress fungal virulence-related genes. So this cross-kingdom RNAi is bidirectional, has been co-evolved during the arms race between plants and fungal pathogens. So during this study, one question always bugs me. I'm sure many of you also ask the same question. How are plant small RNAs selectively transported? I hope you still remember like the small RNA different pairs, only one got transferred, not the other one. So in order to answer that question, I hired another very talented postdoc, Bao He, and he has quite a lot of experience with proteomics analysis. So we hypothesized that probably extracellular vesicles, they contain some specific proteins, such as specific RNA binding proteins. They can contribute to the selective loading of small RNA. So Baoye performed the MSMS analysis and purified the EV used the same quality control as I mentioned before. And then he submitted this purified the EVs for proteomics analysis. And again, use the same 3 biological repeats as our standard approach. And then he only used the proteins that you only select the proteins that are present in all the 3 biological repeats with a minimum 5 distinct peptide reads. So use that cutoff, he still identified about 20 RNA binding proteins from the proteomics analysis, including Arabidopsis argonauts-1, and then dead box RNA helicases RH11 and RH37 and RH52. So these 3 RNA helicases, they belong to the same subclade of the dead box RNA helicases in plants. And then we first confirmed that these RNA binding proteins are also co-localized with the extracellular vesicles. And then we also found another class of RNA binding protein called annexin 1 and annexin 2. They have been shown recently also have RNA binding, small RNA binding activity in addition to their calcium binding activity in mammalian systems. And then we confirmed not only by the cell biology approach, but also by biochemical approach. We can detect those RNA binding proteins like argonaut-1, the RNA helicases, using this in the tetraspanin 8 positive exosomes. So we can clearly detect those RNA binding proteins. But argonaut-2 and argonaut-4 was used here as a negative control. They were not identified in the proteomics analysis, and they are not present in the tetraspanin positive exosomes. And these RNA binding proteins are also inside of the vesicles because we use trypsin protease digestion, and they are still there unless we rupture it using detergent. And what we found is that the small RNAs that transferred into, got loaded into extracellular vesicles, they are mostly associated with argonaut-1. So as I mentioned, in Arabidopsis, there are 10 argonaut proteins, and each have their selective favorite subclass of small RNAs. So argonaut-1 in plants, it tended to bind with 20 to 22 nucleotides long small RNAs with the first nucleotide U. So we found that the small RNAs that got translocated into, got loaded into extracellular vesicles, they are more likely that they are all associated with argonaut-1. But the ones that are associated with argonaut-4 or argonaut-2 or argonaut-5, they are not present. And we further pull down this argonaut-1 protein using argonaut-1 antibody, and we can easily detect those EV localized small RNAs in the argonaut-1 binding fraction. And then similarly, we use the antibody to pull down the RNA helicases 37, 11, and 52. We can also detect those AEV localized small RNAs. So this tells us that this host argonaut-1 and the RNA helicases, these RNA helicases contribute to the selective loading of small RNAs into EV. So in summary, we found the cross-kingdom RNAi in plant-fungal interactions. So several years ago, we wrote a review, and although we know that fungal pathogens can deliver small RNAs into the host, but we predicted that these kind of interactions are bidirectional, and then this the communication, the small RNA level communications should be present in many interaction settings. But at that time, I put all the prediction using question marks. But now our current study can remove the question marker. We definitely demonstrated that plant and fungal pathogens have a bidirectional cross-kingdom RNAi. And then Michael Axtell's lab also showed that parasitic plants can also deliver small RNAs into the host and suppress host gene expression. And then even the cross-kingdom RNAi not only present in this pathogen-host interaction setting, it is likely to be present in also the friendly symbiotic setting, because people now are using host-induced gene silencing to express small RNAs in the host and then let them to transfer into the mycorrhizae fungi and to suppress mycorrhizae gene expression. So so this small RNA trafficking also present in the symbiotic interaction setting. And then also many studies show that, recent studies show that this cross-kingdom RNAi also present in the host insect interaction and host nematode interaction. So recently we wrote a new review, but inherited the same theme, but come up with a new set of questions. I predicted that probably extracellular vesicles play a more important role in the communications, especially RNA-mediated communications between the interacting organisms like a host and pathogens, or host and other parasite or symbiotic organisms. And I hope in the near future, at least some of those question markers also can be removed. So in the end, I would like to use the last 5 minutes to show our application work, how we translate what we learned from the basic research to the real agriculture. And this work were mainly done by Ming Wang, Jonathan, Lulu, and Rachel. Jonathan was also a postdoc who recently become a PI back in Spain. So he is the 11th postdoc from my lab, got a faculty position. So we show that the small RNA effectors are transferred from the pathogens to the host, and we found these small RNAs are dependent on both DICER1 and DICER2. So when we knock out both DICER1 and DICER2, but the pathogen virulence are largely inhibited. So in this case, we can use DICER1, DICER2 as a major target for inhibiting fungal virulence. So we generated transgenic plants that express small RNAs or double-stranded RNAs that can target botrytis, DICER1, DICER2. Those plants are become more tolerant or resistant to botrytis infection. And this method can be used to control multiple pathogens at the same time. So we generated some transgenic plants that express small RNAs target both botrytis dicers and the verticillium dicers in the same plants. And these same plants become more resistant to botrytis and the verticillium infection. So that tells us that cross-kingdom RNAi can be used to control pathogens, even multiple pathogens in the field. However, even though this host-induced gene silencing is working, but we have to generate a GMO. And the GMO is not easily be generated in many species like trees or flowers. And also it's still a big concern. So we needed to have a faster non-GMO and especially eco-friendly approach, because now we mainly use fungicide to control the fungal diseases. So environmental RNAi was observed in C. elegans, but not it didn't work in vertebrates. And then it's not clear if it works in plants and fungus. Many people said that it probably won't work. But when I dig into the literature and ask about this with my colleagues, and it seems like people didn't try very much on this. So I asked Ming to give it a try, to label all the RNAs, like small RNAs and double-stranded RNAs with fluorescent, and then just mix the RNA with the fungal cells, and we can see those RNAs can get into botrytis cells. Even when we remove the fungal cell wall and then use a micrococcal nucleus treatment, those RNA are still there. So that means fungal cells can indeed take up RNAs very efficiently. So in that case, we can design double-stranded RNAs and small RNAs that can target important virulence-related genes in botrytis. and then we spray those RNAs onto fruits, vegetables, and flowers, we can see that the RNAs that can target botrytis dicer 1, dicer 2 can inhibit the botrytis infection pretty effectively. So this RNA-based so these RNAs can serve as new generation of fungicide for disease control. So in order to check if this RNA uptake ability is only for botrytis or for many fungal cells, we did a survey and tested on all the fungal pathogens that we can get hold of, and including botrytis, sclerotinia, rhizoctonia, which is a rice fungal pathogen, verticilliums we talked before, and aspergillus, a very serious post-harvesting fungal pathogens. What we found is that all of these aggressive fungal pathogens, they can take up RNA very efficiently within like 6 to 12 hours. So in that case, we can use this RNA approach to spray double-stranded RNAs or small RNAs that can target fungal virulence-related genes like dicer 1, dicer 2 genes, or the genes that involved in the trafficking pathway, as we detected as a host small RNA target, and we found that these genes really can suppress a fungal infection.
I'll have to stop you there, Hailin. Absolutely fascinating. A talk filled with facts and just thoroughly data. We really have to move on now.
Okay, so yes. So in the end, I just want to say this RNA-based fungicide is working well in other for many to control many fungal pathogens. So this is the acknowledged slides for the funding support and all the collaborators and the people who work on the project. Thank you.
A wonderful talk. And we have no time for questions, but there's a lot of comments in the chat, and maybe you can answer in the chat some of the questions. Wonderful talk.
Okay, thank you. Sorry about the time. Sorry about that.
Thank you.
Bye-bye.
That was a great talk. So we continue now to the next talk. So it's very nice to see so many people connected, seeing many friends. Welcome all. So the next talk will be by Osvaldo Valdez Lopez from the FES Iztacali UNAM, and his talk. So go ahead, share your screen, Osvaldo. So his talk will be about modulation of the root nodule symbiosis by plant host phosphate status. Take it away. Go ahead.
Thank you very much for the invitation. So today I will present some of the data that my lab generated, and I will try to convince you that the plant host phosphate status play a major role in the establishment of the legume-rhizobia symbiosis. As you know, legumes are able to interact with different microbes, but I would say the most agronomically important interaction is with mycorrhizal fungi and also with rhizobia. So with this symbiosis, legumes can obtain phosphate, but also can obtain nitrogen without the application of any synthetic fertilizer. But today I will focus only in the interaction between rhizobia. So as you know, to establish this symbiosis, plants need to communicate with the rhizobia. So for that, plants need to grow in a very low concentration of nitrogen. So when the plants are in that condition, so the plants will release different molecules, including flavonoids. So these flavonoids will recognize by the compatible rhizobia, which in turn will release lipocyto-oligosaccharides, which will be recognized by the legumes host. So this is a very a summary of how the legumes communicate with the rhizobia. But in detail, I will not go in detail with all the genetic behind this interaction, but I will just make a summary how the legumes communicate with rhizobia. So as I mentioned before, once the legume released to the rhizosphere the flavonoids and isoflavonoids, so the rhizobia will be detected and will release the lipocyto-oligosaccharides. So those LCOs molecules will be detected by different receptors that are localized in the plasma membrane, particularly in the root hair cells. So once these LCOs are detected, it will activate different molecular response, including, and I would say the most important, is this oscillation of calcium concentration in the nuclei that we call calcium spiking. So this calcium spiking are decoded by protein kinase, particularly calcium calmodulin-dependent kinase. And after that, it will trigger different molecular responses, including the activation of the expression of different symbiotic genes that are required for the first step of this symbiosis, which is the curling of the root hair. And this response is necessary to entrap one of those rhizobia, and then the plants will form this structure that we call infection thread that is required to transfer the rhizobia into the cortical cells that in parallel is experimenting a different cell division to form a new meristem that we call the nodule meristem. So in this part, participate different genes, and I would say one of those is NIN, or nodule inception, which is a transcription factor that will coordinate the rhizobian infection process, but also it will coordinate the nodule formation. So we know that legumes can form different types of nodules, and it depends on the legume species. One of those is the indeterminate nodules, which is which has a persistent meristem and different zones: the meristem zone, the infection zone, the fixation zone, and the senescence zone. So this kind of nodules are particular in Medicago truncatula or Medicago sativa. And the second type of nodules is the determined nodules that only has the fixation zone that later it will become the senescence zone. So this type of nodules are particular in common bean or soybean, or those legumes that grows in tropical or more in tropical zones. Sorry about that. So this symbiosis is really important because through this symbiosis, legumes can obtain nitrogen from the atmosphere. So which means that the bacteria, which has the nitrogenase enzyme, will transform or convert the atmosphere nitrogen into fixed forms of nitrogen, like nitrate or even ammonium or amino acids. So in exchange, legumes will provide different carbon sources like carbohydrates to maintain this symbiosis. So as you can imagine, to convert this atmosphere nitrogen, it requires to invest a lot of energy, particularly ATP. So here we have the equation that for one single mole of atmospheric nitrogen, the plants need to invest 16 ATPs to have 2 molecules of ammonium. So this is only for the conversion of atmospheric nitrogen into fixed forms. But if we consider, sorry about that, all the steps required to establish this symbiosis, the plants need to invest a lot of energy in form of ATP. So for the plant, it's a cost-energy process, and the plants need to balance this symbiosis in order to have an optimal development and also yield. So to have a balance between growth and this symbiosis, legume plants has a particular signaling pathway that we call as autoregulation of nodulation. So in summary, so when plants begins to interact with the rhizobia and start to activate this cell division in the cortical cells, immediately 2 genes are activated that encode for these small peptides that we call rhizobia-induced peptides, 1 and 2, or RIG1 and RIG2. These 2 peptides are produced in the roots and are transported through the xylem to the upper part of the plant. And in this part of the plant, these 2 peptides will be recognized by a receptor that we call as NARC receptor. So once these 2 peptides are recognized and still unknown molecules is produced in the leaf and transported through the phloem to the root, and it will stop the nodule formation. So the only component downstream of the NARC, or downstream of the perception of the RIG1 or RIG2 peptides, is this gene that we call too much love. So too much loss encodes via E3 ligase ubiquitin. And we believe that too much loss is targeting this master regulator, which is NIM. And once it's targeted this and transcription factor, the nodule formation will be stopped. So this is, in summary, the autoregulation of nodulation pathway. So if one of those components fail, like, for example, if the plant doesn't produce these two peptides, or the NAR receptor has some defect, so we will have this phenotype that we call supernodulation phenotype. And we can say that if the plant has more nodules, it will be able to fix more nitrogen and it will grow faster and happy. But it's not the case, because we know that if the plants produce more nodules, it will require it to invest more energy in form of ATP. So the plans to avoid this unnecessary ATP investment, so the legumes activate the AON pathway, just to have a balance between the growth and the nitrogen fixation process by itself. So plants, and particularly legumes, and also have another issue to deal in natural condition, which is the phosphate availability. And we know that phosphate is very important to make different biomolecules, including nucleic acid, including phospholipids of the plasma membrane, but also it is very important to decode any signal molecules, either coming from inside of the cell or outside of the cells. And in the case of the legume rhizobia symbiosis is not the exception, because in this slide, I can show you that to activate many of the symbiotic response to host this rhizobia, plants need to activate different phosphorylation and dephosphorylation cascades. If those phosphorylation and dephosphorylation cascade fails, this symbiosis is not established, or it will have some defects like few nodules or nodules that are not colonized by rhizobia. So phosphate is also important, and legumes need to deal with both nutritional stress, which is nitrogen availability and phosphate availability. As you know, phosphate is playing a major role in the agriculture, because it regulates many aspects of the plant development and plant production. So as you can see here, in the case of legumes, it is very important for the biotic nitrogen fixation, but also for seed production, even for any of these development process. So phosphorus is also very important, and that's why it's considered as the second most important nutrient for plants, or even for any organism in this planet. So the issue with phosphate is we have, plants in the soil have a lot of phosphorus, but the plants will only take phosphorus in form of phosphate. And phosphate is not really abundant in the soil. So basically in the soil, we can have around 10 micromolar of phosphate. But the issue with phosphate, it is a molecule that interacts rapidly with other cations. Here we have two scenarios in acid soils where aluminum is very abundant, so it can interact with phosphate and it will become inaccessible for plants, or insoluble precipitates. A similar scenario in the basic soil, where we have a lot of calcium and it will interact with phosphate, and making this complex, phosphate is not available for plants. So as you can see, phosphate is also a major issue for plants. And this problem is not only for Mexico, it's an international problem, because we have many acid or basic soil around the world, and basically most of the plants are dealing with the phosphate availability. So here you can see the effects of phosphate deficiency. Here we have plants growing on phosphate deficiency condition, comparing with plants growing on optimal phosphate condition. You can see that the growth is severely affected. And also one of the response to phosphate deficiency is the modification of the root architecture. So this is just to find different or potential part of the soil that can contain phosphate for the plants. So some of these responses to phosphate deficiency are regulated by a single transcription factor that we call phosphate starvation response 1. And basically, we call this as a master regulator of the plant response to phosphate deficiency, because this transcription factor is in charge to coordinate the process to take phosphate from the soil, to translocate the phosphate from the root to the other parts of the plant, but also to coordinate and regulate the phosphate homeostasis. And also, there is more evidence indicating that this transcription factor also can play a role in other adaptation of the plants. So this transcription factor that we call PHR1 is present in many land plants, including basal plants. In the particular case of legumes, so one of the effects of phosphate deficiency is the reduction of the number of nodules. So here you can see plants growing in optimal phosphate condition, and you can see the number of nodules. Here you can see also in this plot, and comparing with plants growing under low phosphate condition that form few nodules, and many of those nodules are not infected by rhizobia. So the other effect is the reduction in the nitrogen fixation. Here we have the nitrogenous activity of nodules growing under low phosphate condition and comparing with phosphate growing under optimal phosphate condition. You can see that the reduction is more than 50% in comparison with the control plants. So since this is a very important effect on legumes, there are many studies demonstrating how legumes, or particularly how nodules, respond and adapt to phosphate deficiency condition. Now we know that which transcription factor are involved in nodules, what phosphate transporters are involved in nodules, and basically we know how nodules obtain phosphate from the rest of the plant. So with this, we can say that we know a lot about how nodules adapt to phosphate deficiency. But there are many questions related to this, to this nutritional stress. For instance, how does phosphate deficiency reduce the number of nodules? Is phosphate deficiency affecting the rhizobium infection process? It is possible that the autoregulation of nodulation pathway is involved in regulating the number of nodules when a legume is growing under phosphate deficiency condition. Or perhaps there are other regulators that are participating under these particular nutritional conditions. So to answer this question, so first, we try to answer whether phosphate deficiency affects the communication between rhizobia and legumes, and particularly, we were more interested to see if phosphate deficiency affects the rhizobial infection process or not. So here we have a summary of this rhizobial infection process. I will not go in detail on all the genes involved in this process, but I will say that if the root hair is not correlated, like in this picture, this symbiosis can fail because legumes are not able to entrap any of those rhizobia and internalize it into the plant cells. So to answer this question, so that we did is we generate rhizobium tropism, which is the symbiont of common bean that express a marker gene, in this case, the GOS gene or UA. So that we did is we inoculate our plants growing on the optimal phosphate condition or low phosphate condition, and we can see here how rhizobium is attached to the root and how it's attached also in a low phosphate root. So you can see that the attached process, which is very important for this symbiosis, is reduced under low phosphate condition. But it's not the only aspect that is affected by low phosphate condition. We also observe that the root hair curling process is also affected. Here you can see how under optimal phosphate condition, rhizobia is triggering this root hair deformation. So here you can see many of those deformation events in those roots, in comparison that roots growing under low phosphate condition that have few of them. But also, there are many aberrant root hair deformation that, based on the literature, are not efficient to entrap rhizobia, and it will affect the establishment of this symbiosis. So with this data, we can say that this rhizobial infection process is affected by phosphate deficiency, particularly the root hair deformation process, and probably also the colonization of the nodules. Just to have more evidence that low phosphate affect the rhizobial infection process, we analyzed the expression of 3 marker genes that are related to the infection process, which is this transcription factor NSP2, NIN, and this flotilin gene that is involved also in the infection process. So in summary, you can see that when plants are growing under low phosphate condition, the expression of NSP2 and NIN are either not induced or has a delayed response. In the case of FLOT2, it has basically no response to rhizobia. So having this data support our hypothesis that low phosphate affect the rhizobial infection process. So to have a better idea what other genes are affected under low phosphate condition when a legume is interacting with a rhizobia, we perform an RNA sequencing analysis, and we found different genes, and I will not go in detail what we found in this transcriptional analysis, but I want to bring your attention in some of those genes. So we know that auxins and cytokinins are very important to activate many of those genes involved in the rhizobial infection process. So what we found is that genes involved in either in the biosynthesis or perception of these positive regulators, auxin or cytokinins, are not activated in plants growing under low phosphate condition. In contrast, what we found is genes related to the biosynthesis and perception of other hormones like ethylene, gibberellic acid, jasmonic acid, which are very important for plants to properly adapt to phosphate deficient condition, particularly in an abiotic context, are upregulated. But we know that ethylene, jasmonic acid, and gibberellic acid are negative regulator of this symbiosis. Basically, if plants have a high concentration of these 3 hormones, this symbiosis will fail. So with this data, we came with this model that we propose that low phosphate deficiency conditions will affect the rhizobial infection process, making this aberrant root hair deformation that probably will lead to the formation of few of those nodules, but also hormones, particularly ethylene, jasmonic acid, and ABA, are playing also a role in regulating the number of nodules. So to answer the question whether the autoregulation of nodulation signaling pathway is involved in regulating the number of nodules under phosphate deficiency conditions, we also perform different experiments. So first, we check the expression of the RIG1 and RIG2 peptides, and you can see that even during the first hour of interaction with rhizobia in plants growing under low phosphate condition, we have a high increase in the expression of RIG1, but also we got some expression in RIG2, particularly after 48 hours of interaction with rhizobia. But if we look, these panels show only fold change, but if we look only in expression levels, we can see that the expression of RIG1 are always higher, not even in the absence of rhizobia. So this observation indicates or suggests that phosphate deficiency by itself is able to activate the expression of these clepeptides that for several years, we believe that those peptides are only activated by rhizobia. So to confirm that, what we did is we checked the expression of RIG1 and RIG2 in the absence of rhizobia, but under optimal or low phosphate condition. And we confirmed our previous observation that low phosphate is able to activate the expression of these two peptides, or the genes that encode for these two peptides, sorry. Interestingly, when we put back our plants from low phosphate condition to optimal phosphate condition, we observe a significant reduction of this expression, basically about the same level of plants growing under optimal phosphate condition. So just to confirm that this is a particular case for phosphate deficiency condition, we check the expression in other nutritional stresses, including nitrogen deficiency and iron deficiency. And basically, we don't see any induction of the expression of these two genes. So with this data, we conclude that phosphate deficiency activates two members of the AON pathway. To have better idea and confirm that phosphate deficiency activates the expression of these RIG1 and RIG2 genes, we clone the promoter region, particularly of RIG1. We got some technical issue to clone the promoter region of RIG2. And basically, you can see that where RIG1 is expressed in the absence of rhizobia, but in the growing under low phosphate condition. And basically, we confirm our observation that low phosphate activates the expression of RIG1. And it is very interesting because we can see that the promoter activity is basically close to the vascular tissue, in particular in the pericycle cells, which is very similar that it is observed in other plants inoculated with rhizobia. This is the promoter activity of RIG1 in Lotus japonicus in the presence of rhizobia. So you can see that basically low phosphate condition activates in the right cell types of these peptides. So we check what cis elements are present in the promoter region of RIG1 and RIG2, and we found that these genes has at least one PHR1 binding sites or P1BS cis regulatory elements. That this is very characteristic in those genes that are involved in phosphate deficiency response. And these genes are regulated by PHR1. So the presence of these cis elements suggests that RIG1 and RIG2 probably are regulated by this master regulator. So just to remind you that PHR1 is the master regulator in any plants to properly adapt to phosphate deficiency. So basically PHR1 binds to the P1BS cis elements to activate the expression of genes involved in phosphate homeostasis, phosphate transport, phosphate translocation, and in other responses that are required to adapt to phosphate deficiency. And this is interesting because so far there are no evidence indicated that PHR1 are required to establish symbiosis with rhizobia, or at least to control. So to have more clues about PHR1, so we clone the promoter of this transcription factor, and we generate transgenic roots that express the construct of the promoter of PHR1 fused to the reporter gene GOS. And as you can see, we can see a nice activity of this promoter in the root, but also in the nodules. So this is something very similar observed in soybean nodules. In this study, they demonstrate that PHR1 are also expressed in nodules, but in this paper, they suggest that PHR1 is required to activate this phosphate transporter to keep providing phosphate to the nodules and maintain the nitrogen fixation. In this paper, there is no information about the role of PHR1 in the controlling number of nodules. But we generate some evidence about that. Just to demonstrate that PHR1
Thank you.
Just to demonstrate that PHR1 is relevant for this symbiosis, we silence this transition factor, and we observe that when PHR1 is silenced, RIG1 and RIG2 is not expressed. So with this data, we demonstrate that PHR1 is controlling part of the autoregulation of nodulation signaling pathway. And basically, we found about the same for the other regulator, which is NIN and too much love. I will go faster. That basically, NIN is also playing a role, and PHR1 is controlling the expression of this component, NIN, too much love, RIG1 and RIG2, and with this we conclude that the autoregulation of nodulation pathway is required to regulate the number of nodules under low phosphate conditions. And just to, this is the last slide, just to conclude, we did some physiological analysis just to demonstrate that the receptor is also involved in this process. So we have mutants that the NARC receptor is mutated, and basically with NARC receptor is mutated, you cannot see the reduction of the number of nodules even in low phosphate conditions. So with this is the model that we are proposing, that together PHR1 and NIN are playing a role in controlling the number of nodules under low phosphate condition. And we believe that PHR1 is not only required for the response in an abiotic context, we believe that is required to communicate with other microorganisms, including mycorrhiza. And just 2 weeks back, a paper in Cell demonstrated that PHR1 is a master regulator to establish this symbiosis under low phosphate conditions. And with that, I would like to thank the people in my lab, my collaborators, and also the agency that provide funding for my research. Thank you very much.
Thank you very much, Osvaldo. A very, very nice talk. I'm very sorry, but the time is up, so we have to stick with the schedule. So please, all the participants, when you have questions, please write them in the chat, and I'm sure Osvaldo would like to have a look at them. Thank you very much for the talk. And then we go back to Dan. Go ahead.
Our next speaker is Laila Partida Martinez from Irapuato, Sinvestav, and her talk is going to be deciphering the secrets of the plant microbiome in drylands. Take it away, Laila.
Thank you, Dan, and thank you very much to this for the invitation to this great meeting. Are you hearing me very well?
Yes, we hear you perfectly.
Fine? Okay, thank you very much. So I'm really happy to be here today and to be able to share with you some of our results about the plant microbiome in dryland. So we have seen now, well, all the different kind of symbiosis, and I'm really very keen to explain you. Well, first of all, thank you for the invitation. I'm really excited. And I hope, well, I also thank my lab, who I will be presenting most of the work is actually from 3 former PhD students that you can see in this slide, Citlali Fonseca Garcia, Damaris Desgarenes, David Camarena Pozos, Victor Flores Núñez, and also Raúl Alcalde, and now also Jose Daniel Chavez. So they are the main the drivers of this project, and I'm very happy to say also that we have had the funding from the GTI, from CONACYT, from the Senckenberg Institute in Germany, and also from a lot of the producers in Amatitan, and also here in Guanajuato, and even in Oaxaca. So I will tell you today about something that we have seen in this conference already, that we are very worried about the climate change. And of course, we're worried about the climate change because they will affect all the different biomes of the earth in different ways. And one of the most important for me, and as you can see in this map, is actually the drylands. These drylands represent about 41% of the world's surface area. And the characteristic of these drylands, biomes, is actually the lack of water. No? So precipitation that comes to these areas actually very likely evaporates. And by this aridity index, we can actually consider 4 different types of aridity biomes, the hyper-arid, semi-arid, arid, and dry subhumid. And as you can see from the map, in North America, especially in the case of Mexico, 50% of our territory are considered dry or arid or semi-arid. So this is very important because in the challenge of climate change, we will see that the vulnerability of these areas is really increasing. So we will see scattered periods of rain and, of course, increased droughts and even floods, as we have seen before, also with the Julie stock and many others. So this is something that will promote, it's really challenging how we will make agriculture in these drylands and in order to survive and to maintain our ecosystems and also our livings as humans. So something that we have learned constantly is actually that plants, well, we can learn a lot from nature. Nature has already had many millions of years of evolution, and they have created different solutions for the problems that we face. So actually plants, as you can see in this slide, plants from with these succulent bodies that you will all recognize as cacti and also agaves, they already develop many physiological and biochemical innovations to adapt to these dry environments. But up to now, of course, we didn't know so much about the role of microbes within these plants. For example, one characteristic that is very important is actually the water-efficient photosynthesis, that is the crassulacean acid metabolism. And by this, but using this way of photosynthesis, these plants really can save a lot of water during the day when it's very hot outside, close the stomata and make all the CO2 transport during the night, and then also the fixation. So this, but until now, we didn't know so much about the role of microbes. And probably you know that the Cactaceae family is an American plant family. So it originated in this continent, and it has a lot of different members, like 2,000 members that you can, with the highest diversity in Mexico, and they span from Canada to the south of Patagonia, well, to Argentina. And for Mexico, it's really one of our most important symbols, even for the flag. We have the Tenochtitlan that was really when an nopal, an eagle stepped in a nopal. So these are very important plant family. And of course, the agaves, we cannot say something less. The agaves have been used in many different ways since pre-Hispanic times. And of course, most of the people know that are not from Mexico, or tequilas and mezcals. And they are really very evaluated distilled beverages. And as you can see, well, the market for these beverages is really increasing a lot, especially in the last years. And so all these beverages are derived from different species of agave. I have to mention here that agave have diversified and originated in Mexico. So most of these 200 different species of agave sensulato are actually native to these arid regions of our country. And of course, they have been also important, not always for the beverages, and so they are also seen as a source of new biofuels. So this is something that also help us to get some funding from the GTI. So the research question that we have addressed in my lab since I started working at Simvestab in Irapuato are, you can imagine like this. Now, you see very commonly that agaves and cacti share the same environment. So you see this picture when they are really sympatric. And the questions that we have asked is which kind of microbes are associated with these plants, how they are selected, how the communities are built. And very important, but because we think now we cannot understand plants, plant biology without the interactors like microbes. What do these microbes do for their host? And so with these questions, we started, I will show you our first experimental design that has been very important for all the different developments later on. So we selected 5 different species of agaves and cacti. You can see the pictures here. In the north of California, we have agave deserti. in Guanajuato and Jalisco, we could screen and investigate the communities associated with agave tequilana, the species that is used for cultivation and that for production of tequila. And in the state of Guanajuato, where we live, we have 2 different sites where 3 of these sympatric species live. This is agave salmiana, that is also used for mezcal and in an artisanal way, Myrtillocactus geometricans, columnar cacti, and Opuntia robusta, and opal, right? So one of the approaches that we follow was actually to compare not only different sites, but also the abiotic factor that is very strong in these plants, no? The dry season and the rainy season. Of course, the patterns of rain are different in the different sites that we sampled. And one of the strengths of our survey was also to look for all the microbial communities, not only in the rhizosphere or the roots that are supposed to be the most, of course, the most studied and also the most important for the communication with the plants and the microbes, but we have also investigated the endosphere in the aerial part and the surface, so the phyllosphere, the roots, and of course the soil to analyze and to compare to the plant-associated communities. And we have combined culture-independent and culture-dependent methods to really know what these microbes are and what are they doing for their plants. So with this idea, well, I will show you very briefly all our main results. Many of these papers are already available, so you can go in more detail if you feel like. And something that we really were amazed is actually that we have a very high diversity, both from bacteria and archaea and fungi. You can see that the numbers are 4,000 and 4,000 fungi. So we have like 8,000 different taxa associated with these plants. And from this, of course, because of the time, I will go a little bit quickly. But to show you, we went wondering how these communities are assembled. And you can see this is when you compare each of these different samples and to see how many different microbes are associated. So when they are very close, it means that they are sharing the same microbes and in the same abundance, more or less. And if you see these points very separated, it's because they are more different and they have different composition. So what I'm showing you here is actually how the prokaryotic communities in agave and cacti look like. And you can recognize something, this pattern, that all the soils, independently of in California, 2,000 kilometers away from central Mexico, all the soils were clustering together. And then along we move in the coordinate 1, so in the x-axis, we see how the different communities separate depending on the niche of the plant that they are occupying. So you will see the rhizosphere in red, the phyllosphere in blue, and then the root endosphere and the leaf endosphere in green. So this was the most important pattern that we identified in the bacterial, and I would have to say the archaeas are very few in these plant-associated communities. So mostly we can speak about bacteria, and these are the most important factors. And secondly, it was the host. But this picture looks quite different. It looks using the same DNA from all the plants that we sampled in the field, but when we use the fungal marker, no, the ITS2. And as you can see, as we work, for example, in the river, not separating U.S. and Mexico, we see all the Mexican populations in the left and the one from California in the right. And the other clear division of these communities is actually what is inside the plant. So you see the endosphere in the upper part of the coordinate 2, and all the soil and epiphytic fungal communities clustered in the down part. So we learn from this analysis that fungal communities have a biogeographic pattern. So this is important. And something that we were also keen in investigating was actually how diversity looked like. If you remember what we have now in this graph, we have three different species, and the one that is cultivated is only agave tequilana. And you may know that this species is a monoculture and is not allowed to flourish, no, and to get genetic mix. So I'm highlighting here two important aspects of this graph. So first, we saw like the diversity using the Shannon index is very high in the soil, in the root zones, and then it lowers in the epiphytic compartments, and it's even less in the endophytes, in the endophytic compartments, something that was expected. This is very clear in the prokaryotes and also to some extent in the fungi. But what is intriguing from this slide is also that the phyllosphere, that is the communities above in the surface of the leaves on the plants and stems, they are really so high as the rhizosphere. So that means that there's a lot of diversity, bacterial diversity, but something that we also recognize from this picture is actually that agave tequilana that has an agronomical management has a reduced diversity. And of course, this could prompt us to think that this species is in risk because of our management. And it's something that we will address later. And the other thing, despite of these different patterns of diversity and assembly, we could identify that agaves and cacti share a core microbiome. So I'm showing here the 5 different species and how in different compartments you see more or less the same bacterial and fungal taxa. Of course, in different proportions sometimes, but they do represent a big part of the community, not only in numbers, but also in their relative abundance. So we think that by understanding the plant microbe, these plant microbe relationships and what do they do, we will be able to have general rules for not only for agaves and cacti, but probably also for other plants, so that we could transfer some of this knowledge to tomato, maize, or whatever other plant you like. So this is our hope. So from now, so this was the first part that was studying diversity and to know actually who is there, no, and how they are arranged. So now the difficult part with such a complex community, imagine 8,000 different taxa. So we have to move on how to create different strategies to study their function. So we have made some progress in 3 different areas. One is the microbial volatiles, how chemical molecules can be used for communication between microbes and plants. We have also used the approach of using synthetic communities, so that is reduced complexity communities that can resemble the functions that we presume are working on the plants. And of course, we also have some insights into fungal bacterial interactions, how they are very important, the interactions between the microbes, also within the microbiome. But for the sake of this presentation, I will focus on the first one, that we have already more advances. So volatiles, as we have seen now, even the Hailin talk that is very, very beautiful to how she has shown the communication at the RNA level, right? So something that has been also present in our minds in ecology and in plant science and insect science is actually that molecules, compounds can also have be infochemicals. So this one has been from the 70s already, 60s already, that some molecules can be used to attract your partner or to see where is your prey. No? And in plants, this has been, plants produce 20% of the carbon that they fix are emitted to the atmosphere as volatiles. And you know, probably like in these pictures that I have, that some volatiles are meant to attract pollinators, some of them to defense, not to, that herbivores cannot eat them. And of course, parasitic plant like Cuscuta can distinguish even between the volatiles from tomato than from wheat and go to the tomato plant because he likes, or this plant likes tomato more than wheat. So something that was really very new, I mean, at the beginning of the 2000 was this paper that actually showed for the first time that bacterial volatiles, so bacteria, all living organisms emit volatiles, but bacteria volatiles were first screened with Arabidopsis, and by Roy and collaborators, they showed with different, using even E. coli DH5 that you can see in this one, do not produce, these volatiles do not produce growth in plants, but some others can do, right? So this has been very important to try to understand not only, well, microbes and plants have many, many different ways to interact with each other. And of course, one of them would be this chemical communication. So now that we knew that our agave and cacti microbiome is complex, it has diversity, and we know the patterns, we were also interested in investigating if there are some chemical compounds that these bacteria and fungi produce for influencing plant growth. And I show you here the screening that we have made using 40 bacterial strains and 22 fungal ones. And they are from the core, the ones that are shared between different species and sometimes even in different plant compartments. So the important thing, just to test the proof of concept, if they have really a potential, you can see in this divided Petri dish, so there is no physical contact, only through the atmosphere, we can see how the phenotype of Arabidopsis is really being affected by the emission of volatiles from the bacteria or fungi that you can see the different names here, that they come all from the agave and cacti microbiome. So this one was really inspiring us to really go further and characterize in more detail the plant phenotype. And we use Arabidopsis, of course, instead of using agaves because they are very much easier and shorter time to respond. And of course, what we can learn also from Arabidopsis, potentially we wanted to test if they will occur and see the same effects in agave. So of course, you can measure all these different patterns, the length, the shoots, and the chlorophyll. So this will be the summary of this. All these different measurements. And so David actually was very patient to really phenotype many different plants in all these parameters. And we will learn that using the bacterial volatiles, that the main lateral root was affected. You can see in green, we have like 3 different responses. In green, the very positive ones, in yellow, the slightly positive, and in red, the ones that inhibit even growth. And of course, you can see that there's no phylogenetic position of the microbe or the bacteria that tells you which kind of response you will have in the plant. And when you see the fungal screening, the difference was actually to see that we have more difference in root architecture and also in plant biomass. So you see differences very green, well, 5 times more plant biomass than the controls. And interestingly, some fungi didn't produce any effect as if they were not there, right? Like very neutral. So the ones that are in white. But this one was not telling us what kind of volatiles were produced. So for knowing which is the diversity of the compounds emitted by this set of microbes, we decided to use solid phase microextraction and then gas chromatography and mass spectrometry. And of course, this one is a batch process that you can expose your fiber to this Petri dish when you have the plants and the fungi or the bacteria, or either alone, to really check which kind of volatiles were produced during the interaction, and also when they are alone. So what we have learned from these experiments is that 40 different compounds, especially esters, alcohols, and aliphatic compounds, and also ones with sulfur, are produced by bacteria. You can see that in blue, there are many that are shared and some that are specific. But the most important and more diverse are the esters and alcohols. In the case of fungi, even if we have less and a reduced number of species, we have really a wider chemical diversity. And something that attracted us attention was the terpenes. Terpenes are very important, not only for plant development, they have different functions, and the fungal here are also producing a wide diversity of terpenes. But of course, this altogether, bacteria and fungi are producing almost 120 different compounds that we could detect in our analysis. How can we select the ones that are really promoting growth, that we can study in deep and in more detail, and also to try to answer and to use them for agriculture? So David screened many of them, I think around 15 different compounds that were available that we could buy from the different suppliers. And he tested how much in a dose response assay in different doses, how the phenotype of the Arabidopsis was changing. And you can see that we identify now 8 different compounds, esters, alcohols, one with sulfur, and a terpene, a sesquiterpene, and that are mentioned here. And also you can see the different structures. And something that is very important to keep in mind in this chemical communication is that these responses are dependent on the doses. So you can see here in the pictures below that when you have 0, you see a normal growth, 10 or 50, you see really a promotion of growth. But of course, if you use a lot of more of this compound PA, then there is some inhibition. So this change says already what I say, those response assays. But of course, all organisms, bacteria and fungi that we screen produce not only one compound, but several compounds. So we were interested in testing if we synergize these compounds, if we could have even better effects. And this is in fact true. So when we combine different volatiles, we see that even higher effects, as you can see here in terms of some of the parameters. Something that I was very surprised, because some of the experiments I did it myself when I was in the sabbatical year in Germany, is that some of these compounds were able even to increase the number of seeds of plants, like Nicotiana attenuata in the left, and also in Arabidopsis. So these small compounds have a really strong effect on plants. And of course, we were wondering if what we have learned in these model plants could be applicable to agaves. And this is also the case. So you can see like the doses that we identify, and this is David, who did all this work. And you can see how these doses, and in agave, we see the same effects, and we see growth promotion. So this was very important. And we continue monitoring these plants after the first treatment of volatiles. And these are almost the same plants that I showed you before, but 8 months later, without any treatment, additional treatment, and they were already, of course, in soil. And you can see that the effects on the development are long-lasting. So we are very enthusiastic because we want to help to make this mezcal production also sustainable. We don't want to lose these beautiful landscapes, and also we don't want to lose the opportunity to taste some mezcal later in the future, right? So that's why we decided to test as well. You probably know that many of the mezcal, the species of agave that are used for mezcal are wild, no? They don't have populations or cultivation. So we need to reduce this, to try to help to address this. And you can see from this slide, the time, 7 years, 11 years. So we need a lot of that. So that's why we have focused on the time, because they are very long life plants. And after 1 year of experiment, so we applied in November 2019 all the different, some volatiles, the best that we have found. And after 1 year, we have measured the plants again in the field, both in agave tequilana and in the other mezcal species. And we have seen that the diameter of the rosette, the number of leaves, increased. And in the case of agave tequilana, we were even able to look for the number of hijuelos. And this was increased in this 1 year. So we think that this application, is really something feasible for the future of agaves. And we are very happy to provide this tool for the producers and to try to make this production sustainable. In the last 1 minute that I will use for this talk, I want to say that we have also made some steps to to study volatiles in a dynamic way. And this is thanks to the collaboration with Robert Winkler and our student Raul, who really developed a new system to track volatiles online, no? That we really know when they are emitted, what happened with the plant, and if the volatile, the plant reacts also with volatiles. And we're also trying to investigate how these volatiles are actually produced. What is the biosynthesis behind? So we have genomes and all these things that we use. And just very briefly before we finish, is actually this is how this mobeams looks like, and this is that allows us to measure in milliseconds the production of and the consumption of volatiles, and we can see that the dynamic in these graphs changes when the plants are present. And also, of course, we can also, using some other mass specs, spectrometry devices that the lab of Robert have developed. We can see also where these volatiles goes. And here, for example, this volatile goes to the stem and not to the leaves and not to the roots. So this is also an opportunity for us to understand how plants sense different volatiles from external origin. And of course, we would like to know the whole communication. So this tool will be very useful for that. With that, I already said most of this. So I'm very happy to take any questions if you may have one. Thank you for your attention.
Thank you. Fascinating talk, Laila. Please raise your hand if you have a question. Oh, we have many questions arising. Ulises. Oh.
Better Alejandra. I will ask Laila later.
Oh, okay. Alejandra.
Ladies first.
Hi, Laila. How are you?
Hi. Thank you, Alejandra. Hi.
Nice talk. I'm just wondering whether some of these volatile compounds may have a positive function for growth in some plant species, but the contrary in other plant species. Have you seen something like that?
Yeah, of course, this is to some extent specific to the plant. I mean, we have seen most of the positive effects in Arabidopsis. We could translate them to agave plants. And we have seen that depending on the species, we have like differences in the doses that they react to. For example, to agave tequilana that is a little bit thinner, is like 5 micromolar, while to agave salmiana that is a little bit thicker, it could be 50 micromolar. But of course, something that we want to learn is why they react to these differences in a different way. Right. Of course, when we know that, we will be able to design in more detail what could happen. And of course, some of them are probably negative, inhibiting the growth, no? Especially, well, even as I showed with one of the alcohols that I showed, they can inhibit, if this is in a very high doses, it inhibits growth. In a adequate doses, it promotes growth. So of course, it has also the potential to be used not for promoting growth, but also to inhibit the growth of plants that you don't want to have.
Exactly.
But it's something that we have not really, and something that we have to learn why as well, right? That we can really develop this and design these solutions for the agriculture.
Exactly. Thank you.
Great talk. We're at time and we have some more questions, but if you can please write them in the chat. I'm sure Laila can answer them there. Thank you.
Of course. Thank you. Thank you.
Thank you very much. So we go to the next talk. We have Professor Georgina Hernandez from the Center for Genomic Sciences of the UNAM. And her talk will be about deciphering novel common bean regulators for the rhizobium nitrogen fixation symbiosis. So welcome, Georgina. Please, if you can share your screen.
Do you see my presentation?
It's coming. Yeah, see your screen.
Okay.
There it goes. Yes,
okay,
go ahead. Thank you.
Okay, thank you very much for the invitation. And I really would like to congratulate and to thank a lot the organizers, Felipe and other organizing committee for this meeting. It is obviously a new form of having a meeting, this virtual form, and it has been great so far. And it was a great effort for you organizers. So thank you very much for this very nice Congress for all the community. I will present my work, as Stefan already mentioned, and So my lab works with legumes, with legume plants, and I would like to present my work that is it's the same theme that we already heard from Osvaldo Valdez. So really, he did a great introduction of this theme, and I will just briefly say some introduction because Osvaldo already speak about this. As the legumes are a very big family, around 700 genera and 18,000 species, and they they form 1/3 of the primary crop production in the world. And as we have already heard, and we have seen this same cartoon, one of the very important attributes of legumes is that they establish symbiosis with mycorrhiza and with nitrogen-fixing rhizobia. And this will allow them for better nutrient allocation. And as well as Osvaldo, my talk will be about the nitrogen fixation in these legume plants. This is an introduction to the theme, and as already say, Osvaldo already introduced it. So I really just mention few details about this, and that already has been shown. So in the top of the slide, in the left, we can see that the interchange of different biochemical signals between the plant and the rhizobium that will secrete these node factors that are perceived by specific receptors in the root hairs of the legume plants. The specific receptors are seen in this other scheme in the right-hand side. And then the infection of the rhizobia begins, forming these infection threads and infection threads within the formation of the infection of the root hair and the rhizobium here, and it penetrates. And here is the signal transduction pathways that are going around. And Osvaldo also mentioned this calcium spiking in the nodule that is a signal that is decoded, and the transcription of different genes start. Regarding the infection, after the infection thread comes to these other layers of the root, the cell, there are several signals that are perceived, and the nodule primordia begins, and the nodule, the terminate nodules in the case of Phaseolus is formed. And in these several stages and in others, there is a very important actor that Osvaldo mentioned, the NIN transcription factor, or nodule inception transcription factor, that is expressed in the very early stages in the root hair, and also in the pericycle and cortical cells of the root, and that induces this nodular organogenesis. When the nodule is formed, the bacteria will take the nitrogen from the atmosphere and will reduce it to ammonia and give it to the plant that will synthesize organic nitrate compounds that will be used as nitrogen source, and the plant will give the bacteria carbon from the photosynthesis to have the required energy for the nitrogenous complex that will reduce the nitrogen. So as Osvaldo mentioned, this is a very costly process, because lots of ATP are needed to reduce the atmospheric nitrogen. I work especially with common bean. My group has been working with this crop, for this legume, for several years. This is a very important legume, the most important grain legume for human consumption, not only in Mexico, but in other countries from America and Africa. This common bean, Phaseolus vulgaris, originated in Mesoamerica, specifically in Mexico, and it diversified in two genetic groups in Mesoamerican region and in the Andean region. These two were differently grown and
domesticated, and we have genotypes from representing these different genetic pools. In this work, I used the Mesoamerican genotype BAD93, that was the first Mesoamerican genotype sequenced. The original microsymbiont of Phaseolus vulgaris is Rhizobium etli, also originated in Mesoamerica, and these two microsymbionts have co-evolved for thousands of years. Despite the complex the several regulators that we already know that participate in the common bean plant or in the legume plant Rhizobium symbiosis, we know that this is a very complex process that has to be very precisely regulated, and it is many of other regulators that are important as are yet unknown. So the theme of the research in my group is specifically this one. We are trying to decipher novel common gene regulators that are relevant for the control of symbiotic nitrogen fixation. And we have focused in two different regulatory molecules that are very important global regulators, the macroRNAs and the transcription factors. And we have used reverse genetic approach to try to demonstrate the relevant role in the symbiosis. Here, I listed some of the previous work from my group that have allowed us to demonstrate this novel common bean regulator that includes microRNAs like microRNA 398, 172, 319. And so several of these microRNAs have as target gene a transcription factor. So we investigated the role of these nodes. For example, this one, the microRNA 172 and APT2, a transcription factor that is essential for the regulation of nodulation and symbiotic nitrogen fixation. This other one and this other one is still research in progress. And I will not talk about these examples because in fact I have talked about this in previous meetings. So I will focus in the new, I cannot, sorry. In this other work that was recently published, and we investigated the role of the AGL or MATS box transcription factors from Phaseolus vulgaris and showed that it is a very important regulator in the Rhizobia symbiosis. This work was done mainly by Litsi Aria, a PhD student from my group, and we collaborate with Osvaldo Valdez Lopez, that you already heard, Lourdes Girard, a colleague from my center, and other colleagues from my center and from Osvaldo's group. So, as you may remember, the MATS transcription factor family is widespread in different organisms, and the name that we use, the MATS name, comes from the first transcription factor of this family discovered in different organisms like yeasts, plants, and humans. In Arabidopsis, this first gene described from this family was called agamus. So this family in plants is also called agamus-like protein transcription factors, and I will refer basically to this name, to AGL, a transcription factor family. As you probably remember, this family in Arabidopsis constitute very important homeotic genes that determine the identity of each floral organ, thus functioning in flower development and flower transition. And this is a regulatory model that you might remember that comes from different years of research of these transcription factors in the Arabidopsis flower development, and it is very well known. However, besides having important roles in flower development, these transcription factors are expressed in many other plant organs. And of course, we know about functions in these of these genes in these other organs, okay? So I don't know why I cannot, okay. So there was a recent review by Alvarez-Buya et al. That summarizes the information about MATS transcription factors that regulate Arabidopsis root developments. Here are depicted some of these factors that are known to regulate, for example, primary root, lateral roots, root hairs, and that are known to be expressed in different root cells of Arabidopsis, and some the functions of some of these are known. And in this review, some information is summarized about legumes, about this transcription factor in legumes, but this information is rather few because we really don't know their function specifically in the roots of legumes and if they are important for symbiosis. So this is something that interested us for studying, okay? So then, I don't know, but I have problems passing the slides, sorry. So another thing that was important for us when we began this project is to know that during evolution, the legumes have recruited regulatory elements from different developmental programs to participate in the coordination of root nodule development. And here are just 3 examples of these. One are the LBD genes that in Arabidopsis participate in lateral root development, and it was recently demonstrated in the 2 model legumes, Medicago and Lotus, that they activate overlapping lateral root and nodule developmental programs. Another example are these NBCL genes that are orthologs for Arabidopsis bob genes that participate in flower developmental programs, and in nodules, they participate in repressing root identity and all symbiotic organs. So with these kinds of examples, we ask the question, do common bean AGL transcription factor orthologs orthologs of Arabidopsis AGLs that regulate root development, regulate root nodule development? For this work, Liziara first did the phylogeny analysis of mad proteins, including Arabidopsis thaliana, Phaseolus vulgaris, and these other 3 legumes, soybean, Medicago, and Lotus, with different number of proteins and genes present in these legumes for this transcription factor family. And here in this root, in this tree, we highlighted these transcription factors from Arabidopsis that are known to have important roles in the root. And in all of these clades, we also find legume transcription factors. So we wanted to see how was the expression in the different plant organs of the transcription factors present in these clades. In the top of the clade is always the Arabidopsis gene, known to be expressed in root, and here it is depicted in these brown bars. And in the other genes from the other 4 legume species analyzed also showed very high expression in roots, and in the red bars we can see that in most of the cases they are also highly expressed in nodules. So in a way this supported our hypothesis. And then what we did was to try to investigate more about this family in Phaseolus vulgaris. We have previously published an analysis that there are 16 of the 93 AGL genes from Phaseolus that are expressed in root and nodules. This has been published, and here is the qRT-PCR analysis that indeed, this is from RNA-seq data, and this is from the analysis that also we know that we see that these transcription factors are expressed in fertilized roots, inoculated roots with rhizobia, nodules, and leaves in a different expression level in the different genes. The ones that I highlighted with this star are those that are more highly expressed in roots and nodules, because other ones have really a very low expression level. This is a finer expression study, expression analysis of these 6 genes that present higher expression level in roots and in nodules. We analyzed roots fertilized with nitrate and roots inoculated with rhizobia that eventually around day 10 in Phaseolus vulgaris will form nodules, and we also saw the expression level of each of these genes in these tissues from 6 hours of inoculated roots until 35 senescent nodules. You can see that the expression is, again, different levels, but always higher than the other genes. And we named these genes according to the Arabidopsis orthologs that we found out in our phylogeny. Some of the genes are more highly expressed, for example, in non-inoculated roots, And some of these, like this one, fool-like, is more highly expressed in inoculated roots and in nodules as compared to fertilized roots. So we asked if these genes would have a function in the symbiosis, and for this we used reverse genetic studies that we have used in our work, and we use the agrobacterium transformation, agrobacterium rhizogenous transformation that help us to form transgenic roots and transgenic nodules. These roots, for example, have the constructs of the PB fool-like gene overexpressed, and obviously it has a higher expression in these roots and nodules. And for the gene silencing, we use the RNAi strategy. And here, just to show you that the 200 base pair sequence that we use to do the usual construct for the RNAi approach contains or codes for the MATS domain sequence. This is the characteristic sequence for the transcription factor family that is used to recognize the binding site in the DNA. Therefore, it is very similar among all the genes of the family. Because of this characteristic, perhaps we found that not only the full gene was silenced, but the other genes that in both in roots and in nodules, the other 5 genes that I just mentioned are also silenced in these roots that have this construct. So perhaps we are avoiding the functional redundancy of these genes in roots and nodules. And as you will see, we have a strong phenotypes of these silenced plants with these constructs. First, we saw that the AGLs have positively regulate hairy root formation that is depicted here. These are the roots that are emerging from the sites when we infect with Agrobacterium misogynis, and the amount and the growth speed of these roots is much decreased in the silenced plants as compared to the other ones. These roots have a red fluorescent protein reported, and it's obviously here again the different amount. And these are non-inoculated roots, not in symbiosis, but also we see this decrease in their size and in their weight. Obviously, what happens with symbiosis, that is our main interest. And so we analyze these composite bean plants with the transgenic roots and nodules expressing the 3 constructs that I just mentioned, the silencing, the overexpression, and the control vector from 5 days to 28 days post-inoculation. We first saw that these roots do not have an effect in the I don't know why, do not have an effect in the root hair development. The density of the root hairs are very similar in the 3 constructs. However, we did see an effect in the infection of the rhizobium. This is the same kind of measurement that Osvaldo mentioned, the deformation of the root hairs. And we saw that a very decreased root hair deformation of these effective root deformations in their silenced plants versus a higher formation of these non-effective deformed hairs that will not conclude in an infection thread and in a nodule. In all the graphs that I will show you, first I will put the RNAi construct, then the empty vector, and then the overexpression construct. Then what about other stage? We also measured the expression of early symbiotic genes. Also, Osvaldo referred to some of these, and these 6 early expression genes that are very important for the early stages of the symbiosis, including the main regulator NIN, NSP, etc., are decreased their expression in the silenced plants, and as compared to the control or the overexpression plants that in some cases have an increased expression of these genes. We also saw that in the promoter of some of these genes, we found the CARG boxes that are the sequences that are known to be recognized by the transcription factor in the DNA. Here is depicted this NIN gene and the NSPT2 gene have these CARG boxes in the promoter sequence, indicating that perhaps the AGL are direct transcriptional regulators of these genes, although we did not further demonstrate this feature. We also saw that these genes affect I will take out this instead. This is these are affecting the root development. Here these are symbiotic roots inoculated with rhizobia, and we saw that the root length, the root area, and the root dry weight is diminished in the silence plants as compared to the other constructs. We also asked obviously what is happening with the nodulation, and we saw that indeed the nodules are affected. Here we see that the silence plants make more nodules, especially at this time, 28 days after inoculation, but these have a lower perimeter. There are smaller nodules, as you can see here, and they present a very small infected zone as compared to the other nodules that are bigger and have this very big infected zone, and therefore the nitrogenase activity is also diminished in these silence plants. So this effect that we are looking, Osvaldo also mentioned it, because this nodule phenotype with increased number of small, pale, less effective nodules is shown in those plants that are affected in this regulatory circuit that Osvaldo mentioned, that is called the auto-regulation of nodulation. That is a synthetic process that regulates the number of nodules in the root. Because as he's mentioned, it is not good to form a lot of nodules because the plant will have a negative effect of this. So we asked, are the AGL silence plants altered in components of this AON process? And the answer is yes. Here we see that the RIC gene, that is the one that produces the peptides, the signal that goes to the leaves, is reduced. The expression of the TML gene that is here is a negative regulator that senses the signal coming from the leaves, and a negative regulator of the nodulation is also lower expressed, and also NIN, this master regulator that regulates the RIC gene, is also reduced, as I have already told. Also, the RIC gene and the TML have CARG boxes that may indicate that they are regulated by AGL. So we propose that the AGL affects systemic autoregulation of nodulation process. What are the conclusions of this work and the proposals that we do is that the common bean AGL transcription factors are relevant positive regulators for the rhizobium ethyl symbiosis through their control of root development and architecture, rhizobial infection and the expression of early symbiotic genes, nodule organogenesis, nodule number through the AON process, and nitrogenase activity. We propose that the positive effects of PV-AGL transcription factors in the rhizobia symbiotic process results from its potential interplay with NIN, the master symbiotic transcription factor regulation. Why do we propose this? I showed you already some of this data, but I will show it again. The NIN gene, we detected a CARG box in its promoter region, and we saw that the expression of this gene is very low in silenced plants. In the symbiotic condition with rhizobium ethyl, but I have not shown this result. Also, in non-inoculated and nitrate fertilized roots, we saw a very low expression of this gene. And in this case, there are no other or very basal expression and quantity of other symbiotic regulations. So we can propose that this regulator, the AGL transcription factors, are important for the regulation of this NIN gene. So this is a model that we propose based in these results that I just showed you. Here are the AGLs. They regulate expression of some symbiotic genes that have to do with a formation of infection thread. They also regulate root development. They also regulate nodular organogenesis through NIN and other regulatory factors that are known, and perhaps also a nodal number with the AON process affecting the expression of RIC and TML negative regulators of the nodal numbers. We propose that these transcription factors, AGL in common bean, that are orthologs of Arabidopsis AGLs that regulate root development, have been co-opted by common bean for the regulation of root and nodule development during rhizobia symbiotic nitrogen fixation. And we wonder if this also holds for other legumes. And it will be interesting to analyze if these AGLs are also regulating in other legume symbiosis. So with this, I finish, and I thank all the people from my group that in participate and in this and in other projects that we have, postdoctorals participating in this and other projects. A PhD student, Lizzie Ira, I already mentioned her because she did most of the work that I presented, and technicians Mario Ramirez, Alfonso Lechka, and Sara Fuentes also participated in this work, and we collaborated with Osvaldo Valdez from FES Iztacala and her member of the group, Mariel Isidra Arellano, Lourdes Girard, and Luis Lozano from my center, and Luis Pedro, a previous postdoc in my group. Thank you for your attention.
Thank you very much, Georgina. Wonderful talk. Well, I love Mensebox protein, so very nice to hear that you also work in nodule development. We are almost going to the break. I don't know, we have maybe 1 or 2 minutes for some questions. If someone would like to ask a question, raise your hand in Zoom, please. There's a question from Alejandra, please. Go ahead.
Okay. Hi, Gina. Nice talk. I wonder whether these Metabox transcription factors are responding to hormonal cues in the plant. Do you know that?
No, I don't know this in this case. Yeah, it's a very interesting question that we have also thought about, and we don't know. We think that perhaps an easier way to know the answer to this question would be to use mutants, for example, from Medicago or from Lotus that are affected in cytokinin perception or in cytokinin synthesis, or even also in auxins. So perhaps we will like to try this and answer this question perhaps with these tools that we do not have in Phaseolus. Yeah. But yeah, thank you for your question.
Thanks.
Okay, we have a few questions. Maybe we can take these 3 questions. So please make them short and then we go to the break. So first, Jose Luis Reyes, please.
Hi, Gina. Morning. Nice.
Hi, Pepe.
As you know, I had some other questions pending from the other day. So I'll be brief with the first one, which is, do you think the same AGL factors have been recruited to other symbiotic processes, like mycorrhizal specifically?
We don't know, and also it's a very interesting question, and obviously we would like to know about this. Yeah, but I don't know any report even in the mycorrhiza symbiosis. Yeah.
Okay. Thank you.
Luis Cárdenas, please.
Yeah, actually, Gina, you already responded my questions. Yeah, my question was regarding to that. Have you seen any phenotype in the mycorrhizal? Are you looking for that?
No, we haven't looked to that really. I repeat, it would be very interesting. In my group, we really have not analyzed the mycorrhizal symbiosis, so it would be very nice to collaborate with colleagues that do this. Yeah.
That would be nice to look at.
Yes.
Yeah. Thank you.
You're welcome.
Then we go to the last question of Adriana Garay, please.
Hello, Gina. Nice talk.
Hello.
I already put it in the questions. Do you think that any gene that participates in root growth would affect nodule formation?
No. I don't think every gene would do that. There are some examples, and obviously we don't know perhaps how many would participate, but I don't think it would have to be all of them. No.
Okay, thank you.
Okay, thank you again, Georgina, for your very nice talk. Thank you everyone for participating and for asking questions. So now we go to the break, and we will be back at 11:45 with Mario Serrano. Thank you.
Thank you. Thank you.
Thank you. Bye, Georgina.
Bye.
Thank you for being here. We are going to continue. So today we have the next talk of Mario Serrano, also of the Center for Genomic Sciences of the UNAM. And his talk will be about differential defense response induced by changes in the plant cuticle. Welcome, Mario. If you are ready,
Thank you. Let me share my presentation. So
perfect. Go ahead.
I hope you can see it, right?
Yes.
Okay. So first of all, thank you so much for the organizers to give me the opportunity to present this the results of this topic that is related, as they say, in the plant cuticle and defense responses. So the main goal for all the people who is in this Congress is actually the production of better food and to have not only better quality, but more food. And this has been a task since the beginning of the time of the evolution of the food. So since the agriculture started 10,000 years ago. And one of the driving forces to do this is because the population is always growing. So the man actually has been really successful to produce enough food for the moment, since due to different changes in the technological and from the synthetic point of view. And here I just show you the change that had been driving this evolution from the Neolithic revolution that they start to have, we pass from collecting the food to produce it ourselves. Then the agricultural revolution, when they start to use the animals to prepare the soil, among many other things. Then the machine revolution, that they start to use not only the machine, but also the agriculture start to use fertilizers, and they start to make a new varieties of different plants to produce more. And right now we have this biotechnological revolution. Why I say this? Because right now, let's say the thing is that we have enough food to feed all the people, but still there is a lot of people who doesn't have enough food in their tables. Why? What is the problem? There are many problems. There's a multifactorial problem, but one of them is because we lost a lot of food. Between 25 and 50% is lost post-harvest. So this is really, you can see it, it's really a lot of food that can be used to feed and to be in the table of somebody else. So who are the responsible of these losses? Well, most of them, they are the microorganisms. Within all of these genera, there is Botrytis cinerea. And in my group, we use Botrytis cinerea as a model, as a micro model. So Botrytis cinerea is a necrotroph pathogen that actually, as I say, is very important for the agriculture. As Hailin today showed, is the second most important pathogen in the agriculture because they affect many different crops, and they affect many different organs, such as the fruits, the flowers, and the leaves. So let's take a look how the Botrytis cinerea infect. So in the upper part of the plant, let's say on the leaves, that's the main focus on my talk, the Botrytis cinerea spores arrive over the cuticle, and then they have this first mechanism, the defense mechanism, this physical mechanism that the plant has in the cuticle. However, Botrytis cinerea has the meaning using different enzymes to degrade the cuticle of the cell wall. But once they degrade it, they will release different MAMs and DAMs for microbial and damage-associated molecular patterns that we know that they are recognized. Yeah, we don't know yet how they are recognized, but we know they are recognized. And once they are recognized, the plant can induce the early defense response, including the innate immune response, the reactive oxygen species accumulation, and the late defense responses induce it or drive it by the accumulation of the salicylic acid, jasmonic acid, ethylene, and abscisic acid dependent signaling pathways. But still, for most of the plants, the interaction with Botrytis cinerea is a compatible interaction. That means that the plant is infected. Why? Well, at least there are two possibilities. One is that the induction of the innate immune response and the late different responses actually are late. So they are very late in the body. And that's why botrytis cinerea infect. And the other one is that botrytis cinerea actually use either an effector, as oxalic acid, or even the small RNAs, as today in the morning we nicely saw, to modulate and to put it down the different responses. So what would we like to know in this model, in this pathosystem, botrytis cinerea and the plants? So we wanted to understand what happened in the first moment when the pathogen interact in the cuticle. What are released? The DAMs, MAMs, how are recognized, and how the plant can recognize these DAMs or MAMs, or both. And also we wanted to further characterize the innate immune response, in particular the reactive oxygen species. And also we wanted to know the whole picture that might lead to the resistance against this important pathogen. In order to do that, we have the following research projects in my group. So we have, in the first part, in the cuticle, we analyze different cuticular mutants to understand, as I say, the first events in their interaction between the pathogen and the plant. Also, we are performing a chemical genomics to try to characterize how the reactive oxygen species can be modulated in order to have a resistance. Additionally, we have a genetic screen to identify and characterize different genes, early response genes that modulate the plain innate immunity and the late different responses. And finally, we have also mutants related with the resistance. These mutants are more related to the salicylic acid, jasmonic acid, ethylene, and ABA-related signaling pathways. And in addition to this basic research, we have a more applied research that is the identification of characterization biostimulants that might allow us to change any of these signaling pathway, but in the end, we can either modify or interrupt the growth of botrytis cinerea, or we can modify and induce a different response that in the end might lead to the better different response against this important pathogen. However, today I will just focus on one of these projects, and this is the characterization of the cuticle mutants, in particular the molecular changes in the cuticle. So, and first we wanted to know that the cuticle is the layer that actually covers all the organs on the upper part of the plant. So, and they are mostly formed by two important polymers, the wax, that they can be epicuticular, so outside, and intracuticular. And the other important component, chemical component, are the cutin. So these two polymers, they form most of the 90% of the cuticle. So we have recently shown, or not shown, but described, together with Wendy Aragon, that the cuticle not only regulates the abiotic stresses that previously was very highly described, but also the biotic interactions with herbivores, with bacteria, and also with the fungi. And during the last years, I have been working with this idea that change on cuticle composition leads to resistance against botrytis cinerea. And in the following slides, I will show you the evidence that points out this statement. So the first evidence is that when we apply cutinase, that is an enzyme that degrades the cuticle, we release the DAMs. And then if we remove the cutinase and now in the same place we infect with botrytis cinerea spores, we have full resistance. The second evidence is a genetic evidence, because cutin-related mutant present a syndrome, a defense syndrome. And the defense syndrome is the following. So all of these are cutin-related mutants, Bodigar, LAX2, Filhead, and Lacerata. All of them, they are resistant to botrytis cinerea. All of them accumulate more reactive oxygen species with this green color. under the basal condition, not during the interaction with botrytis cinerea. And also, since they are not, the cuticle is not properly formed because there's the lack of reduction of the cutin, they have holes in between, and that's why they have more permeable cuticle. And this we can detect it by several means, but here I show you one using the toluidine blue. This colorant, it can only penetrate if the cuticle is permeable, and then it can interact with the cell wall and stay over there. So they have these three phenotypes: resistant to botrytis, reactive oxygen species, and permeability, more permeability. So it's important to say that this phenotype or this syndrome is not only observed in Arabidopsis thaliana. Also, they have been described at least in other two plants, in tomato and also in Camelina sativa. So here I have to be very clear. Not all the plants that modify the cuticle have the same defense syndrome, but at least it's not only in Arabidopsis, and then it's okay for us because we can use this Arabidopsis as a model, and we can transplant the information at least for these two plants. So now, this is an important slide because how we explain this resistance, it was our model, and we proposed this model in 2014. And we trying to explain why we got this resistance as a follow. So when we make changes in the cuticle, either by mutation or by treatment with a cutinase, we make more permeable cuticle. And then the DAMs and PAMs can go inside faster, and this faster can then make the plant recognize also faster. So they can recognize the DAMs or the PAMs more efficiently, and they can build up and the defense response more efficiently, also the innate immune response, the reactive oxygen species, and the resistance. So this was our model, that we proposed. But however, this model applied only when we changed the cutin. Then we take a look, okay, what happened with the other polymer that is important for the cuticle? And those are the WACs. And there was a lack of information about the interaction with this pathogen and the WACs. And that's why, together with Wendy Aragon, this is a student, a PhD student, she tried to answer this question. If change on wax composition also might lead to the resistance against botrytis cinerea. And this is what I'm going to talk about it. So first, what we do is to select the plants that we have to work with. Unfortunately, we have several mutants in wax and in cutin. So for cutin mutants, we use Bodyguard and LAX2 that they have been properly described, and they are, as I say, more resistant and more permeable cuticle. Then the wax mutants that we don't know much about them, so they have been described, but not much about them in relation to the interaction with the botrytis cinerea, and there were SER1 and SER3. And finally, we use one or own mutant, that is XII, that actually, as far as I know, is the first mutant that has reduction in both polymers, in cutin and wax. And also, we have described that it is more resistant to botrytis cinerea. So this is our material that we would work on it. So the first question that we have was, okay, what happened? Let's base our research in the model. So what happened at the cuticular level? All the mutants are more permeable, and the answer is yes. So we use different strategies to determine that the cuticle is modified and is more permeable. Here we have the wild types. Here I use two different ecotypes because ICA2 is in the background C24 and call 0 for the rest. So that's why you saw these two wild types. So when we use the toluidine blue, the chalcofluor, and also measuring the chlorophyll leaching. And for all of them, what I can say is that the cutin mutants, they have a strong cuticle permeability, but still in SER1 and SER3, we still have more permeable cuticle than the wild type. Then the next question was, okay, what about the basal reactive oxygen species accumulation? Once more, we observed that all the mutants, Q10 and WAX mutants, they have more reactive oxygen species. But this is under basal condition. And where it was more clear was when they interact with the pathogen, with botrytis cinerea. Here we can see clearly that they have more reactive oxygen species than the wild type. So we have more permeability and more reactive oxygen species. The next question, the logic question was, what about the interaction with botrytis cinerea? And here we have a very nice surprise, because as I say, for the Q10 mutants, we have the full resistance. However, for SER3 and SER1, they were susceptible as the wild types. So what does it mean that? So that means that the model, it was not complete or it was incorrect. And as George Box said, all the models are wrong, but some of them are useful. So we used this model for several years, but now I was very, very excited because right now we can improve our model. We can ask new questions and to see what else is involved in this cuticle-dependent resistance against botrytis. So the first question was logic. So why these Q10 mutants were resistant? And then in collaboration with Damian Formey, a colleague, also a researcher from the CCG, and Wendy Aragon, what we performed was RNA-seq to determine the transcriptional changes during the interaction with all of these set of mutants. And we analyzed two conditions. The first one was a basal condition without a pathogen, and afterwards an infected condition after 6 hours of interaction. So Damian and Wendy, they make a very precise and extensive analysis. However, I don't have time to go through all these results, but what I can tell you is the following. So first, the Q10 mutants induce the defense-related genes, and the WAX mutant induce an abiotic stress-related genes. When we saw this result, we say, oh, that's why the Q10 mutants are more resistant to botrytis cinerea. Then we move, okay, what else the information we have? So since botrytis cinerea is so important, we have a lot of information at the transcriptomic level. And we know this model, all the late defense responses are dependent, or the defense responses against botrytis cinerea are dependent of the canonical defense responses, including salicylic acid, hasmonic acid, abscisic acid, among others. So we took a list and we asked the question, okay, can we see a clear pattern where all the Q10 mutants are induced in one signaling pathway or another? And the answer is no. The canonical defense responses are not completely induced is in yellow, sorry, repressed in blue. So not all these canonical defense responses are induced for all the Q10 mutants. Just an example, here we have for bodyguard and LAX2, we have an induction, but not for ICA2. So that means that probably they might have a role in this Q10-related defense response, but we feel that it was not valid for all the mutants because we can expect that all of them, they have a different Q10 composition, reduction in Q10 composition. So we expect that the signaling pathway might also be all induced, or at least some members of them. So we didn't see it. So that was really interesting. And the way that we looked, it was, okay, so let's identify these genes that actually are shared in induction on the expression pattern for all the Q10 mutants. And fortunately, Damian and Wendy identified a set of genes that actually have this expression pattern. So all the genes were induced in all the 3 mutants and repressed in the SER3 and SER1. We were very excited. Why? Because among these genes, we have genes related with the cell wall remodeling that actually we verified using not only the RNA-seq, but also by qRT-PCR. And why we are excited? Because these cell wall remodeling genes actually have been described very recently involved during the interaction with botrytis cinerea. And this is the first time that the cuticle-related defense response are linked to this kind of genes. So we were very excited. And right now, as usual, we have a new model to work on. So we now know that the Q10 and wax mutants, they are more permeable. Yes, all of them are more permeable in different degrees, but they are more permeable compared to the wild types. So that means that they might allow the DAMs and BAMs to go inside the cell wall. Also, we know that they have more reactive oxygen species, again, with the difference, but very strong when they interact with botrytis cinerea, and this is important. But afterwards, they are different because in the Q10 mutants, there is the induction of the different-related gene expression, while for the wax mutant, we have a different set related with abiotic and photosynthetic-related gene expression. And this difference in the Q10 mutants, in particular, there are genes related with the change or remodeling of the cell wall. So now we have this starting point to ask questions. And one of the questions, we have several, but one of the questions is, what happened? What is the change between the accumulation of ROS and the activation of the early defense response genes? That it doesn't happen in the wax mutant. And now we have the tools, we have these mutants, and we are looking forward to build up a new model and to have more information to change our point of view. So this result was recently published last week, actually, in Frontiers in Plant Science. So please take a look, and you can tell us new ideas and ways to collaborate to improve our work. But now I wanted to finish with the work that we are working on. So the first one is working with ICA2. This mutant that, as I say, they have more cuticle permeability, they have more induced basal and induced reactive oxygen species. As I say, is the first mutant that we have less wax and Q10. They have a faster induction of the plant defense responses. And the most important thing that really excites me is that they are not only resistant to botrytis, they are resistant to Pseudomonas and also to herbivore. But not for all the pathogens. We have also is more susceptible for Ptyphthora, for instance. So it's not a general defense syndrome, but specific ones. And then we can not only analyze the interaction with botrytis cinerea, but also with other important pathogens. And the way that we are trying to do it is, first of all, well, we saw already a very nice talk for the small RNAs, and we wanted to study the role of these molecules in this kind of mutant with a cuticular mutant. And this is a work from Emil Padilla, a postgrad master student, and once more, Damian Fournet. And what we are doing is to analyze what happened with the small RNAs. And we are very excited because in the differential expression of the small RNAs, we identify a large amount of small RNAs that actually are present in ICA2 and not in the wild type. So right now, the table is served to try to answer and to try to understand if these small RNAs are playing an important role to have the cuticle-related defense responses. Another project that we have is in collaboration with Delia Narvaez and Claudia Martinez. Claudia Martinez is in the Institute of Biotechnology. Delia was her student, and right now she's in the Weizmann Institute in Israel. And now we wanted to ask, okay, this Q10-related defense response is present in all the necrotrophic fungi, in all the necrotrophic organisms. And to answer that, we use their model, Pectobacterium, and we use the same strategy. Let's infect Pectobacterium with the cuticular mutants. And here, surprise, surprise, is exactly the opposite result, because all the Q10 mutants, ICA2, Bodyguard, and LAX2, actually, they were more susceptible than the wild type and the SERV mutants. So right now, we are trying to build up an history to try to understand, to give the first steps to understand why this is a susceptibility. So, but important mentions is here that we cannot say it works for one, it works for all. So the plant-microbe interaction is really a complex history, even with the same strategy. So both of them, they are necrotrophic pathogens. We have a different response. So it's a very interesting and challenging subject. Finally, I wanted to mention, that we are working with a new plant model. And this is thanks to the collaboration and the opportunity to work with them, with Ana and Mario Arteaga from Bioteca. And this is to analyze what happened, not only the interaction with botrytis cinerea, I focus here with botrytis cinerea, but also with Ana in particular, we are working with a biotic interaction. And to see if we can learn from this very interesting model, Marcantia. And we have already our first results. The student of Mario present, I think on Monday, present the results. And so we are really excited to have this kind of collaborations. Finally, I just want to thank the organizations that they give us the money. So in Switzerland, in the United States, and also at UNAM, in my center. And of course, I have to thank the people, the driving forces of this. I just present the results, but actually we have a very enthusiastic and driving force of several young people that working in my group. And I want to thank them, all of them. Unfortunately, I didn't have time to present all the other projects, but sometimes we might have the opportunity. And in the end, I just wanted to thank also the institutes with we collaborated. We have several collaborations, and we are always looking forward to have more in Mexico and abroad. We have several lines and several collaborations. And that is one of the most important and I think more exciting parts to make science, because we can collaborate with many different profiles, many different knowledge. And I'm looking forward to have the next collaboration. So with this slide, I wanted to thank you. Here is my email, but also I wanted to include the email from Wendy Aragon. Right now, Wendy is finishing her PhD and she's looking for a postdoc. So please, if you have an opportunity, she's a really intelligent and driving force colleague. So I will really highly, highly recommend it. So with that, thank you. And I hope I can answer your questions.
Thank you, Mario. Very nice talk, very interesting. So we have some time for questions. Please go ahead. Let's see if we have some questions from the audience. Alejandra Covarrubias, please.
Yeah. Hi, Mario. How are you? Good. Very nice. Very nice presentation. I wonder whether you know, cuticle and wax covers in the shoot parts of the plant are very important to keep the water inside the cells, right? So so for sure, both structures. If you alter both the structures, you will have responses that had to do with abiotic, at least with water deficit conditions. So what do you think about it? I mean, that is something that always goes together, no? The response to pathogens and the response to abiotic stresses.
Thank you so much, Alejandra. Yeah, this is a very important question. Actually, I missed to tell you. So in these mutants, when there is a reduction of cutin, there is always a compensatory effect. They have more wax. But still, all these plants, some of them, they are very, very, very bad in shape, I must say. But many others, for instance, ICA2, they are a little bit smaller, but they survive perfectly. So they lost water for sure, but they are not so affected because they have these compensatory effects. And that's why we can still use it. I'm not, so the message is not that, okay, to improve the resistance to botrytis cinerea, we need to modify the cuticle, because then all the pathogens will be more easily to go inside and infect the plant. They will lose water, they will produce less food. But this is a model to understand how we can improve what is behind. That's why it's important to see the genetic elements, all the genes, proteins that can be improved, and they were identified using this model, modifying the cuticle permeability.
Yeah, just suggesting maybe it would be good to test them under water deficit conditions, right?
We are doing that with actually with Anna. Yeah.
Okay, good.
Thank you so much. Yeah.
Okay, thank you very much. So the time is almost up. I see Aida Martinez with her hand. If it's a very short question and a short answer, we can take it. And later, please check the chat.
Okay.
Yeah. Mario, hi. Very happy to see you again. I'm not sure if I missed, but your ICA mutants is in which gene? I mean, they are in biosynthetic, no, because they're very different routes. So please.
Okay, thank you. You made the hardest question. So we still don't know what is ICA2. So we hope to publish these results. We are working on that for several years, but it's really hard to crack. And so we don't know, to be honest, but the way that they were identified, actually ICA2 modified expression of early response genes, and probably is not related with the biosynthetic, but at the regulatory, at the transcriptional level. But we are working on that. Yeah, this is really a nightmare for me because we haven't known exactly what is ICA. Yeah.
Great. Thank you so much, Mario, again.
You're very welcome. Thank you so much again.
You're welcome. Bye. So we go to Dan.
Our next speaker is Saskia Hogenhelt from the John Enos Center. And her talk will be how bacterial phytoplasma pathogens change plant architecture and plant defense to insect vectors. So if you can share your screen, Saskia.
Right. Do you see my screen?
Yes, we can see it perfectly and hear you. You are ready.
Brilliant. Thank you. So I really want to congratulate the organizers for organizing this exciting and informative conference. I'm very honored to be invited to give this talk, and I thank the organizers for inviting me. And I'm going to talk today about phytoplasma vectors. And this is a long-term research topic in my lab, which has had the contributions of many people. And this is my current lab, and they have all contributed some way or another. And so this is let me see if I can sign. Yeah, this is about molecular plant microbe interactions. And it's we are working on the leafhopper plant interactions and leafhopper phytoplasma interactions and phytoplasma plant interactions. So it's a tritrophic interaction. And what's really interesting about the system is that the phytoplasma, which are bacteria, are inducing all this interesting developmental phenotype, such as vicious brooms and phyllidi in plants. And we're interested to know how they do this. So there are a number of plant pathogenic bacteria that are insect transmitted, and a few of them are maybe known to you, which is the Liberibacter species, which is also introducing the citrus greening disease, and also Salmonella fastidiosa, which is transmitted by sharpshooters and fruithoppers, and are also making all kinds of economically important in grapevines and other trees. And then there's Spiroplasma citris, Spiroplasma conchyli, and also Spiroplasma phoenicium that are insect transmitted pathogens. And then there are up to 1,000 different subspecies, or more than 1,000 different subspecies of phytoplasmas, which are all leafhopper, planthopper, and psyllid transmitted. And so I'm going to talk about the phytoplasmas today. Phytoplasmas induce all kinds of interesting symptoms, as one can see here. It's found in many different plant species. I cannot imagine one plant that is not infected with the phytoplasma at one point. And these are a few examples: rapeseed, for example, and this is phyllidi symptoms. And also carrots are getting infected with phytoplasma, and you get these hairy roots. Rice is showing this yellowing kind of and tillering kind of symptom. Then there's palm lethal yellows, witch's broom, disease of Lyme, and grapevine yellows, just here as examples. But in Mexico and elsewhere, there is a few important phytoplasma diseases, for example, in papaya. So papaya, not banana. Papaya Bunsi top phytoplasma is a global problem. And as you know, papaya is the third most cultivated tropical crop worldwide. And Mexico is one of the main exporters. And I know that this particular phytoplasma is causing an issue for Mexican papaya growth. And also, it's interesting that papaya is mostly originated from Mesoamerica. And so it would be interesting to see what kind of diversion of kind of variation there is among these varieties to be infected with phytoplasma. Now, a lot of other phytoplasmas found in Mexico, in cacti, in coconuts, in palms, and marigolds, in potato, and tomatoes, in maize. And probably this is an underestimation of the diversity of phytoplasmas in Latin America. So phytoplasmas are very prevalent in Mexico. So phytoplasmas induce this interesting kind of phenotypes. It includes vicious brooms, but also phyllodes, as you can see on the right. And in many different crops, they induce these symptoms. But what they also do is they make these plants more attractive to leafhopper vectors. And the phytoplasmas are obligate inhabitants of their plant hosts and insects. And so they are really requiring both for spread. So by making the plant more attractive to the insect vector, they also guarantee their own survival and spread to other plant species. And so we call these plants zombie plants. And that's because the plants appear to stay alive only for reproducing or spreading the pathogen. The plant itself is not producing seeds or flowers. The flower, if they produce flowers, they don't produce seeds, and they remain sterile, and the plants become better hosts for the insects. So that's why we call them zombie plants. And there are a lot of phytoplasmas described, as you can see here. These are just a few. This is actually quite a small fraction of the phytoplasmas that are known. For these, we have some sequence information. And the Mexican citrus, papaya, and maize phytoplasmas belong to this Aspergillus clade that are up in the tree here. And we are working on one of the Aspergillus phytoplasma called Aspergillus witch's broom. And the reason we are working on this particular phytoplasma is because it can infect Arabidopsis thaliana. And you can see that in Arabidopsis thaliana, it also induces this typical witch's broom kind of symptoms, and also the phyllodes symptoms, so the leaf-like flowers. So witch's broom and leaf-like flowers. Witch's broom is actually a stem proliferation phenotype, and also leaf-like flowers are induced by this phytoplasm in Arabidopsis thaliana. And this is the life cycle. It's transmitted by an insect called the oyster leafhopper, Macroceles corneatus. And the whole cycle between the insect and the plant is 10 days. It takes 10 days for the phytoplasma to colonize the insect and 10 days to colonize the plant. And so the whole cycle is 20 days. And also within 10 days after inoculation into the plant, you can see that the Arabidopsis thaliana is showing these typical symptoms. The phytoplasmas are in the phloem of the plants. And when they're in the phloem, they're intracellular. So when the phytoplasma secrete their effectors, so they have a signal peptide, then the effectors will directly arrive or be deposited inside the cytoplasm of the plant cell. So there's no need for a type 3 secretion system that are typical possessed by Pseudomonas or Xanthomonas. These bacteria can deposit their effectors directly into the cytoplasm. And in the genome, we have identified 56 secreted Aspergillus witch's proteins, which are putative effectors called SAPs. And we have identified or functionally characterized many of them so far. And so what happens is the leafhoppers, they feed on the phloem. And when they feed from the phloem, they inoculate the phloem with these phytoplasmas. The phytoplasmas that migrate via the sieve cells to systemically, so to distal tissues. And while they infect the phloem, they also secrete these effectors called SAPs, and these include SAP11, SAP5, SAP54, etc. And these SAPs are released in the phloem, but they can also unload from the phloem and migrate to adjacent tissues. The phytoplasma stay in the sieve cells, however. So we have functionally characterized quite a number of them, and I'm going to talk about 3. And these are called SAP11, SAP5, and SAP54. And SAP11 and SAP5 induce these vicious brooms. SAP54, the leaf-like flowers, and SAP11, and all 3 of them actually are promoting the insect fecundity or attract the insect vectors. So I'm going to tell you a little bit about SAP11. And SAP11 is degrading or destabilizing TCP transcription factors. And when they do this, they induce this stem proliferation called vicious brooms. And TCPs are plant transcription factors with key roles in plant development. And this is what we see when we overexpress the SAP11 effectors, and these are 2 of them, SAP11 of Aspergillus vicious broom, but also SAP11 of maize bushy stunt phytoplasma. You can see that the Arabidopsis thaliana plants produce more stems. And that's similar to a BRC1, a BRC2W mutants, which are mutants in null mutants in the TCP transcription factors. And this is the mechanisms that we have uncovered. So the phytoplasma is in the phloem, and when the phytoplasma secretes the SAP11, it degrades the TB1, which is the TCP transcription factors, and then you get outgrowth of more stems. And that's how you get this stem proliferation or vicious broom-like phenotype. And this is true for many lines. And so we have measured the number of branches and the different transgenic lines induce this branch proliferations. And this also happens in maize, for example. Also, maize bushy stunt phytoplasma is a maize pathogen and also has a SAP11 effector. And we find that this SAP11 factor also is degrading the TB1 transcription factor of maize, and you get these bushy plants, as you can see here. And so this is really interesting because maize, as you know, is having female and male organs, and the tassels on the top, and females are the cops that are on the stem. And the TB1 mutant, which is the TCP mutant, you get this kind of phenotype with lots of tassels and lots of branches. But the SAP11, when you overexpress the SAP11 of maize bushy stunt, you get a very similar phenotype as the TB1 mutant. And Teosinte, which is why maize is domesticated from, is also having a TB1-like features because TB1 has been involved in the domestication of maize. And also maize bushy stunt phytoplasma is inducing, is basically making the maize plant go back to the Teosinte ancestor. So it's interesting how maize, the phytoplasmas are inducing the symptoms in maize plants. SAP11 also makes these crankled leaves, as you can see here. And so the Arabidopsis thaliana plant looks like kale. And that's because the SAP11 is also degrading other TCP transcription factors called syn-TCPs. And by doing that, the syn-TCPs are normally promoting the jasmonate production, and SAP11 inhibits this. And then you don't get jasmonate production. You then get more proliferation, less expansion, and less senescence of the cells in the leaves, and this causes then the leaf crinkling. So SAP11 is inducing leaf crinkling by destabilizing these syn-TCPs. And as you know, jasmonates are very important hormones also in defense. And this affects the leafhopper factor. This leafhopper factor is laying eggs on the plants, and the eggs hatch into nymphs. And then when we silence the LOX2, or we knock it down the LOX2, then the insect will lay more eggs on the plant. And it does the same on the SEP11 transgenic plants. So, it seems that the SEP11 is promoting also the leafhopper egg laying. Now I'm going to say a little bit about SEP54, and SEP54 is a factor that induces phyllody and also promotes the leafhopper factor feeding and traction. And this particular factor is destabilizing the METS transcription factors, and they are involved in flower development, and you get phyllody. So when we express the SEP54 factor in plants, you get these typical symptoms like phyllody. And you can see that in the middle, when the SEP54 is there, what you get is that you get an indeterminate flower development in which the stems are still developing from the flower, whereas in the healthy flower, you don't see stems. Also, the structures of the flower stay leafy and stay green. And that is similar in the Astealis, which is bloom-infected Arabidopsis thaliana. And what we found is that SEP54 binds METS box transcription factors. And METS box transcription factors are involved in all kinds of different processes in flower development, including sepal, petal, and stamen and carpel development. And we found that SEP54 binds these METS box transcription factors, and the METS box transcription factors are then degraded by the cell garbage bin, the 26S proteasome. And it does that by also recruiting another protein called RED23. And RED23 is a shuttle factor, and it shuttles the SEP54 METS box transcription factor complex to the 26S proteasome, and then it gets degradation of the METS box transcription factors. And so you don't have METS box transcription factors, or less of these factors, and then you get leaf-like flowers. And we found that this RED23 is actually a susceptibility factor. If you make mutants that do not have a RED23C, and you infect those mutants with phytoplasma, then you do not get phyllody, as you can see on the lower right. However, if the plant does have RED23C, as you can see on the left, and is then infected with phytoplasma, you get still the phyllody. So it suggests that RED23, and especially RED23C, is a susceptibility factor for phytoplasma induction for phyllody. Now, we also find that SEP54 makes the plant more susceptible to the insect factor. And you can see that here. When we do choice tests, then the insects are much more attracted to the GFP SEP54 plant, as you can see here. And also, when you infect the plants, and especially the plants that are still making a leaf-like flowers, and a RED23BT mutant, then you can see that the insect also is more attracted to the plants that produce the leaf-like flowers. So this suggests that SEP54 and in its activity is making the plant more susceptible to the insects. I'm going to say a little bit about SEP5. And SEP5, now see this is, SEP5 is making bushy plants, and also is making the plant more susceptible to the leafhopper factor. And we found that SEP5, when you overexpress SEP5, SEP5 is making the plant more bushy, as you can see here. It's making more leaves and more stems, and the plant looks like completely not like an Arabidopsis plant any longer. It's an amazing phenotype. And we found that SAP5 interact with multiple plant SPL and GATA transcription factors. And there are many of them, as you can see here, they're big families. And we found that SAP5 bind to the zinc finger domain of these transcription factors, both the SPL and the GATA family. They have zinc finger domains, and the SAP5 is binding specifically to the zinc finger domains. And they do that of many of them. And we also found that these GATA and SPL transcription factors are completely degraded by SAP5. And this an important transcription factors because SPLs are involved in a juvenile to adult transition in plants, and also they promote the vegetative to reproductive transition in plants. And whereas GATA is inhibiting the stem production after when the plants become adults. And what SAP5 does then is by degrading both SPLs and GATAs, it promotes the neoteny, so it makes the plant more juvenile, and it promotes it then continues making leaves. But at the same time, it also initiates the stem production because it also degrades GATAs that inhibit stem production. So you get then these very bushy plants. And there are many SAP5 factors characterized in phytoplasmas. The phytoplasmas are diverse, as I told you. However, the SAP5, SAP11, SAP54 factors are found in many of the divergent phytoplasmas. But in the SAP5, there's something very interesting going on, and is that the majority of the SAP5 homologs of phytoplasma do bind both GATAs and SPLs. However, some bind only SPLs and some bind only GATAs. And in the case that they bind only one or the other, the phytoplasma often have two. For example, peanut witch's broom phytoplasma has two different SAP5s. One binds SPLs and the other one's GATA. And if you express only the GATA or only the SPL binding SAP5s, you can see that the symptoms are, the phenotypes are consistent. So when only this WDLA, which is binding only the SPLs, you can see only leaf production, but not the stem that's growing. If it only expresses WBDLB, which is only binding the GATAs, you can see a lot of stem production, but not leaf production. So you can see that it's very specific in this case. So what we found is that SAP5 acts by recruiting also a 26S proteasome component, but in this case it's RPN10. And RPN10 binds the 26S proteasome and then drags the complex to this proteasome and degrades then the cargo, which in this case are the SPLs and the GATAs. And what's interesting is that it's a very unique mechanism, because normally the proteins need to be ubiquitinated by E1, E2, and E3 ligases, and involving ATP. And the ubiquitin chain has to be put onto the target, and only then will it be degraded by the 26S proteasome. However, phytoplasma SAP5 has circumvented this ubiquitination and just drags the entire complex to the 26S proteasome without ubiquitination. And we found that we can actually make plants resistant to this SAP5. And that is by making changes in the RPN10 and just two amino acids from GA to HS. And GA to HS mutation is then inhibiting the interaction of SAP5. And when we express SAP5 in this RPN10 changed plants, then we don't see the symptoms any longer. And we can also see that when we do when we infect the plants. So we have this RPN10, the wild type RPN10 still interact with SAP5, and this particular plants show really nice symptoms that are characteristics of SAP5, degradation of SPL and GATAs. But if we make the plants resistant by introducing the GA to HS conversion, then the RPN10 is not interacting with SEP5 and the plants do not show these symptoms. And what is interesting is that SEP5 is, when it interacts with RPN10, it makes the plant live longer. So, and this is really important because if the plants live longer and they have all these symptoms, the insect can live longer as well and can also acquire the phytoplasma for longer. So it really has, SEP5 has also not only in phenotype in induction of the symptoms, but also makes the plant more attractive to the insects. So this is what I just showed you. So there's this wild type plants that are both SPL and GATAs are involved in making the plant look into the adult, generate the adult version of the plant. However, SEP5 is interfering with this process and you get then these very bushy plants. But we can make this plants resistant to the SEP5 action. And then by making it not interact with RPN10, and then you get normal plants again. And we can introduce this kind of mutation with CRISPR-Cas editing. And so this is a nice approach for creating resistance against vector-borne diseases, in this case, phytoplasma. And by doing that, we also inhibit the number of insects that are carrying the phytoplasma, because less insects will grow on the plants that are infected by the phytoplasma and be able to spread it around. So this is the summary. So we find that SEP11 do all kinds of things, the SEPs do all kinds of things. They degrade TCPs, they degrade METs, and they degrade SPL and GATAs. They are all very important transcription factors, and they are all very conserved in plants. And also the RAD23 and RPN10 are conserved in plants. And by doing this, they get these phenotypes. And also the insects are more attracted to these plants, and then they can carry the phytoplasma and spread the phytoplasma around. And we discovered approaches to increase plant resistance to phytoplasmas and their insect vectors. And I think this is important because maybe we can now use our tools and our discoveries to help find ways to get all these different plants that are infected by phytoplasma and see if we can find resistance to phytoplasma diseases. So with that, I would like to really thank my lab, and especially Vijay Wang, who has been really crucial for the last part of the story about SEP5 and the zombie plants. And also all my former lab members, and especially Alison McLean and Akiko Sujo. Akiko has really done a lot of work on the SEP11 project together with the Heather Kingdom. And Alison McLean and Bo Sigmunds have done excellent work on the SEP54 part of the work. And Christina, Ali, and Pascal have done an excellent job with the SEP11, the projects, and also with SEP5 and SEP54. And Melissa is doing the modeling, has done a lot of modeling on the system. Many collaborators, very grateful for their contributions, and also the JLC entomology staff, of course, for the insect work. And at last but not least, I would like to really advertise our latest paper that is on the SEP5 project, and it came out in the issue of Cell in September. And this is also a really, really cool story because this is when the arts and science meet. And this is Suan Pai. And Pai made this beautiful cover that made it to the cover of Cell. And you can see here the witch's broom, the phyllodes, the phytoplasmas, and the insects that are there, but also the witch that's on the lower left. So it's a beautiful, beautiful design, and I thank Pai and VG for this amazing artistry. So with that, I'm happy if there's time, make any questions.
Gorgeous talk and gorgeous cover too. I think we have time for one very quick question in the chat. Jume asked, great talk, Saskia. Are the SAP proteins related? Are they mobile?
So they are related. Yes, they are having the signal peptides, and they have similar signal peptides. But there are different for different groups. Ones that are CEP11 and CEP54 are somewhat related, and CEP5 are a little different based on the signal peptide sequence. However, they've much more diverged. I mean, they've evolved fast, it seems, and they all located on specific mobile units, in particular arrangements. And because they are on these mobile units, it seems that they are having the similar origin. So I think they have, they're a big family, it looks like, although they have diversified and it's hard to find any sequence conservation. But they probably have structural conservation.
Thank you, Saskia, again. If you have more questions, please put them in the chat and Saskia can get to them when she can. Thank you.
Thank you.
Okay, thank you. So we go to the next talk. It's my pleasure to introduce my colleague from Irapuato, Alfredo Herrera Estrella, Director of Uga Langebio Simestaff, and he's going to talk about the trichoderma plant dialogue. So Alfredo, if you are ready, please share your screen and take it away.
Thank you. Thank you very much for the invitation. It has been a great meeting, really great labor in terms of coordination and setting up the meeting. So I will share my screen now, and I will talk about the trichoderma plant dialogue, as Stefan said. So many of you might be wondering, what the hell is trichoderma? You know the plant, of course, but what is trichoderma? So let me tell you, it's very easy. It's the love of my life. And this is what trichoderma does in the interaction with plants. So it actually has many features. It's a wonderful organism. It interacts with microbes in the soil. It's a mycoparasite that attacks directly phytopathogens, and it also produces antibiotics and competes for the niche with bacteria and other fungi as well as nematodes. It also emits a lot of different compounds that might be effectors that allow it to interact with the plant. And finally, it colonizes intercellular spaces in the plant root system, and this affects growth of the plant. At the end, you see an enhanced growth of the plant. You see changes in root architecture, and this is done basically because it somehow interferes with plant hormonal homeostasis, and it also triggers plant defense responses. And this is the ultimate result of what happens in this interaction. You can see an improved yield when you have plants that have been colonized by trichoderma. And that's, as Mario said, what we all want to see. Okay, so let me show you some of the evidences of what I just said. So this is Arabidopsis plants, and you see here on top plants that have not been inoculated with trichoderma, and these ones in the lower part have been inoculated with trichoderma. And you see the difference in growth. The foliage of the plant is much bigger. And you can see also here, I hope you can see, but if you have trichoderma Arabidopsis growing in the presence of trichoderma, you can see that there is an increase in the production of lateral roots and actually root hairs as well. And this leads to increases in fresh weight in the shoot. And these observations led to the proposal that trichoderma produce auxins, and it was actually demonstrated by the group of Jose Lopez and Sergio Casas more or less at the same time that it produced auxins. So I'm not going to talk much more about that. But we have also observed that volatiles produced by trichoderma can increase lateral root appearance in the plants and development and change the whole root architecture. So there are different factors which trichoderma uses to control root architecture and therefore plant growth. Recently, we have also observed that trichoderma has the capacity to produce cytokinins. So it's not only auxins. We have found all the genes necessary for the production of cytokinins in trichoderma. We have actually demonstrated that it produces free base cytokinins and it secretes these cytokinins mainly. I'm sorry, it secretes all these cytokinins like mainly cis-zeatin, and the mutants in those genes in the log and the IPT gene in trichoderma do not produce the cytokinins anymore. However, we think that this is not especially designed to interact with plants because many fungi you see here on the right that produce or that have these genes do not interact with plants. So we wonder what's the role of this cytokinin production in filamentous fungi. As I say, trichoderma also induces defense responses in the plant. And you can see here the induction of the salicylic acid response. When the plant is colonized by trichoderma, you see induction of this reporter's gene PR1A, and also the jasmonic acid pathway. And you see here again, induction of the reporter's gene LOX2 when trichoderma interacts with the plant. But it actually works in the field. So it's not just that we see in vitro and it's very nice. No, it works in the field. And this is some of the evidence. You see here what happens with wheat plants. You see when they are inoculated with trichoderma, you see increase in size and you see also the production of grain. And in maize, you can see changes in the root system. You see with no trichoderma and with trichoderma. But you know, there are always incredulous people. Even in my life, they have been people who do not believe in trichoderma. They see the in vitro and say, oh, it doesn't work. But now, I'm not going to say names of the people who were very good postdocs and very good students have been incredulous, and now they even produce trichoderma and sell it. So it's a very nice system. Okay, so today, I wanted to talk to you more about these enzymes and what's their role in the trichoderma plant interaction, NADPH oxidases. These enzymes are normally known to be membrane-bound, and it's a complex of different proteins. The catalytic subunit is considered membrane-bound, although in fungi it has been found in different places, and I will not get into that. But in the filamentous fungi, we find two different catalytic subunits that are called NOX-A and B, or NOX-1 and NOX-2, that are regulated by NOX-R, the regulatory subunit, and of course, many other components like in animals and in plants. And these enzymes produce superoxide, which can be converted then in hydrogen peroxide by SOD enzymes. These enzymes have been found to be involved in fungal pathogenesis. So there's an example here with Fusarium in wheat, where you can see that when you infect wheat with a wild type Fusarium, you see these symptoms. And if you knock out the NOX-A and B, you see a reduction in the symptoms caused by Fusarium. And here on the right, you can also see in the case of Magnaporte infecting rice, what is the effect of these mutations. So normally Magnaporte produces this infection system. You see a presoria, then an infection thread here that goes into the cells or into cellular spaces. And if you mutate NOX1, there is an attempt to do this, but it doesn't develop any further. NOX2 doesn't even attempt to do this, to make this structure. And of course, the double mutant doesn't make anything. It resembles more the NOX1, the NOX2 mutant. And NOX-R has a similar defect as the double catalytic subunit mutants. So we wonder, this is in fungal pathogens, and all these effects have to do with the development of different structures in the fungus, and therefore a defect in colonizing the plant and attacking the plant. So we wonder what happens in a beneficial interaction. So in trichoderma, just some years ago, we made mutants of the NOX1, NOX2, and NOXR proteins. We observed that they are defective in conjugation in response to mycelial injury, and you can see here more closely, and that they were defective also in the production of ROS, as you can see here, which is produced very rapidly upon injury in the mycelium. And this blue precipitate indicates the production of reactive oxygen species, which is no longer produced by NOXR and NOX1. But we wonder what happens then in the interaction with the plant. And this is what we observe first. You can see here an experiment of co-cultivation of Arabidopsis plants and trichoderma. And you see the effect of the wild type, the NOX1, the NOX2, and the NOXR mutants. And when we look at the root system in these plants, we could see that the wild type strain of trichoderma increases slightly primary root length when growing with the plant. And then lateral roots, these are strongly increased. So the wild type trichoderma has a strong effect in the production of lateral roots and root density and root biomass. These characteristics are partially lost in the NOX1, NOX2, and NOXR mutants. The effect actually in lateral root number is stronger in the NOXR mutant. And root density is similar in the NOX1 and NOXR mutants. And again, root biomass is decreased more strongly in the NOX when the plants are exposed to the NOXR mutant. So we ask ourselves why? How can this happen? And this is what we observe in terms of the formation of lateral root primordia in the plant. What you see here is the quantification of lateral root primordia, and it is separated in different stages of development. In the first 4 stages, we basically see no major difference if we apply the wild type trichoderma or if we apply the mutants. But at the later stages, in the maturation stage of root primordia, we see a very strong difference. And actually, the wild type trichoderma increases very strongly the formation of this mature root primordia, lateral root primordia, and this effect is strongly lost when we apply the NOX mutants, the NOX1 or the NOXR mutants. We see this defect. And if you quantify the total number of root primordia, you see clearly the increase when we apply the trichoderma wild type mutant, and this is decreased almost to the level when the plant is growing alone if we have the NOXR mutant. When we look more in detail and we look at the expression of DR5, an auxin responding gene here during the formation of this primordia, we can see here how the expression of this gene is increasing during maturation of the root primordia, and then you see during lateral root emergence what is happening. The wild type trichoderma actually increases this response and accelerates the development of the primordia. You can see it here and you see here, and it continues increasing the expression of DR5 in this region. And the NOXR mutant basically behaves like as if there was no trichoderma in the interaction. So it's very interesting to see these differences. And we thought, well, maybe there is no colonization by the mutant, and we look at colonization patterns, and what we can see is that the NOXR and the wild type mutants colonize more or less at the same level. So we look at the transcriptional level, taking samples before trichoderma establishes contact with the plant root system. That's 3 days plus inoculation, post-inoculation, and then 5 days post-inoculation when there is when contact starts. And you see here in the plant there is a small number of genes that respond 3 days after inoculation with trichoderma. This number increases at 5 days, but if you apply the NOX-R mutant, you see that the response of the plant is stronger, and actually much stronger by 5 days. So we wonder what's happening, why is this going on, what's wrong with the dialogue. And if you look at the heat map here down here, you can see that particularly at 3 days post-inoculation, so before there is any contact between the plant and the fungus, you can see that there is a small response when the wild type strain of trichoderma is inoculated, but it's much stronger in the NOX3 mutant. And actually it's not only the number of genes, but also if you look at the same genes, the level of expression is much stronger when we apply the NOX-R mutant. And this is shown here also in these plots, where you see that if you compare the genes that are responding in the plants when you apply the NOX-R mutant or the wild type trichoderma, with what happens with the plant when you apply the wild type trichoderma, you see many genes that are induced in this comparison, so they are more responsive to NOX1, and many that are repressed in this comparison, so that they are not actually repressed. And that is at 5 and at 3 days more or less the same behavior. So there is a stronger response of the plant in terms of gene expression. When we look at the type of genes that are responding in the plant, and we look at functional categories, what we find is that defense responses are much stronger when we apply the NOX mutant. You see response to wounding, response to kiting, response to indole-containing compounds, oxygen levels, immune system, response to insect, regulation of defense responses. There are more, these genes are responding more strongly to the NOX mutant. And if you look in a different way at all these comparisons, and we see just the expression when we have the wild type or the wild at 3 days or 5, and the NOX at 3 and 5 days post-inoculation, we see basically at 3 days that there is almost no response in these categories of genes in the wild type plant with the wild type fungus. But if you look at the NOX3, you see these differences very strongly marked. And if you look more in detail at what type of genes are these, you can see here genes that are involved in the stress responses, in damage responses, in the jasmonate pathway is certainly more strongly induced by the NOX-R mutant than by the wild type trichoderma, and also other genes that have to do with camalexin production and other defense responses, even to abiotic stress challenges. So we wonder whether this was reflected in protein levels in the jasmonate pathway. And what I'm showing you here is that actually, if we look at just one expression in the fusion with GFP, at 3 days post-inoculation, you see that there is activation here by the wild-type trichoderma strain, but there is a much stronger response when we have the NOX-R one. And if you measure this as relative fluorescence, you see a clear difference. And this happens in the root. You see these nuclear bodies where the just one protein is clearly staying there and very strongly. If you look at the aerial part of the plant, then you see here that this induction of JAS1 is earlier than when you have the wild type trichoderma. So this let us think that maybe there's an increased resistance to infection in the plant. And that's what we look at. And you see here, when we infected the plants with botrytis cinerea, we see in the first graph how inoculation with either the wild type or the delta NOX mutant of trichoderma reduces symptoms in the lips. So this is a very strong reduction, maybe 50% reduction of symptoms in the lips, but there is no difference between the wild type and the mutant. And if you take 5 days, it's the same. You don't see these differences. Also, you see that the disease symptoms have increased. So if you look more carefully and look at different types of damage or necrosis caused by the pathogen, you can see here how there is a slight reduction of severe symptoms when we inoculate with the NOX-R mutant, and there is an increase also in no symptomatic leaves. So that's correlates with our observation, although not as strongly as we expected. And if you look at lesion diameter also, you can see a difference, but not significant statistically with the wild type mutant. So this is what happens in the plant. But what happens in the fungus? So how is this communication? And again, we did a transcriptomic analysis of this, and you see here, very interestingly, how in the fungus, there's a very strong change in gene expression by 3 days of the interaction. This is in contrast with what happens in the plant. And by 5 days, it is back to normal, basically. And the NOX-R mutant seems that it doesn't really change its expression level. So it is not responding to the presence of the plant. And you can see this here in this heat map, and you can look at genes that are normally repressed in the wild type. They are actually induced in the NOX mutant, and you see this increase. And if you look at this set of genes, what are the functional categories involved here? You see here more clearly what happens in the trichoderma wild type strain. There's actually repression or no expression, or very little expression of these genes here. But in the mutant, at the same time, these genes are responding, and there is almost no repression of these genes. And they are related basically to nitrogen metabolism and to complex carbohydrate metabolism. So what we think is that these genes have to be shut down in trichoderma. So trichoderma produces many polysaccharide degrading enzymes, and this could harm the plant. So what we think is that at certain stage, early during the interaction, these genes have to be shut down to allow the interaction to progress. And you can see here what happens if we look at peptidases and we look at glycoside hydrolases. This is the normal response that we expected for all genes, and you see how these are less expressed in the wild type or more expressed in the NOX1 mutant. So particularly the glycoside hydrolases are controlling their expression. And if we look at a few of them more closely, if we just see what happened with a few of them to see more clearly the response, you see here that these glucoside hydrolases, at 3 days post-infection, post-inoculation, there is a reduction in their expression, very clearly. And they are back at 5 days. But if you look at the NOX mutant, they are expressed even without the presence of the plant, and they don't change their expression at all in the presence of the plant. So this is, we believe, a major problem in this interaction. And if you look at the communication, at functional categories that have to do with communication or perception of the environment, what you see is very interesting also, because you see that cell communication processes are activated in the wild type at 3 days, but they are not activated in the NOX mutant at 3 days. And if you look at DNA metabolic processes, they are activated also in the wild type at 3 days, but they are only slightly activated in the NOX mutant at 3 days. And what we think is that this has to do with this capacity of communicate between the plant and the fungus, and shutting down the expression of these polysaccharide degrading enzymes. And we did a very simple test. So why is this happening? So what we thought is that sucrose that is released by the plant was not being sensed by the fungus. So we simply grew the fungus in different concentrations of sucrose and cellulose, and you can see here that the NOX1 and the NOX-R mutants are defective in the utilization of sucrose compared to the wild type, whereas the NOX2 is not affected. And they don't seem to be affected in their capacity to degrade cellulose. But if we combine sucrose and cellulose, what you can see here is that the NOX-R mutant is also reduced in its capacity to utilize or to grow in the presence of these two carbon sources. And you can see that on the pictures here at the left. So to conclude, ROS produced by Trichoderma play a major role in interkingdom communication with plants. The Trichoderma plant communication begins before physical contact, leading to the activation of signaling processes in both organisms for the successful establishment of this mutualistic relationship. And the Trichoderma plant dialogue responds to nutrients released by the roots that lead to overall metabolic adjustments, particularly nitrogen and carbon utilization. And on the other side, the plant establishes a delicate growth defense balance in response to Trichoderma. You must remember that Trichoderma induces these defense responses, but there is no apparent cost in terms of growth to the plant. So this is very particular. And I just wanted to thank the people who participated in the work that I presented to you. Fabiola Lopez and Jose Manuel Villalobos were once working mostly in what I presented today, together with Ramon Pelagio and Pedro Martinez. Daniela Porres from my lab works on the cytokinin production. And the other half of Daniela is at the University of Guanajuato with my collaborator Vianey Olmedo. And of course, I want to thank the group of Jose Lopez Bucio, with whom we have established a very strong collaboration, in particular Sarai, Leon, and Jose himself, and Kai Abreu, who has collaborated for a long time with us in the transcriptomic analysis, and Luis Viva, who is now collaborating with us in cytokinin production. Thank you very much for your attention, and I hope you like my presentation, and if you have any questions, I'll be happy and try to answer them.
Thank you, Alfredo. Very nice talk. So let's see if we have some questions. There is Luis Cardenas. Please go ahead.
Hi, Alfredo. Can you hear me?
Yes, Luis.
Thank you. Very nice presentation. I just want to ask you a question regarding the effect of Trichoderma in inducing the lateral root increase in the lateral root productions. So as you said, in the mutants in Trichoderma, it induces less lateral root, isn't it? But in the wild type, it induces the proliferation of lateral root formations. Have you looked at the level of expression of the NADPH oxidases in the plant? Because it has been well described in the plant, the role of the NADPH oxidases in promoting the lateral root formation, just to see if the Trichoderma is able to modulate expression of the NADPH oxidases in the plant?
No, the short answer is no. We haven't looked at the plant enzymes, and we believe it's more, it could be related to auxin production also, right? And maybe there is an increase actually in ROS, which is also linked to auxin production. So it would be very interesting to look at that, but we haven't done it.
Yeah. So my other very short question is, have you looked at the root hairs under Trichoderma infections? Do they proliferate?
Oh, yes,
or increase the length? Yeah, okay.
Yes, there is an increased length in root hairs, very clear.
Okay. Thank you, Alfredo. Nice talk.
Thank you.
Thank you for the question. There's time for one more short question. If anyone likes to ask a question. There's a question of Alejandra Covarrubias. Go ahead.
Hi, Alfredo.
Hi, Ali.
Nice talk.
Thank you.
I was wondering whether the association with trichoderma in some plants that have a symbiosis with other organisms, it means like it is a competence, like, yeah, well, whatever.
Yeah, this is a question we have had for a long time, because it could actually even attack mycorrhiza, right? They are fungi.
Right.
And there have been several studies on that. And some people say they attack mycorrhiza. Some people say they don't attack mycorrhiza. And some people even say trichoderma facilitates the entry of mycorrhiza into the plant system. So it's very controversial so far. I don't think there is much going on in that direction. And but it's interesting that these molecules that mycorrhiza, the name is gone from my mind now, that are produced to establish interaction with the plant, they are produced basically by all fungi. They have slight differences in structure. So it is not clear how they are recognized anymore, but we haven't looked at those produced by trichoderma, so it might be helpful in terms of the plant mycorrhiza interaction.
Exactly, yeah. Okay, thank you.
Thank you.
Okay, great. Thank you, Alfredo, for the great talk.
Thank you very much.
We will move on to the next speaker. Thank you.
Thank you, bye.
Bye. So we continue with Dan. Go ahead.
It is my great pleasure to introduce our next speaker, Dr. Neelima Sinha from the University of California, Davis, and she'll be continuing the theme of biotic interactions with her talk, Interactions Between the Parasitic Criscuta campestris and its host, tomato. Neelima, the stage is yours.
Thank you very much, Dan, for that beautiful introduction. And I want to thank all the organizers for, organizing this wonderful symposium during these very challenging times. So I have to just make sure my Zoom screen is sitting in the right place and all of that. Give me a second to do that. Okay, here we go. So yeah, I want to tell you all about Criscuta campestris and its interactions with tomatoes. So these are biotic interactions between two angiosperms. And it's not a benign interaction because Criscuta is a parasitic plant and it parasitizes many different species within the Solanaceae. And here it's also shown growing on Nicotiana benthamiana. In this little video shot by my graduate student, Minyao Zhu, you can see that it's trying to grow on a morning glory plant. So wide host range and a pretty damaging parasite in agriculture. So I want to start out by acknowledging people and thanking them just in case I go over and I don't want to miss this most important part. A lot of this work was funded by USDA and also individual fellowships to several people who were involved in the work. Much of what I'm going to talk about was done by Minyao Zhu, who a former grad student who is now a postdoc at Cambridge, and Moran Farhi, a postdoc in lab, Richard Philbrook, who was then an undergrad in lab, and then many other people also contributed, including collaborations with Patrick Shee and with Ken Shirasu. So what exactly is a parasitic plant? I want to start there. Parasitic plants, turns out, are pretty widespread, and 1% of all angiosperms are parasites. Some really interesting ones, like this Rafflesia here with a human as a scale bar. The snow plant that seems to just grow out of the soil as it reproduces. And of course, really damaging and devastating pests like Striga hermontheca that can devastate cereal crops. So what makes a vascular plant a parasite? It turns out that it's the presence of this very specialized organ known as a hostorium. So here I show you examples of Cuscuta campestris, which is in the Solanales, growing on tomato. So here's a Cuscuta strand, this is tomato, and these are the haustoria that are being formed, and then they eventually penetrate into the tomato stem. This is a transverse section through the Cuscuta and the tomato, showing you these haustorial projections that have actually penetrated into the tomato stem. And so this is a stem parasite, Cuscuta is, and it's growing on the tomato stem. Here is another example, and this is Striga hermanthica in the Lamiales. It's a root parasite, and it's growing on a maize root. In this transverse section, you can see the maize root, and you can see the Striga hermanthica root, and here's the haustorium that is connecting the vascular systems of these two entities. So parasitic plants, it turns out that they are pretty widespread, phylogenetically speaking, and have evolved more than 11 times, but in every instance, we see haustorium formation. So these haustoria are used by the parasite to obtain water and nutrients from their host. So this is an example, a fascinating example of convergent evolution. So with that introduction, let's move on to the rest of my talk, and I want to divide it into two parts, the parasite and the host, because we're trying to understand this interaction from both those aspects. And the connection between the two is very intimate in the sense that when Cuscuta, for instance, grows in tomato fields, you cannot use herbicides or other control measures to kill it because it's intimately connected to its host. So here are a series of interactions described in this paper by Clark and co-workers in 2019. So here you can see Cuscuta. It's an obligate parasite. We can have obligate parasites in Orobanchaceae and facultative parasites also in the species, in the family. And these are all root parasites. So we have stem parasites and we have root parasites. Much of our knowledge is coming from root parasites because they're more widespread and they cause more damage. Work from many labs, Ken Shirasu's lab in the main, but also other people who then were trained by him and now have their own research programs, has shown that the lateral root development pathway was likely co-opted to make haustoria in these root parasitic plants. When we did some work with Cuscuta early on, we showed that Cuscuta may have repurposed the shoot development program for haustorium formation. And so here in these, in the Lamiales, there is a projection that the haustorium makes, and root hair development seems to be very important for this contact between the parasite and the host, and then eventually the haustorium penetrates and vascular connections are established. So we wanted to know if Cuscuta also co-opted the lateral root program into haustorium development, because that seemed to be an unanswered question. So many years ago, 2014, we had, when genomes seemed to be much more easy to obtain, we had done a transcriptome of Cuscuta and done a de novo assembly of it to identify many genes that were associated with parasitism. Then 4+ years later, Kirsten Krause's lab actually did a genome of Cuscuta campestris. So we then decided to take our transcriptome data and map it to the genome and then do an analysis on this more robust set of data, because now it was genome enabled. And so we used methods that were actually pioneered in our lab by Dan Chitwood, and we continue to use them. So I'm going to describe them once, and then I'll just say we did principal component analysis and self-organizing map clustering. So we conducted a principal component analysis and noticed that the genes that are highly expressed in the invasive tissues are separated from those that are expressed in non-invasive tissues. And we then also did self-organizing map clustering and showed that there are several clusters of genes. Let's see if I can get there. Several clusters of genes that are of interest to us, particularly this cluster 3 that is pre-haustorium enriched, cluster 2 that is Hostorium enriched, and cluster 9, which is enriched in genes that are expressed in both prehostoria and hostoria. And so let me focus first on this self-organizing map cluster 9, which has genes enriched in prehostoria and hostoria. So we took this cluster of genes and we made a gene co-expression network using these genes. And so this gene co-expression network shows us 3 major modules. I'm going to focus on 2 of these. So module 1 is enriched in extracellular domain and plant cell wall loosening type genes. And then module 2 doesn't seem to be enriched in much. There's not a whole lot of genes here. Module 3 is more enriched in response to hormone, lignin biosynthesis, and transmembrane transporter activities. So we wanted to know what some of the higher level genes might be, like transcription factors, receptors, etc., that might be regulating the expression of these genes. And so they're highlighted here. Brassinosteroid related TFs are important here, but I point your attention to these 3 TFs: a lateral organ boundary domain 4, lateral organ boundary domain 25, and a Werke 71. So lateral organ boundary domain 25 had already been studied a little bit in Arabidopsis, a fair bit, in parasitic plants a little bit. So I don't want to misspeak. In fact, most of our knowledge about LBD25 is coming from Arabidopsis, and it's expressed in developing vasculature, in pollen, but also in developing roots. So Yasu Ichihashi, who was a postdoc in my lab when we had started this work, then went on to Ken Shirasu's lab and showed that in this root parasite, Thesium chinense, LBD25 is upregulated during hostorium formation. So we looked at the first layer of interactions that LBD25 had. And this also seems to be, of the 3 TFs that we looked at, also seems to be a major hub and is connected to a lot of genes related to cell wall loosening. So I want you to hold that thought, and I'll go on and show you a couple more things. So then we focused on the clusters that are hostorium enriched, pre-hostorium enriched, and this SOM9 cluster. And we made a gene co-expression network using all of those. And now we see that cell wall loosening seems to be a major enrichment for Go terms in this module 1. Module 3 is enriched for morphogenesis of branching structures, plant organs, and module 2 is more a defense response related cluster. So now if we look at the LBD25 first and second layer connections, we see that it's connecting to both module 1 and module 3. And these are cell wall loosening and morphogenesis of a branching or organ forming structure. And so this suggests to us that LBD25 might be involved in some of the early processes leading to cell wall loosening to allow the hostoria to go in, but also in the process of hostorium formation itself. So how do you tease these things out? We cannot transform Cuscuta. So we focused on this technique that we had developed, which we call host-induced gene silencing. And so we had shown that this works really effectively in Cuscuta campestris many years ago. Now it seems to be quite a favored technique to look at interactions between 2 organisms. So the upshot is you generate an RNAi construct that is going to target a gene in the parasite, and you express it in the host. As the parasite connects to the host, there is small RNA transfer between the host and the parasite, and this enables downregulation of the parasite-specific gene. Stephen Bruno, when he was a graduate student in my lab, had called this the Trojan horse mechanism. So we used 2 different promoters to drive this Cuscuta LBD25 RNAi construct. We used 35S and also SUC2, which is a phloem-specific promoter. So what happens to LBD25 expression levels? Let's just first look at what we can tell just from Cuscuta grown on normal tomatoes. So expression goes up in prehostorial and hostorial stages for this gene. Then we can start to look at LBD25 expression levels when it's grown on these RNAi lines. So we take Cuscuta that is attached to these Higgs lines, and then we query the same gene expression. So here in wild type, expression is up in hostoria, but in hostoria that are formed on Cuscuta grown on the Higgs lines, you can see that there's substantial downregulation of this gene. What is the consequence of this? We see a couple of different things. The first thing is that while wild-type Cuscuta is growing quite normally, and only about 20% of it is flowering at 14 days post-attachment, on these LBD25 lines, up to 60% of the Cuscuta can start to flower. And we think that this might be a stress response because Cuscuta may not be able to very effectively parasitize under these Higgs conditions. And indeed, we see that fresh weight of Cuscuta is reduced when it's growing on these Higgs lines. So that's one thing. That's like a whole plant phenotype. Cell walls were implicated in these gene networks, so we wanted to look at what the response at the cell wall level was. So I want you to focus on this panel first and look at this little key that I have for toluidine blue staining of sections. So when we see reddish to purple staining, which you can see here, this is pectin and middle lamella in the parenchyma and collenchyma that seems to be stained by toluidine blue. As the Cuscuta hostoria penetrate, I want to bring your attention to the cells around the hostorium, and they seem to lose this pink-purple stain, suggesting that indeed pectins are being degraded as the hostoria are growing through. And so then you can see on the LBD25 Higgs lines, we see a couple of things. Hostoria don't penetrate deep, and in fact, the pectin layers seem to be pretty intact under these circumstances. So I want to bring you back to this network that I had shown you, where I had shown that LBD25 connects to many. So this is pectate lyase, pectin methyl esterase inhibitors, and I just want to focus on one particular pectin methyl esterase inhibitor. So here are LBD25 expression levels when they are grown on this Higgs line. And here in those same samples are the expression levels for the pectin methyl esterase inhibitors. So this is telling us that indeed LBD25 seems to inhibit pectin methyl esterase formation in these hostoria and impact cell wall loosening as the hostoria are trying to grow through. So now let's go to some other hostorium higher level phenotypes. And one thing, so one is the formation of hostoria themselves, and this more important part is the formation of searching hyphae. So as the hostoria penetrate into the host, they make these projections. These are single cell projections, and these grow between cells and contact the host vascular system. And then they transdifferentiate into xylem or phloem, allowing a continuous vascular connection between the host and the parasite. So here you can see in wild type, there's lots of searching hyphae and then connections established. But in the LBD25 RNAi lines, not only is the penetration not complete, but searching hyphae formation itself is inhibited. And here you can see this a little bit better where, they form maybe, but they aren't able to grow and elongate and get all the way to the host vascular system. So the consequence of this, of course, is the kind of hostoria that are made. So we did a whole lot of sections through multiple different wild type and Higgs lines colonized by Cuscuta, looked for successful hostorial connections where we see this connection between the host and the parasite, and then also looked at unsuccessful connections where the searching hyphae do not go all the way in and the hostoria sit more on the surface. And as you can see, so we have to look at the red. So yes, connections in wild type can be unsuccessful too. But in these Higgs lines, we see many more unsuccessful connections versus successful connections, suggesting that LBD25 expression is a key feature of successful hostorial penetration. What about just the formation of hostoria themselves? So my student Minyao, or former student Minyao, developed a system for in vitro hostorium development. So unlike other systems like in the Orban-KC, where parasite and host connections are involved and signaling is needed, Cuscuta can be pretty promiscuous and will make hostoria just on contact. So if we take Cuscuta strands, sandwich them between two layers of agar, and give them far red light, they will start to make hostoria without any host. So this is interesting because what we can do is we can grow them on a Higgs host, detach strands, and then say, what is the number of hostorial connections that you are capable of making? So we just count these numbers of prehostoria. And as you can see, compared to wild type, the number of prehostoria that these can form is highly, highly reduced. So LBD25 then seems to play a pivotal role in many aspects of hostorium development, cell wall loosening to allow hostoria to penetrate, searching hyphae development, and we think it's interesting that it's LBD25 is expressed during pollen tube growth because searching hyphae are also single cell extensions. So that's interesting. And then, of course, the formation of hostoria themselves. So it seems likely then that stem parasitic plants like Cuscuta also co-opted the lateral root program into hostorium development. This suggests that common mechanisms were involved in the root and shoot parasitic systems, perhaps giving us more hopes for devising control mechanisms that could work on both different types of parasites. So I'm going to quickly go in and show you also how we have looked at parasite and host interactions in one other way. So Moran Farhi, when he was a postdoc in the lab, did laser capture microdissection, where he essentially used a laser to cut out the region of Cuscuta and collect those tissues, and then also sample the host right around the Cuscuta and look at those samples using RNA-seq. So I won't belabor the details. I already described how we used principal component analysis and self-organizing map construction. So I will just show you what he found when he looked at early, intermediate, and late stages of hostorium penetration. And I will focus on the Cuscuta first and then go into the host. So in the early stages of Cuscuta penetration, he found a cluster of genes that were enriched. And in this, he focused on this transcription factor HB7. He looked at genes that were in a cluster that was expressed in mature hostoria, and he focused on this pectin methyl esterase inhibitor. And the final one, I'm going to tell you, this is something that's expressed early and late, and he found an ERF1, an ethylene response factor 1. And remember, these are Cuscuta genes. So he then went in and made Higgs lines for all of these. So I'm going to show you some of these. And this is wild type Cuscuta colonization. You can see penetration, searching hyphae, vascular connections. So HB7 Higgs lines, essentially just the hostoria just sit on the surface. There seems to be some kind of hyperplasia in the host in response to this kind of attachment. And we see very few successful hostorial connections. The same thing for the pectin methyl esterase inhibitor, again, hyperplasia, but most of the hostoria seem to sit on the surface, and then the same thing for ERF1. So I want to move on now to show you what we saw in the host LCM samples. Let me see how am I doing for time, Dan? Do you have 5 more minutes?
We do have 6 minutes. We usually have a hard time on it, but you are coming up against a lunch break. So a few more minutes and we can have questions.
Okay, wonderful. So then in the host LCM samples, there were again early, and in these, there was a pathogenesis related one, an NLR. And here is, oh, and I wanted to mention one more gene because I want to spend time on that, NORC16. So very quickly, the PR1 CRISPR showed us that Cuscuta becomes like hyper invasive when host PR1 is CRISPRed. The same thing for this NBS LRR. Cuscuta becomes hyper-invasive, makes these massive connections. And then finally, I want to talk to you about WRKY16, which is very interesting. This is a gene that's upregulated 4 days post-attachment in hosts. It's upregulated in many different cultivars. A Cuscuta receptor has been described by Marcus Albert, a collaborator of ours, and we've, worked with him on this and other aspects. And this Cuscuta receptor seems to track the WRKY16 expression levels. So we did WRKY16 CRISPR and found something very interesting. I'm not going to, focus too much on the fact that these plants look smaller, but they're developmentally at the same stage. What I'll show you is that in these WRKY16 CRISPR lines, there is massive lignification in the stem cortex. And of course, then this really inhibits Cuscuta penetration. With that, I will skip to my summary slide and skip over a couple of slides that I had in there. So the LCM data reveals that there are several key genes involved in Haustorium development and also genes that are upregulated by the host in response to parasitism. HB7, a negative regulator of ABA response, ERF1 and PMEI are involved in the Haustorium development and parasitism process. And then CRISPR mutagenesis shows that the host upregulated genes, PR1 and this NBS-LRR, play a key role in resisting Cuscuta attack. And WRKY16 probably has a role in suppressing lignification in response to Cuscuta attack. So I'm going to stop right there and leave the summary slide on there and be happy to take any questions you may have.
Thank you, Nilima. That felt like zooming in through anatomy and gene expression networks simultaneously to get at the root of all this. Please raise your hand if you have questions, and we will start with Dr. Dubrovsky.
Yes, hello. Nilima, thank you very much. That's beautiful work.
Thank you, Joseph. It's so good to hear from you.
Thank you. And yeah, I'm very happy to hear you. And my question is related to the classification. Let's say you say that this is lateral root development co-option of the program of the lateral root development. However, we know that these are not lateral roots, and actually the adventitious roots have a bit different program. Wouldn't you think that this is an adventitious root developmental program or pathway co-option?
I think it could be totally true that it's an adventitious root development program. But, I am coming from the more Kaplan-esque idea where Don said, so I'm going to show you a slide and ignore everything else on this slide and just look here. So the fact that these are not shoots, because lateral shoots arise superficially on the stem. Mm-hmm. These arise internally. They go through several cortical layers to come out. Okay. So I'm calling them roots.
I see. Yeah.
And, let's just not get into, it's not semantics. I totally hear you that adventitious roots and lateral roots can be very different. But at the core, when they are roots, they have some common programs.
Okay, okay.
And that's where I'm going with this.
Yeah, I see your point. And another small question, at what moment during haustorium formation the meristematic activity stopped? Let's say, is it correlated to penetration, after penetration, before contact with the xylem of the host, or there are some other observations on that?
So we think that there is some meristematic activity that is happening and happening until fairly late, because one of the earliest demonstrations that we had of Higgs was when we tried to downregulate the Cuscuta STM gene. And that we know is involved in maintaining indeterminacy. And we saw that when the Cuscuta STM was downregulated, it did impact both haustorium formation and searching hyphae formation. So that's where we had come from. Well, maybe it's the shoot program, but it could just be meristematic activity in the shoot, because these are shoots. I don't know how we could say exactly when meristematic activity stops. I don't know how we would assay that. If you have any ideas, I'm more than happy to listen.
When you have, let's say, modified hairs, root hairs, well, haustorium hairs.
Yeah, see that there is no indication for that in the stem parasites. All of that modified hair data is coming from the root parasites, where we know that the root hair program is essential. We actually don't see that here. And in fact, Cuscuta doesn't make roots.
I don't know. Yeah.
Yeah. Yeah.
Very interesting. Thank you very much.
Any other questions for Nilima? You can raise your hand. We have Nicholas Sacchini.
Hi, a very nice talk.
Hi, Nicholas.
Really nice. Hi, how are you? I have a question. I'm really surprised with the NBS LRR gene. I'm thinking, the parasite plant is injecting some kind of protein inside the cytoplasm that is recognized by the RG, by the receptor or something like that. What do you think about that?
It's highly likely that in the penetration process, there is exchange of molecules between the host and the parasite. We have not specifically studied that at all. So all I can tell you is from our CRISPR data, we see that these genes are very important. We had another LRR that we made CRISPR of, and we could never actually get it to survive in the greenhouse. So we have very few seed left. And, I feel like that may be a more generalized response regulator. So we will need to study that perhaps in more controlled conditions where there aren't other opportunistic pathogens. But yeah, there's no doubt that there's considerable exchange of information between the host and the parasite, is how I will answer your question.
Okay, and I think, I'm not sure, but this is maybe the first example of a R gene receptor of this kind receiving a plant and not a pathogen factor, or maybe I'm wrong, I don't know.
We have another CCMBS LRR that's a host-specific one, but I had too much data, so I took that part out. So maybe you should watch out for that. It's kind of going through some revisions for publication. Yeah.
Thank you, Nilima. That was a wonderful talk. It was great to see that work after so many years come to such fruition.
Yes. Thank you, Dan.
Thank you
for getting us into this very interesting mode of analysis, because we find that doing. Let's see. Oh my God. Okay, stop share. There we go. we find that, using gene expression and these kinds of analysis methods and then using networks to inform us on choice of genes has been very fruitful for us. Thank you all for listening. I hope I didn't take up too much of your time from lunch. Ulysses was saying, well, it's lunchtime. And I said, never stand between a person and their lunch. So
we're virtual now, so lunch is anytime.
I know. Take care.
Thank you so much, Nilima.
Thank you all.
Like we said, we now have virtual lunch until 2:15 p.m., which we will come back for flash talks. And I want to remind everybody that if you would like to organize the next meeting in 2 years for the Mexican section, please come to the organizational meeting, 5 to 7 p.m. Mexico City time on this link. Hopefully the pandemic will be over in 2 years, and hopefully there'll be a wonderful meeting at some great location in Mexico. And you can be a part of that planning process. So we will break for lunch now. We will start the flash talk shortly. They will go from 2:15 to 2:45. But before that, I just want to again remind everybody that we have a business meeting from 5 to 7 p.m. Mexico City time today. Anybody who has participated is welcome to come, but it will be mostly to find the organizers for the next SMB ASPB meeting in 2 years. So if you're interested in organizing the meeting, please show up today if you can, 5 p.m. Mexico City time at this link. We will start the Flash Talk session number 5 now. You can find the presenters and their abstracts on the website. And Sophie and Kamoon will be our next speaker at 2:45 p.m. See you then.
Hi everyone, I'm Rosanna, a postdoc from the lab of Lucia Colombo, settled in Milan. And today I'm going to show you some results on the role of VOX9 STIMPI during ovule development in Arabidopsis thaliana. The topic of our lab is to understand the factors involving the development of ovules, since ovules are the precursor of seeds. In particular, we are studying the role of the homeobox domain 9, called also STIMPI, during ovule development. It is known, as you can see here from this picture, that is expressed in the floral structures, ovules included, but nothing is known about its putative role in this structure. To further characterize its role, we decided to first check the expression of VOX9 during ovule development. And as you can see here, VOX9 is expressed throughout ovule development in the funiculus of the ovule and transiently also in the chalaza and in the nucella. Then we analyzed 2 mutant backgrounds for this gene, an overexpression line named STIP-D and a knockout mutant line named STIP-2. As you can see here in this panel, at the end of ovule development in the wild type, these 2 structures here, named integuments, fully enwrap the female gametophyte. In contrast, both the 2 mutant backgrounds presented defects in integuments development, since they do not fully enclose the female gametophyte that is somehow uncovered. The next step was the identification of new putative VOX9 targets. A mutant showing similar defects in integuments development is inner-outer-5. This gene encodes for a YAB protein, and interestingly enough, VOX9 and INO share a similar expression pattern in the ovule. That's why we checked by in situ hybridization the expression of INO in both the 2 mutant backgrounds. And as you can see here, overexpression of VOX9 lead to a deregulation of INO domain of expression. It is no longer restricted in a certain domain of the ovule. In comparison, in the STIP-2 mutant, we could see that the expression of INO is barely detectable. And these results were confirmed by quantitative real-time PCR. So, in conclusion, with our results, we could confirm that a proper expression of VOX9 is important for a correct ovule development, and inner-no-outer is a good candidate as VOX9 targets. And thank you all for your attention.
Hello, everyone. I'm Miriam Huerta, and I'm happy to present my postdoctoral project titled Plant Membrane on a Chip Platform to Study COP1 Transported Function. In Arabidopsis thaliana, around 18% of the predicted proteins appear to contain 2 or more transmembrane domains. Many of them are related to transport proteins, has been characterized in silico, but not their function. There are some techniques that help us to study the membrane transport proteins. However, not all of them help us to study all the proteins that we want to. That's why we are looking for more options to study membrane transport proteins. Our lab is an expert in form membrane on a chip, which consists in take portions of the cell membrane in the form of blebs, and that with the help of fusogenic liposomes provoke their disruption and form hybrid-supported lipid bilayers, in this case with plants. To study copper transporter 1, which is a protein involved in plant growth and pollen development in Arabidopsis thaliana, but their functions has not been completely understood using the previous techniques. To overcome this, we overexpress COP1 in Arabidopsis protoplast, and as you see, the protein is on the membrane. As a control, we use GFP. Then we form the supported lipid bilayer on top of a conductive polymer, and using FRAP microscopy, we determine the formation and functionality of the membrane. Calculating the diffusion coefficient, in both cases, we observe that a membrane keeps these properties. Next, the membrane was formed on top of organic electrochemical transistors and do some electrochemical impedance spectroscopy measurements. Here, the membrane forms a resistance, and this, after the copper stimulus, this resistance could be changed because the transporter is open. Here is an example of a body plot with the impedance of the epidot, the supported lipid bilayer, and the copper concentrations. After feeding the data, we got normalized resistant membrane. As we observe, in all of the cases, after copper stimulus, decreased the resistance of the membrane, but it's more evident in the ones that we overexpressed COP1. In summary, our plant membrane on a chip helps us to analyze COP1 transporter uptake, and this platform could be suitable for studying other plant proteins involved in different processes in the plant. I want to say thank you to Professor Susan Daniel and my lab mates at Cornell University, as well as our collaborators at Cornell University at Linköping University in Sweden. Thank you for your attention.
Hello, I am Albert. I will present you the research project Identification of Sweet Gene Family in the Physiology Genome and Comparative Analysis of their Expression Profile specific to mycorrhizal and rhizobial symbiosis, carried out at NS León, UNAM. In order to survive, plants create symbiosis with microorganisms such as mycorrhizae and bacteria. From this they obtain nutrients, and as compensation, plants provide them with sugars to survive, since they are important molecules that help to successfully accomplish different metabolic tasks given enough energy. For this reason, we studied the sweet gene family, which are important sugar transporters located in the cell membrane, composed normally by 7 transmembrane proteins characterized in a wide range of species, but has not been analyzed in the legume Phaseolus. Here, we perform a genome-wide analysis to identify sweet transporter genes member in the Phaseolus vulgaris genome and examine the expression profiles of PB sweet genes during mycorrhizal and rhizobial symbiosis. We found a total of 24 genes comprising the sweet family in Phaseolus, classified into 4 clades, in addition to which most are located on the long arms of the 11 chromosomes of this species. Only chromosomes 7 and 10 had no genes. Structurally, most of the genes contain 6 exons H1. These have 3 motifs belonging to duplicated domain in each of the genes. In expression profiling of all tissues, we saw the highest overexpression in genes 1, 6, 14, and 20, while genes 22 and 23 were overexpressed during symbiosis with mycorrhiza and rhizobia respectively. In conclusion, transcriptomic analysis revealed that shared and unique PB sweet genes were upregulated during arbuscular mycorrhizal and rhizobial symbiosis. Overall, the systematic analysis of the PB sweet gene family provides valuable information for further study on the biological roles of sweets in various Phaseolus tissues during diverse biological processes, including Phaseolus symbiosis with mycorrhiza and rhizobium. This work was supported by the GAPA-POPIT UNAM and CONACYT. Thank you for watching.
The regulation of protein activity is fundamental, and part of this regulation resides in their localization within the cells. This includes the secretion of proteins into the extracellular space. In order for protein trafficking to take place, cells have several delivery systems in which cells recognize certain molecular labels in the protein sequences themselves. One of these signals is the signal peptide. A signal peptide is a sequence of between 16 and 30 amino acid residues, generally hydrophobic, found at the amino end of the protein that contains it, whose main function is to direct the translation of the protein towards the endoplasmic reticulum, so that from there it is translocated to the Golgi apparatus, from where it will be transported in vesicles towards the plasma membrane in order to be secreted. Nadarj is an H-type thioredoxin, subgroup 2, from Nicotiana alata, recently characterized as an essential protein in self-compatibility. A peculiar feature of Nadarj is that, although it lacks signal peptide, transient expression assays of Nadarj GFP in onion epidermal cells and in leaves of Arabidopsis thaliana and Nicotiana benthamiana indicate that Nadarj possesses the information to guide its secretion. The N-terminus of Nadarj contains the motif responsible for its extracellular localization, the N-beta motif ALA17 to PRO27, which contains atypical features as a signal peptide. The localization of the N-beta motif within the primary structure of Nadarj and its hydrophilic profile suggested a protein secretion pathway independent of or alternative to the and Golgi apparatus. However, cellular evidence shows that it does use them. This indicates that the N-beta motif represents a novel signal directing protein secretion in cells. In order to determine if the ENS motif might be a signal, transit peptide-like sequence in proteins that, despite lacking any of this typical sequence, their localization is extracellular, we searched proteins with this motif, or a similar one. Then, from the obtained sequences, the synonym proteins, same sequence, different accession number, were eliminated, and the rest were analyzed according to the source, function, and cellular localization. The position in which the ENS motif was located within each primary structure was also analyzed. All sequences were divided in 4 quarters. Preliminary data suggest that when the ENS motif is located in the first quarter of the protein, and maybe the second one as well, most of the proteins appear to be secreted or membrane associated. Apparently, none of these proteins contain a signal or transit peptide suggesting a correlation between the presence of the ENS motif and a particular localization, membrane or secreted. Overall, our data suggest that determining the presence of the ENS motif and its position within the sequence might be used as a tool to predict a putative cellular localization of the proteins that contain it, indicating that this motif is a novel signal transit peptide.
Hi everyone, my name is Eduardo Cázares, and this is a project called Fusarium verticilloides chitinase modifying proteins may play a role on fungal invasion in the tripartite interaction maize Fusarium verticilloides and Bacillus cereus. Maize is one of the most important cereals in the world, being Sinaloa the main producing state. Maize is susceptible to various diseases caused by phytopathogenic fungi. One of them is Fusarium verticilloides, which causes stem and root rot. Bacillus cereus B25 can produce antifungal compounds such as siderophores, antibiotic, and 2-chitinases. We also demonstrated that these chitinases reduce germination of Fusarium verticilloides conidia, having effects on hyphae such cell wall thinning and abnormal jet tubes. Likewise, maize is capable to produce chitinases and induce plant defense when invaded by fungal pathogens. However, Fusarium verticilloides produces chitinase-modifying proteins, or fungalicin, which separate the chitin-binding domain and the catalytic domain of the plant chitinases, causing to lose their ability to bind chitin and hydrolyze it. In our proposed model of tripartite interaction, B25 chitinases might not be modified by fungalicin because they don't have the protein domain that fungalicin recognizes. It has been postulated that the bacterial chitinases may degrade chitin in the fungal cell wall and elicit plant defense response. This work aims to demonstrate in vitro that fungalicin does indeed modify plant chitinases but not bacterial chitinases. For the in vitro modification, a fungal protein extract was obtained from hybrid white cord seeds of Ascrogarañón inoculated with Fusarium verticilloides conidia. The fungalicin assay test was carried out by assaying separately plant and bacterial ketonesis with the fungal protein extract. We also established a tripartite interaction with paper towel assay with for treatment using maize seed, Fusarium verticilloides conidia suspension, and B25 powder formulation spores. We found that maize ketonesis were modified in the presence of Fusarium verticilloides protein extract, while bacterial ketonesis remained intact. We also found that Fusarium verticilloides may produce another protease, possibly a subtilisin, which acts together with fungalicin to modify the maize ketonesis. We observed the accumulation of both maize ketonesis in root of cell tissue, with different levels of accumulation between treatments. So, we postulate that the fact that bacterial ketonesis can attack Fusarium verticilloides from very early stages, inhibiting its growth in maize tissue, also induces the plant defense response due to the ketonesis accumulation, which could lead to an increased tolerance or resistance to infection by fungal pathogens.
Hi, I'm Sergio Hernandez, and today I'm going to talk about determination of heat stress tolerant wheat plants field grown in the Yaqui Valley, Mexico. Heat stress is an environmental stress that limits plant growth, metabolism, and productivity. The WMO stated that it's been an increase of 1.1 degrees on global average temperature in the last century. And in other research, it's estimated that per 1 degree increase in temperature causes decrease in global wheat production by 6%. Particularly in Mexico, the Yaqui Valley in Sonora is the main producer of wheat in this country. However, due to the high temperatures on this state, the heat stress might cause, among others, yield reduction. So that's why it's very important to identify new genotypes tolerant to heat stress to reduce yield losses and enhance the production of wheat in Mexico. That's why we aim to select tolerant and susceptible wheat genotypes based on agronomical and physiological data for further biochemical and molecular analysis. We use 25 genotypes from CIMMYT and a control flow minifab, and at 2 different planting dates, December 2020 as a control and January 2021 as a late heat stress. The field design used was a randomized complete block design, and the parameters evaluated yield, height, chlorophyll, and fluorescence. All experiments were performed at the Norman Borlaug Research Field at Cajeme, Sonora. Heat stress reduced yield in all genotypes when compared with the control, but in a different fashion. That's what allowed us to identify first susceptible genotypes when they had a yield reduction compared with the control higher than 700 grams, identifying 6 genotypes. And on the other hand, 4 genotypes tolerant when they had a reduction when compared with the control lower than 700 grams. The difference between the control and the late planting were higher in the latest at both vegetative and reproductive stage. Heat stress also decreases the height in all plants. As far as for the chlorophyll concentration, genotype 24 had the lowest and 22 had the highest. And as far as for the fluorescence parameters, we only identify differences in genotypes 17 and 22. And being when it's under heat stress, it increases the amount of light absorbed by the photosystem 2, maybe as a compensation for the lack of electron transport in the rest of the transport chain. And on the control conditions, it was quite the opposite. So based on this information, we identified 10 out of 26 genotypes, 6 susceptible and 4 tolerant, which needs to have further biochemical and molecular analysis. Here you have our information for questions.
Hello, my name is Miriam Rodriguez, and today I am going to tell you about suppressor of RNA silencing encoded by pepper huasteco yellow vein virus. Gemini virus replicate and transcribe their genome from a viral mini chromosome. The viral infection triggers the plant defense response, such as gene silencing. The silencing process can act post-transcriptional level that direct mRNA degradation, and at transcriptional level by RNA direct DNA methylation and viral chromatin modification. to restrict viral replication, transcription, and proliferation. As a counter-defense, many plant viruses encode suppressors of RNA silencing. In this study, we analyzed if pepper huasteco yellow vein virus has silencing suppressors. PHYBV is a virus of agronomic importance because it affects pepper and tomato crops in Mexico and Central America. Post-transcriptional gene silencing assays were performed in Nicotiana ventamiana 16C plants that express GFP. We co-infiltrate plants with a mix of GFP plus viral gene. If this viral gene is a silencing suppressor, the infiltrate area expresses GFP. We found that only the expression of REP suppressed post-transcriptional silencing. Nicotiana ventamiana 16C TGS line contains a GFP transgene, transcriptionally silenced. To investigate if virus and its proteins are able to suppress transcriptional gene silencing, we inoculate silenced Nicotiana ventamiana 16C TGS plants with PHYBV or recombinant PBX vector expressing PHYBV encode genes. Inoculated plants were evaluated under UV light 10 days. PHYBV infection induced transcriptional gene silencing suppression, allowing the expression of GFP. Also, the expression of REN is able to suppress transcriptional gene silencing. The level of DNA methylation in the 35S promoter in plants infected with either PHYBV or expressing REN was significantly lower than found in mock-inoculated plants. In summary, PHYBV encodes two silencing suppressors, REP and RED. Thank you.
I am Marina Lopez. I am going to present the work titled Defective in Inducer Resistant 1 Play a Key Role in the Progress of Infection Tree and the Organogenesis of Nodules in Phaseolus vulgaris. Common bean is important for developing countries because it is the mainly protein source for them. Furthermore, on ground, common bean interacts with Rhizobium tropici, forming a new organ called nodules, where the bacteria fix nitrogen. For this, in a previous work by transcriptome analysis, we identified some genes that change its expression pattern during novulation. One of them, DIR1, a lipid protein transporter that mobilizes glyceraldehyde 3-phosphate for activation of systemic response. In this work, we studied the phylogeny of the three genes that encoded to DIR1 in Phaseolus vulgaris genome and nodulation phenotype of two clones with silencing of one of them. Firstly, we compare the protein sequence of DIR1 of Phaseolus vulgaris with the other legumes and Arabidopsis thaliana. The result shows that the three genes of Phaseolus vulgaris are clustered with the legumes independently to Arabidopsis. Then, to determine DIR1 role in ovulation, we inoculate Rhizobium tropicibus in roots with silencing of this gene and find that the silencing does not affect the infection tree number, in the same word infection events, but interferes with its advance to the cortical cells. The effect in the infection tree advance diminishes the nodule numbers at 10, 14, and 21 days post-inoculation, but the most amazing effect caused by the silencing is more one infection event at the same place. Also, the nodules with silencing are smaller or with a multinodular phenotype. In addition, these nodules have defects in vascular tissue development and symbiosome differentiation. However, any alteration does not modify the nitrogen fixation. In conclusion, the IR1 acts as a positive regulator of novulation and prevents the occurrence of more than one infection event at the same site. Thanks.
Hello, my name is Laura Gonzalez and I will talk about the project Identification of Cell Interacting Pathways, a maize protein involved in immunity. In Mexico, with the 8th place in production and the 10th in export, maize is a representative crop due to its economic, social, and cultural importance and has different uses. The mutant Ligulus naro has been identified with developmental defects and is supplemented with a modifier called salt of Missouri 17 or low temperatures. Salt is an orthogonal of the EDR4, which is involved in immunity confirmed by chitin treatment in maize, where induction of salt and genes related to resistance are seen. Nicotiana bentamiana they determine the subcellular localization of Sol B73 in the periphery and the nucleus of the cell, and Sol Missouri 17 only in the periphery of the cell. Therefore, the objective is to see difference in the location of the protein in different genetic backgrounds of maize, generate a specific antibody against Sol to search for proteins that interact with it. For this, specific polyclonal antibody was produced and purified with a specific peptide. Antibody was tested in Western blot and showed specificity. In addition, we are also doing immunolocalization in maize tissues, and as preliminary results, a signal has been detected in male floral primordia. Finally, what remains to be done is to carry out more immunolocalization and standardize the conditions for immunoprecipitation assays to determine the protein complex that interacts with salt. Thanks for your attention.
Hello, my name is Ricardo Ortiz Leano. I will talk to you about the work called change induced by lead in root system architecture of Arabidopsis seedlings are mediated by PDR2 LPR1 phosphate dependent way. The shape and special configuration of the root system is called root system architecture. This presents plasticity as an adaptive strategy to cope with abiotic stress. Lead is one of the most hazardous heavy metals that disturbs development of root system architecture in plants. We found that root system architecture are 800 micromolar of lead is similar to root system architecture under phosphate starvation conditions, with short primary root and increased lateral root density. This makes us to suppose that lead and phosphate starvation stress use the same genetic signaling networks. To find if lead and phosphate are interacting in media culture, we made an in silico analysis, and the results show that phosphate solubility decreased as lead addition increased, suggesting that lead limits phosphate bioavailability. To evaluate phosphate starvation stress in roots, we use construction of PATP2, that is a high affinity phosphate transport fused to Goose reporter gene, and we found that phosphate starvation treatment increased signal of Goose reporter gene. Meanwhile, lead treatment show poor blue colored roots. This indicates that lead stress induced ATP2 downregulation. In order to evaluate if lead stress and phosphate starvation stress share common genetic compounds, we test 3 mutants insensitive to phosphate starvation stress. We use STOP1, LPR12, and LPI3, and our results show that STOP1 had similar root as wild type, demonstrating that STOP1 gene is not necessary for lead response. Also, we found that mutants LPR12 and LPI3 were insensitive to lead, demonstrating that change in root system architecture of plants growing at medium supplemented with lead are dependent of LPR12 and LPI3 genes. we can conclude that change in root system architecture is regulated genetically by LPR12 and LPI3 genes. Under lead stress, activity of ATPT2 is downregulated, avoiding phosphate accumulation, and similar change in root system architecture induced by lead and phosphate extravation are due to similar mediated response by PDR to LPR12 pathway. Thanks for watching. See you later.
Good afternoon, all. So we will have our next keynote talk. So I'm very honored to introduce Professor Sophie Camoun from the Sainsbury Lab in the UK. And he's going to talk to us about how to trick a plant pathogen. Very welcome, Sophie. Please, if you can share your screen.
Thank you. Thank you, Stefan.
The floor is yours.
Thank you. Let me, all right, I suppose my screen now is
yes.
visible. Okay, perfect. Excellent. Thank you, Stefan. Thank you to Ulysses, who invited me, and thanks to all the community. It's really great pleasure to connect with you, although it's from a distance, but it's really pleasure to connect with the Mexican community. I'm a bit tired of giving Zoom talks, to be honest, but with your community, I'm always excited to speak to you and interact with you, and I hope the next time would be in person. So let me get going with the science. So what I will talk about today is really our work on the immune receptors of plants, in particular the so-called NLR immune receptors. And I will give you an overview of our current knowledge of these receptors and some of the work we're doing in my group. And I will talk with some applied perspective on how we can use these receptors for bringing disease resistance to our crops. So let me start by telling you essentially a concept that is very old in plant pathology. I think plant pathologists noticed this as soon as they started isolating plant pathogens, that in general, those plant pathogens tend to be host-specific and infect only limited number of hosts. And now we know that the reason for that is because plants have a very effective immune system, and they actually can actively fight off most pathogens. So disease really in plant pathology is the exception. If you walk around in, for example, in Ulysses' botanical garden, beautiful botanical garden, you see that most plants are actually really healthy. That's because they are really able to fight off a lot of these microbes with their immune system. And the way this works is through essentially this series of receptors that plants have. But before I get to that, I should really get to the side of the pathogen, which is really how a lot of our work has started from studying pathogens and studying how they affect plants. And this work led to the concept that these pathogens, and now we know that this applies to bacteria, fungi, even insects, nematodes, etc. A lot of these pathogens, maybe all of them, are secreting molecules, often proteins that we call effectors, and these effectors will perturb plant processes in many different ways. They will perturb plant processes in the apoplast, and those are apoplastic effectors. Often they are inhibitors of plant hydrolases that are contributing to defense and immunity. But some effectors, quite few of them, get translocated inside the plant cells, and they alter plant processes intracellularly inside the plant cell. But pathogens don't stay idle, and they are able to detect these effectors, and they detect them through a variety of receptors that plants have. There are two major classes of receptors. There are the ones that are on the cell surface, anchored in the membrane. These would be the PRRs, generally called PRRs, pattern recognition receptors. They would detect the apoplastic effectors, and they can also detect conserved molecular patterns from the pathogens, like flagellin from bacteria. And then there are the intracellular receptors, which will detect the effectors that are translocated inside plant cells. They can detect these effectors either directly by binding them or indirectly by detecting the perturbations that these effectors cause. And this is the immune receptors I will talk about, and they are known as the NLRs, previously known as MBS-LRR. And I should add also that NLRs are present in all types of organisms. They even now reported in bacteria, but they're certainly present in animals. We have NLRs, and many other organisms use NLRs also to fight off pathogens or to detect pathogens. So for those of you who have more of a plant breeding, perhaps angle to this work, the R genes that are generally defined genetically as being single dominant genes that are segregating in populations and contribute resistance to pathogens. Generally, it's very specific resistance. The R genes are the often, not always, but often they encode for these NLR proteins. And we recently compiled a database of all the experimentally validated NLRs, and this is now about 500 plant NLRs that have been experimentally studied in one way or another. So it's a very heavily studied family of plant proteins and very diverse in plant genomes. So this is a bit of a description of how the NLRs are organized biochemically. These are multi-domain proteins that have a central NB-ARC domain. This is essentially the ADP-ATP switch domain, which when activated would alter the conformation of these proteins. And in plants, the C-terminal domain is a leucine-rich repeat domain, but the N-terminal domain tends to vary, and it's primarily coiled-coil domain or the so-called TIR domain. And now we know that these two domains, NLRs that carry CC or TIR, function through completely different mechanisms. I should add that we like to use Nicotiana benthamiana as an experimental system to study NLRs. This is very popular for studying NLRs, and the reason is that you can typically reconstruct the interaction between an NLR and an effector by co-expressing them in a benthamiana leaf using the agro-infiltration assay. So you can see here in this leaf that in the bottom right side, we have matching NLR and effector, and that gives you the so-called hypersensitive response, which is a cell death response and is our primary readout for activated NLRs. And if one of the components is missing, you cannot see this immune response. And you can also see the pathogen resistance here. This is with the potato blight pathogen. And on the right side, again, you can see that the pathogen is not able to infect when this NLR matching that particular pathogen is expressed in the benthamiana leaf. All right, and then what happens is that when these NLRs are activated, we have this fantastic model that is now really the consequence of cryo-EM technology and the capacity to get the structures of macromolecules like these complexes here. And now we know that when these NLRs are activated, we have the oligomerization of these NLRs into either a tetramer, as in the case of these TIR NLRs, or into a pentamer, 5 different proteins in the case of the CCNLRs R1. And then this oligomerization is actually what enables these activated NLR complexes, which we call resistosomes. This is what enables these resistosomes to cause the hypersensitive cell death. And this model is very similar to the mammalian NLRs, where they are also known to form oligomers of this type, and these are known as inflammasomes or apoptosomes. So this is really a massive advance in our understanding of NLR biology. It's very recent, as you can see here. It dates back to work by Gigi Shai and Jiamin Zhu, 2019 for ZAR1, and then additional papers with the TNLRs in 2020. And it's really game-changing, and because there was a lot of discussions about how NLRs activate immunity, and there was always this paradigm that there will be a classic signal transduction pathway activated or underpinning activated NLRs. But in reality, the model is a little bit different because now we think that in the case of the CC NLRs, ZAR1 being the sort of the prototype where this is well understood now, when we have an activated form, the resistosome, this results in essentially the N-terminus alpha helix flipping out and forming this funnel-like shape, if you like, structure that is thought to form a pore inside the plasma membrane. And this is functioning as a pore which is perturbing the plasma membrane in itself, but is also now more recently shown to function as a calcium channel, maybe a channel for other molecules. And that activity, the activity of inserting into the membrane and forming this pore-like structure or channel-like structure is critical for the activation of the cell death response. So it's not really functioning as a classic kinase, let's say, pathway or a classic signal transduction pathway. It's really functioning as essentially a structure that is associating directly with the plasma membrane and activating the cell death response through perturbations of that membrane or through the channel-like activity. So I'll get back to this model later, but before we get to that, and to illustrate also the diversity of the way by which these NLRs function, this is the case of the TIR NLRs, which function completely differently, because in this case, the TIR domain is actually an enzyme, and this enzyme has NAD hydrolysis activity, and that NAD hydrolysis activity, that NAD hydrolase, which is only activated when the NLR oligomerizes into a resistosome, that activity is essential for causing the cell death. It's not fully clear how the metabolites that result from the NAD hydrolysis are activating cell death, but this activity is essential. But you can see here that this is totally different from how the CC-NLRs function. So if you're interested in NLRs, please keep that in mind, that they have a lot of different activities and they function through a lot of different ways. And I'll also get back to this later on. Okay, so now I'm going to switch to our work, and I won't start with the end, because the model we have developed over the last years is shown on this slide, and it reflects the evolution of NLRs from singletons, if you like, single proteins that can do both businesses that are the main activities of NLRs, detecting the effector and activating the immune response or the hypersensitive cell death. So this activity in many NLRs is carried by a single protein. However, the majority of NLRs have diversified and specialized into either sensor NLRs or helper or executor NLRs. So this is the terminology we now use for this functional specialization of these NLRs. And we like to call this an asymmetrical evolution of NLR proteins, because the sensors and the helpers have really diverged from each other in very dramatic ways. So the sensors are now essentially specialized to detecting effectors. And they can do this directly by binding them, but they can also do this by integrating domains, which are also often resulting from the targets of these effectors. And these integrated domains would then bind the effector and detect it. This is a fascinating model that I will get to later on, which is basically the NLR or the plant turning the table on the pathogen. These effectors have targets, but these targets can also integrate into NLRs and now become baits or decoys for the plant to detect the NLR. The helpers, on the other hand, they retain the classical NLR architecture. They resemble quite a lot their ancestor, but they don't directly detect the effectors. They rely on the sensors to detect them, but they can still do the business end of causing the hypersensitive response and activating immunity. So this is kind of the end of actually my story, really. This is the model we reach at the end. So now I'm going to sort of take you through the research that led to this model, and you'll see at the end why this evolutionary model is actually very important for understanding how NLRs function. So first of all, let me introduce a very complex NLR network that we discovered about 5-6 years ago, thanks to the pioneering work of my student at the time, Shihang Wu, who is now at Academia Sinica. And what Shihang discovered is that many of these R genes, these classic R genes that are found in solanaceous plants, potato, tomato, and what have you, these classical R genes are actually specialized sensor NLRs. They cannot activate the immune response or the hypersensitive response without the help of these so-called NRC helper NLRs. And you can see here that this system is quite complex because these NRCs are redundant. This is actually a simplified version of what the network is like. Some of these NRCs are redundant. For example, the R gene Rx, which was cloned by David Bolcombe as a virus detecting R gene, can function with NRC2 or NRC3 or NRC4. These are equally redundant NRC helpers that can work with this Rx gene, and so on and so forth. So like RPI-BLB2, on the other hand, is against Phytophthora infestans, is not redundant, but functions only with NRC4. So this network is quite fascinating because it's very complex, and it's also phylogenetically structured. So all these NRCs are actually phylogenetically related and belong to this one clade here. Let me get the pen. I think it's going to be better with the pen. So you can see here that the NRCs are all in this one clade. So these are all the helpers. And all the sensors, everything in this clade, every NLR in this single clade is always dependent on an NRC. So it's quite remarkable. Every single one we tested so far is always dependent on an NRC. So it does look like this, at some point in evolution, there was probably a singleton that duplicated into a pair of NLRs. One became a sensor, the other one a helper. And then that pair expanded into this dramatically complex network. In some species, some solanaceous species, tomato, Nicotiana, as examples, this network became half of all the NLRs of that particular plant. And this network expanded around 100 million years ago, and now is working against literally dozens of different pathogens. So this actually brings up an interesting question, because why do we have a complex network like that? And why is it redundant also? Why is there this redundancy? How would that evolve? I mean, redundancy is always confusing in biology, because you would think that if you lose one component, then the organism would do just fine without it. So why do we have redundancy here? And this slide is illustrating what we think are the two major reasons why we have redundancy. This is a little bit interesting because these are concepts that are very heavily debated in biology, and I think it's kind of fun for us to get involved in this work. So first of all, robustness. If you're redundant, you're robust. So if you lose one component, if one component is somehow perturbed or attacked by a pathogen, then you still survive. You still can function. The immune system in this case would function. And the second reason is evolvability, that by uncoupling the pathogen recognition from the signaling, your system becomes a lot more evolvable, in the sense that there's a lot more capacity for acquiring new activities. For example, the sensors can acquire new domains and so on. So let me briefly take you through some of these. Oops, what's happening here? I'm pressing on the wrong way. Things got messed up. Okay, now here we go. So let me take you through the first part, which is really the robustness, and illustrate to you why robustness or why redundancy in these helpers can actually be useful to them. we made the hypothesis very early in this project that if we have a network structured in this way, this is what is known as a bow tie network, then the nodes here, the hubs, are going to be essentially the weak points of this network. So if you're a pathogen and you attack one of these hubs by suppressing it, for example, through the activity of an effector, you're going to really get rid of a lot of NLRs. So perhaps the duplication of these NRCs is a consequence of pathogens suppressing these helpers or these hubs or nodes of this network. So we went on and screened for a lot of effectors. I don't need to really take you through the whole thing, but there are two effectors I want to highlight. We published this recently. One is from Phytophthora, the potato blight pathogen. Another one is from the potato cyst nematode. So these are both potato and solanaceous pathogens. And these two effectors are able to suppress the activity of these NRCs, even when we express them as autoactive mutants, as autoimmune mutants, which trick we can do independently of the sensor NLR. So I don't need to take you through the whole thing, but the key point here is that these pathogens have evolved different effectors, totally different effectors, one being from the nematode and the other ones from Phytophthora, to suppress the sections of this network, at least, through totally different activities. This particular effector, SS15 from the nematode, is actually an inhibitor of the resistosome. It directly binds the NRCs and inhibits the formation of the resistosome. However, AVR-Cap1b is an effector that acts downstream. It acts on a protein called TOL9A, which is involved in vesicle trafficking. And somehow, that by acting on TOL9A, it's able to suppress the activity of this network. But what's also interesting is that both of these suppressors are only acting on NRC2 and NRC3, but they do not affect NRC4. And this is why we have actually nematode and Phytophthora resistance genes against these pathogens, because these resistance genes function through NRC4, which is not suppressed by these effectors. So you can see how this coevolution between the plant and the pathogen is occurring at multiple levels, including the fact that there are suppressors of different sections of this network, which is perhaps what is resulting in the expansion of these NRCs into family members that are able to evade suppression by the pathogen. So that's one reason. The second one is evolvability. So evolvability is really about making the system more complex. So instead of having one single protein doing the job, you have multiple ones. And to illustrate this, let me go back to ZAR1. So I told you how ZAR1 has this N-terminal alpha helix, which is when this protein oligomerizes forms this absolutely critical pore-like structure or funnel-like structure. Well, it turned out that this alpha helix is quite conserved in many other helper NLRs, including in the NRC. So this is an alignment between ZAR1 from Arabidopsis and an RC4 from solanaceous plants. And you can see that the N-terminal alpha helix is quite conserved. Half of the residues are invariant. And not only is it conserved at that level, but it's also defined by this motif sequence, which we call the MADA motif. You can computationally, using unbiased approaches, identify this N-terminal motif. And that motif is actually present in about 20% of the CCNLRs. And what's remarkable is if you map this motif across NLR diversity from dicots, monocots, etc., here's ZAR1, the NRCs are here, but even like a rice NLR, like from a monocot, so PIC2 is from rice, even monocot NLRs who are executors of cell death, helper NLRs, would also carry this MADA motif. So it's a signature essentially of helper NLRs, or at least some of the helper NLRs, and illustrate that this motif is very ancient, but has been retained in many of these helper NLRs. We showed that this motif is interchangeable, so you can take the ZAR1 alpha helix and introduce it in an RC4. You can even take a monocot, like PIC2 is again from rice, you can take that alpha helix of the rice protein and introduce it into an RC4, and it will still be functional. So this is a functionally conserved ancient N-terminus, N-terminal alpha helix and N-terminal motif that is present in the helper NLRs. So the reason why I'm telling you all of this is because it is really remarkable that the great majority of the NRCs have this MADA motif and this conserved N-terminus, and this is shown with the red color in these different branches in the tree, but absolutely none of the sensor NLRs in that NRC network carry this MADA motif. So you can see how what happened here throughout evolution. The sensor NLRs, they don't require this motif. They rely on the helpers to cause cell death. So they lost the motif. It's what is known in evolution as use it or lose it. If you don't need a motif, if you don't need a particular feature of your protein, then you're going to lose it. There is no selection pressure to maintain it. And that's exactly what happened in this, all of these sensors and NLRs, all of these R genes that have been discovered over the years don't carry that signature of the helper NLRs that allow them to cause a hypersensitive cell death. It's even more interesting because this particular clade here of sensor NLRs, they actually have an N-terminal integration that is before the CC domain and before the MADA sequence. And that N-terminal integration is acting as a bait for pathogen effectors and allows them to detect pathogens through that N-terminal integration, but obviously they cannot function through the pore-forming mechanism like ZAR1 because they have several hundred amino acids before that, actually, that structure. So the model we came up with, and this is really revisiting that figure I showed you at the beginning, is we have indeed functional specialization followed by diversification during the evolution of these NLR networks. And that resulted in sensors that have degenerated MADA motifs, they have N-terminal domain integrations, and that clearly cannot function in causing the cell death on their own without the help of the NRCs or the helper NLRs, which have a conserved MADA specifically at the exact N-terminus. So this shows again how the system is more complex because the sensors are more evolvable because they are not constrained by the maintaining that MADA sequence at the N-terminus, so they can acquire new features like N-terminal domain integrations to detect pathogen effectors through those new domains that they have integrated. And that's really a nice illustration of how these NLRs can really functionally specialize and how the system as a whole is much more evolvable than if they were constrained in a single protein. Okay, so now I'm going to shift gear and finish the talk with some applied perspectives. And this is some work we just wrapped up very recently, and I'm really excited to talk about it for the first time actually at a conference. And this is really relates to the idea of how plant immune receptors, at the end of the day, what they're doing is they're tricking pathogens, because pathogens have effectors that have targets, and they perturb these targets to basically cause a susceptible state in the cells they are infecting. But what happened throughout evolution is plants evolved these receptors that detect the effectors by binding them, by binding or detecting perturbations of the targets. But sometimes even better, they integrate these targets inside the, essentially the architecture of the NLR protein and now directly bait the effector this way. So this particular concept is fairly recent, dates to maybe the last 10 years. And when it was discovered that indeed throughout evolution, some NLR proteins acquired new domains that are evolving from the targets of the effectors. Of course, the idea came immediately that maybe this can be exploited in biotechnology to replace these integrated domains by some other domain and generate now made to order or designed NLRs or designed R genes that would give us resistance to a variety of pathogens. So the question maybe that I would want to raise now would be, what would be the ultimate integrated domain? If you had to actually pick a domain that you would want to integrate to replace the natural integrated domain, what would that be? And we gave that some thoughts, and we realized that actually the ideal domain to integrate will be a domain that is obtained from a mammalian antibody, because antibodies are actually able to bind pretty much virtually any molecule that you expose the mammalian immune system to. That's why we vaccinate. That's how we get our coronavirus vaccines to work. And that's why we generate also antibodies by vaccinating various animals. So the additional maybe twist to this is that in biotechnology, the so-called VHH or single domain antibodies, which are generated from the camelid families, so camels, llamas, alpacas, these are actually even better than the other or the classical conventional antibodies, which have multiple chains, but these have actually single domain antibodies and single chain antibodies. So the whole binding activity of the antibody can be brought down to about a 10 kilodalton domain or of these camelid antibodies. They're known as nanobodies or VHH antibodies or VHH domains. So we tested this, and we, as a proof of concept, we decided to use the existing nanobodies that are known to bind to GFP and mCherry proteins. And these are quite specific, although the target proteins are similar. We have enhancer that is one of the nanobodies that binds GFP, and LAM4 that binds mCherry. And you can see that they bind also in different positions. So we decided as a proof of concept to test this, and we called the system picobodies. So I'm really pleased to introduce these picobodies, which are brought to us by animals, strange animals like this one, like the camel. And what these picobodies consist of, they originate from a pair of NLRs called PIC1 and PIC2, which are originally from rice. And these paired NLRs, so PIC2 is one that is a typical MADA-CC NLR that is an executor of cell death, and it depends on PIC1, which has an integrated domain, and we replace that integrated domain by a nanobody. And so we make a picobody that is detecting GFP and another version that detects mCherry. And we can now test these for the hypersensitive response. And indeed, we can obtain a specific hypersensitive response. If you use the enhancer or the anti-GFP antibody, we get an HR against GFP, as you can see here. But if you use the mCherry one, then we don't see the HR against GFP, and that's a different nanobody here that is integrated. So the key point here really is that we can basically make these plant NLRs to detect a completely artificial protein that obviously the plant system cannot normally detect by introducing nanobodies into its canonical architecture. So does this work against the pathogen? So we did test this against the virus. This is potato virus X, and potato virus X can be engineered in various ways to express fluorescent proteins. And we just did that. And you can see here that the enhancer gives resistance to the virus because we are now looking at fluorescence. So there's very little accumulation of the fluorescent protein. And these are just experiments that allow us to look at the levels of the fluorescent protein. So you can see here that the virus, in the presence of the matching picobody, is not accumulating a lot of fluorescence in a similar level to a native R gene against the virus Rx here. So that's quite similar to Rx. So these picobodies can also confer resistance to a virus pathogen with a matching protein. So what's very exciting about this, of course, is that these are made to order R genes. Now we can actually dream about doing that. So we can take an effector from pathogen, generate nanobodies against it, integrate them into this NLR, and then presumably obtain resistance against any pathogen we start with. And I have to tell you that this is work in progress. We now have our first nanobodies against effectors, and we'll be testing whether this these indeed would function the same way that the anti-GFP and mCherry nanobodies have worked. We posted this work recently in bioRxiv, and I should really congratulate Yorgos and Clemence for doing a fantastic job in coming up with this system and wrapping the work rapidly. So I think with that, and I'm gonna wrap up. So I want to thank my team, particular team NLR. Lida did a lot of the suppressor work, especially working with Mauricio. I just mentioned Yorgos and Clemence with their nanobody work. Ola has actually done superb work in her thesis on integrated NLRs. And also various other people have contributed to this work, in particular Abbas with his help with structural biology. And also several of these folks have now moved on to other positions. And Aki in particular is the one who did the work on the MADA NLR proteins. So I also want to thank our collaborators, many collaborators, Tolga Boskurt at Imperial College, Mark Benfield, John Innes, Ryohei Terauchi in Japan at Kyoto, and Sumeng Dong at Nanjing Agricultural University, and also my colleagues at the ScienceBee Lab. And I hope I didn't go too long, and there is time for questions. So thank you so much for your attention.
Thank you very much, Jofien, for this really great talk. So about the evolution of NLR proteins, and I think a very nice example from basic science coming to applications and talking about pico bodies. So that's a very nice name also. So no, really great. We have quite some time for questions, so please go ahead. Let's see if there are questions. Okay, we have the first question of Jose Luis Reyes, please.
Hi, can you hear me now?
Yeah.
All right, perfect. Thank you for the beautiful talk. Oh, I have my dog here. He may bark, but let me hurry then. Sorry. What about the pathogens tricking the plant? Do we have examples where pathogens do something to? Well, you showed a few. I'm actually thinking of another example that was not in your image of microRNAs. That there's a few of them that target NLR proteins, right? So is it known anything about when do they contribute, and also probably in the context of symbiotic relationships where you don't, I guess the agent doesn't want to be recognized as a pathogen, but more like a friend. What could you tell us about
that? First of all, I mean, to answer part of your question, so the concept of effectors, the effectors don't have to be proteins, so they can also be nucleic acids and RNA. So the same thing I described there could be applied to nucleic acid effectors, if you like. The way, I mean, pathogens trick the plants in many different ways, in dozens of ways. I mean, I showed you one, which is the suppression of the immune system, right? So that's one very classical way. Typically, there's a kind of a dominant view that effectors are often suppressing the immune response, especially the so-called PRR-triggered response from the cell surface receptors. But in reality, pathogens trick the plant in lots of ways. I think you heard Saskia's talk earlier today, and she talked about completely different ways in which pathogens trick the plant. So I think the pathogen have a lot more tricks in their bag than the plant does. The plant is just detecting these effectors, activating immunity, and end of story. I think that's part of your question. The symbiosis field, I think we still don't fully understand how symbionts avoid or have sort of reached a pact with the plant to not activate immunity. So there's probably a lot of different mechanisms. There are examples with rhizobacteria where the rhizobacteria have effectors, which might be actually suppressing a response and suppressing the immune response. But I think that field is really, it's actually one of our unanswered questions in the plant-microbe interaction field. So hopefully, it's not my research, but hopefully we'll get to talk next year or in the coming years about this subject.
Great, thank you so much.
Thank you for your question.
Okay, the next question is from Alejandra Cubarrubias, please.
Yeah. Hi. Great presentation. Thank you.
Thank you.
I have two questions regarding the oligomerization of these NLRs. So the first one has to do with if you know the domains or the motifs that are involved in this oligomerization process.
Yes, so thanks to the structure, it's known that it's the N-terminal domain and also the NBRC, the central domain, that are involved in the oligomerization. There are some variations between, again, the CC and TNLRs, but those are actually the ones that are driving the oligomerization. The loose and rich repeat domains are kind of, they form the petals of the resistosome, if you like. They're sticking out usually. Yeah, so that's what happens. Is when the NLR is activated, it switches to binding ATP. Not always, again, but in most cases, and that switch is resulting in the conformational change that results in the oligomerization of the N-terminal domains.
I see. So the other question is, there are signals or chemical factors that avoid this oligomerization?
Oh, that's a very that's a great question, because actually in the mammalian world, in the biomedical world, actually there's a lot of radar companies that are trying to find inhibitors of NLRs, and they usually inhibit the inflammasomes, and it's important for autoimmune diseases, but also it's important in cancer and various other diseases or perturbations that we have. So in the case of plants, we just discovered the resistance of as a community only the last 2 years, but we're very excited. I didn't show you that data, but we now have data suggesting that this nematode effector that inhibits the NLR is actually binding to it and preventing the oligomerization. So it's very, very recent work in my lab, so not yet ready for prime time, but we think that there are effectors that can inhibit that process.
Sure. No, that's very nice. No, thank you.
Thank you for the great question.
Yeah. Okay, we have a question from the chat. I will read it.
Yep.
From Alexandre Noah. How do you ensure that your nanobody will be stable for a durable effect on treated plants?
Yeah, that's a great question, because obviously we know that the problem in plant pathology and disease resistance is durability, because pathogens, again, evolve very quickly and they evade resistance and all that. So in the preprint we posted, we actually did the stacking experiment. So gene stacking is a classic plant breeding strategy to delay essentially the pathogen evolution by combining multiple R genes together. And so we showed that we can actually stack these anti-GFP and anti-mCherry picobodies together, and they will still work. They will not interfere with each other. So that is actually hopeful, because that means maybe we can actually stack multiple picobodies and target maybe 4 or 5 components in the pathogen, which would delay, obviously, the evolution of the pathogen. So there's that. But the second part I really like about this is actually the problem often is that when you find an R gene in a germplasm, it's not necessarily functioning against the races of the pathogen that are currently present in your field. So my vision really is that we combine this with pathogenomics, where we discovered what races are present actually in our fields and causing an epidemic or a pandemic at the moment, and then extract effector sequences conserved in those epidemic, pandemic lineages or pandemic populations, and then make nanobodies against those. So the idea of having made to order R genes is also exciting because of that angle, that you could actually target the current pathogen populations that are causing problems, not necessarily an R gene that's maybe has been co-evolving with that pathogen for a long time and is already defeated. But yeah, it's a great question that we're thinking about, but the idea of stacking is obviously quite exciting.
Okay, thank you. Well, maybe I can ask a last question. So the picobody, so thinking about an application, so what you showed is that it works against proteins, let's say, for instance, GFP. Yeah. But could it work exactly the same against metabolites? So to really make sensors in plants?
Yeah. My understanding is that so this is, if this is based on immunization, right, of an animal to generate an antibody against it. So my understanding is that usually you need a certain size of the molecule to be able to make it antigenic. So usually it has to be conjugated to something else to be antigenic. So it's not impossible, but it's, I think, a little harder. But I would say that if we step back, I mean, the nanobody concept is obviously very exciting, but I mean, considering the very recent advances in protein structure, and for example, with AlphaFold and the capacity to predict with reasonable success structures of proteins, just the simple idea that we can integrate a domain that will bind anything is actually quite exciting. So I'm pretty sure that within years we'll be able to design protein domains that bind anything we want. You know, I think that now, especially with things like AlphaFold, I mean, that's the next step, right? With AlphaFold, is to predict protein interaction. I think there are people already. There are, like, I think, a preprint every day or something on this topic. So probably, I mean, the idea of having an effect or having a target, whether it's a metabolite or protein or peptide or an RNA, and generating a domain that binds it and that you can then integrate in your receptor is really exciting, and I think it's probably going to be feasible in the coming years. So I think the concept as integrating new domains that bait and the pathogen anyway, I think it's going to be a long-term strategy for this field.
Okay, no, really great. Thank you very much. We are very grateful that you shared your latest results with us, and hopefully we see you soon in real life.
Thank you, Stefan.
Thanks a lot.
I appreciate the invitation. Yeah, thank you to all for your attention and the questions.
Thank you.
Bye.
Goodbye.
Bye-bye.
Okay, so now I hand it over to Dan. I think we will go to Flash Talks again.
Not much to announce here. Flash Talks are next. They will go from 3:30 to 5 o'clock, Mexico City time. You can go to the website under Flash Talk sessions and see the participants for this session and the abstracts on the website as well. And importantly, after the Flash Talks at 5 to 7 p.m. Mexico City time, we will have the business meeting. This meeting is principally to identify new organizers for the next meeting in 2 years. So if you would like to be involved in organizing the next meeting, please stop by, and we'd like to hear your thoughts for the next meeting. So we will start with the Flash Talks now. See you all.
See you.
Hello everyone, my name is Eddie Mendoza, and today I'm going to present a short talk entitled Communicating Plant Science Using Comics, Lego Bricks and Beyond. Plants are the basis of our daily life. We eat them, we drink coffee and tequila, and we drive our cars using plant roots from millions of years ago. But does everybody see them as we do? If I show this picture to a non-plant science person, people mostly will talk about the animal, but not the plant. The world population is growing, and yes, the industry has been really close to scientists, but we cannot assume farmers have the facility to incorporate high technology and research in their current problems. And I don't mean we must pursue practical knowledge only, but there is a huge void between our work and what happens in the Mexican field, in reality. Thomas Kuhn emphasizes science not shared is like it doesn't exist. So, is our work invisible? We must put effective science communication in front of making citation numbers. Once work is done and published, it's our responsibility to share as far as possible. And one of the best ways to achieve this is by recognizing field work and each lifestyle by ourselves. In order to know how to properly communicate in every context, we should first realize how and who is listening to us. This pandemic has shown how important it is to communicate science and the impact of misinformation on society. Thus, I propose to incorporate friendly tools to illustrate plant science to the world. The film industry has shown everyone loves superheroes. Then, maybe we should create a superhero who can be heard. In this way, I've created Phytoman, whose messages about plant life have reached whole families on social media. Creating a plant biology comic book, it's an ambitious project, but I'm pretty sure it will become a feasible and powerful teaching tool. On the other hand, if K-12 scholars like Lego bricks, then we should use them to show how we study plants. That's why we will start a workshop journey in rural schools to close them up to STEM. As an example, we created Phenom, a Lego robot that shows how plant genotyping works. Teaching kids programming principles, plant biology, and next generation data can be fun too. Maybe the hardest public is farmers. This implies a revolution of exchanging experience. We should reach them first because the field is their niche. We are foreigners to them and nothing will change without their hands. Keeping in touch with farmers, kids, and society will also improve creativity to keep going in our labs. I encourage academia to communicate their work, being always inclusive. Science should be for everyone, not only for journals. Thank you for listening.
Hi, my name is Isaura, and I will be talking about cacti flowers transcriptomes. In the world, there are 5 times more flowering plant species than vertebrate species. The causes of the rapid diversification of flowers are enigmatic. Charles Darwin called this phenomenon an abominable mystery. But what is a flower? A flower is a short shoot that carries reproductive organs. The genes that determine the flower organs have been studied in Arabidopsis, and their interactions were organized in the ABC model. Although cactus flowers share features with Arabidopsis, they also have modifications that make them intriguing. A spiral perianth formed by a gradation from bracts to tepals, and the presence of the tissue which covers the ovary, called the pericarpel, which looks like a stem, so cacti flowers have been called branch flowers. I am interested in understanding the genetic and developmental mechanisms of floral diversification in cacti. I have worked with 2 cacti species with very different flower morphologies. Disocactus speciosus, which has big red bell-shaped flowers, and Disocactus exchami, which has small magenta tubular flowers. Why these 2 species? Well, because they are very closely related to each other, but have very different floral morphologies, thus they are a very interesting model for cacti. A way to understand which genes are expressed in each species during floral development, I extracted RNA from 3 developmental stages. I sequenced all the transcripts and put together 2 de novo transcriptome assemblies, one for each species. Then I analyzed the difference in expression profiles during flower development. What did we find? While the size and protein number in the transcriptomes of the 2 species are very similar, their expression profiles are different. We also found that some genes that have been previously correlated with the control of organ size were differentially expressed between the 2 species. 2 of these genes are the transcription factors big prodel and big petal, which are overexpressed in the perianth of the small magenta flower in comparison with their expression in the red big flower, suggesting that gene expression patterns between these species could be controlling floral size. Finally, if we want to understand which changes in the spatial or temporal expression patterns of genes that are involved in flower development and explain the vast array of flower forms we see in nature, it is important to study angiosperms with diverse floral morphology, such as cacti.
In this work, we investigated about the co-expression networks associated with auxin metabolism during transition from flower to fruit in Vanilla planifolia. The transition from flower to fruit is defined by 3 critical events: pre-pollination, pollination fertilization, and the fruit development. So far, the phytohormone auxin has been used as a reference to understand this process. In some groups, there is a variation called post-pollination syndrome. This syndrome is characterized because during anthesis the ovule is not developed, and the pollination is the signal that activates the development of the ovule until the fertilization. For this reason, we propose that plants with post-pollination syndrome present a different co-expression network than model plants. We use transcriptomes of 2 genotypes, the CH1, fruit fault tolerant, and CH6, susceptible to fault, in 4 developmental stages. Then, we modulate the co-expression networks, determined the number of functional modules, and calculated the hopscotch. As a result, we determined that co-expression networks had characteristics of a complex system, such as free scale, high clustering coefficient, and minimal path among nodes. Also, the network had different numbers of modules, and we determined that the CH1 had 29 authoritative hop genes, while the CH6 had 55 genes. The network shared 11 authoritative hop genes. The CH1 had 16 genes that were related with development, for example, flower development, fruit development, and size algo and division. On the other hand, the CH6 had 38 authoritative hop genes that were related with responses to stress. For instance, osmotic stress response, response to ultraviolet, far red, and blue light, response to hypoxia, response to bacteria, co-response, and response to salt stress. In conclusion, we propose the following model. In CH1, the conjugation genes generate a low level of auxins that is a characteristic of the fruit development. Also, participate some specific genes related to signaling and transport. On the other hand, in the CH6, the biosynthesis genes produce high level of auxin that is a characteristic of immature stage of the ovary. This situation provokes an incomplete fertilization and premature fruit drop. Finally, a lot of genes related to signaling and transport participated in this process. In this sense, we are trying to understand the transition from flower to fruit in Vanilla planifolia.
I'm Greta Moran, and I'm going to talk about the effect of heat stress on biomass production in broad wheat genotypes in the Yaqui Valley, Mexico. In the agricultural system, heat stress has the major impact in crops yield, and as shown in this graphic, the increase in annual temperatures has been evident around the world since approximately 1982. This increase in temperature has caused the loss of total zone area for wheat in Mexico. The sensibility of this crop at this particular abiotic stress has led us to analyze the performance of 4 genotypes of wheat. The genotypes in question are the ones shown here, coded genotypes 3, 6, 23, and 26. For these evaluations, 2 field experiments were carried out at the dates of December 2020 and January 2021, labeled the control and the heat stress respectively. For this, the anthesis time was evaluated and all the samples collected had this treatment. Afterwards, the data was analyzed by a descriptive statistical analysis. In this graph, we can see how all the genotypes under heat stress suffer from around 20% less total days of anthesis compared to the control ones. In the spikes, we can see how all the genotypes under heat stress suffer from around 20 to 25% less total weight compared to the control ones, being the most affected the genotype 6 and the genotype 23. In regard to the length, this one didn't change significantly. In the biomass, we can see how also the number of seeds didn't change significantly, but in their weight, we can see how genotype 26 and genotype 3 were the ones with less total weight change comparing the control and the heat stress. In the genotype 3, the total weight change being of 7%, and in genotype 26, 1.5%. These results led us to the conclusion that genotype 26 is the one better able to maintain its biomass under heat stress. However, further biochemistry and molecular evaluation should be taken to explain the ability of this genotype to confront heat stress and maintain its biomass. Thank you.
Hello everyone, it's Carlos, and today I'm going to present to you my current project, which is called Investigating the Effects of Retrotransposon Activity during female gametophyte development in Arabidopsis. So, Retrotransposons are the most prevalent class of transposable elements in the genome of Arabidopsis. Also, their mobilization can be a source of genome instability and modifications of gene expression. It's not clear what is the effect of their activity in the haploid female germline. Also, EBD is a single copy retrotransposon that belongs to the copia family. It is one of the most mobile transposable elements in the Arabidopsis genome, and it has the capacity to escape transcriptional silencing. So, to better understand the consequences of retrotransposon activity in the development of the female gametophyte, we are studying the transcriptional state of EBD in developing ovules and targeting its constitutive transcription through the CRISPR-Sontag system. Then, by RT-PCR and in situ hybridization, we showed that EBD transcription is mostly repressed in the female gametophyte and lives. However, we detected a spontaneous expression at a frequency of 15% that was correlated with the expression of antisense transcripts in the same locations, suggesting a residual failure of the mechanisms that repress EBD. Then we transformed Columbia zero plants with the CRISPR-Sontag system, and we tracked GFP fluorescence to confirm its expression. We observed GFP signal in the nuclei at premeiotic and postmeiotic stages of the female gametogenesis, where EBD is mostly absent. Finally, we compared the female gametophyte development between transgenic plants and wild-type plants. We did not identify any difference between wild-type plants and the transgenic plants. Finally, we conclude that at least two different mechanisms could prevent alteration upon activation of EBD. Either insertions of our transgenic constructs landed in neutral genomic locations, or yet to be discovered, post-transcriptional silencing mechanisms prevent EBD mobilization in transgenic plants. Our results suggest that gametophytic cells possess additional mechanisms to prevent developmental alterations caused by retrotransposon activity.
Hi, my name is Luis Ayala, and I'm talking about an antioxidant activity of the major terpenes of Callistemon citrinus in rat's feet, a high-fi diet. High-fi diets increase the risk of development of obesity, type 2 diabetes, mellitus, and cardiovascular disease. The increase in saturated fat activates various signaling pathways that promote and increase in reactive oxygen species and inflammation in different cells, affecting the cell function. Callistemon citrinus is a plant that had been reported biological activities. In this study, we evaluate the antioxidant activity of the major terpenes of Callistemon citrinus, 1-hexenol, limonene, and alpha-terpineol in rat's feet, a high-fi diet model. 36 male albino Winston rats were randomly divided into 6 groups, n equal to 6 to each group. Group 1 was the control, Group 2 was the high-fi diet. It contains 41.66% of Purina Robin Chow food, 20.83% of Inca vegetable fat, 20.83% of lard, and 16.68% of sucrose. The group 3, 4, 5, and 6 were administered orally with terpenes at different concentrations and the terpenic mixtures of dust, plus the high-fi diet daily for 15 weeks. We obtained the liver for the rats and did homogenase to analyze the levels of the biomarkers. High levels of oxidative stress biomarkers, malondialdehyde and 4-hydroxynonenal and advanced protein oxidation products were found in the liver and high-fi diet group. Conversely, the levels of the biomarkers in the groups with cineol and alpha-terpineol and limonene plus the high-fi diet showed a decrease in these parameters similar to the control group. Reduced glutathione showed low levels in all the groups compared to In the control group, the activity of the paraxenoson 1 significantly decreased in the high-fat diet, limonene, and cineol groups when compared with the alpha-terpineol and terpenic mixture groups. These results demonstrate the alpha-terpineol was the best to improve the damaged CASB oxidative stress as a result of the high-fat diet for 50 weeks. On the other hand, limonene and cineol has shown protection against the certain biomarkers of the oxidative stress. Thank you.
Good afternoon, my name is Sandra Mancera Lara, and I will be presenting the project named Obtention of Betalains from beta vulgaris out of an in vitro culture. This project was made in Universidad Nacional Autónoma de México and in Universidad Anáhuac, Mexico, and it was done by myself, Dr. Laura Martínez Montiel, and Professor María Teresa de Jesús Olivera Flores. Betalains are natural pigments present in multiple plant species, such as beetroots. They are considered as an alternative for red 40 usage in cosmetics and foods, as well as in vitro culture is considered as an alternative for obtaining pigments from cells and tissues instead of using complete beetroots that were originally cultivated for food. In this project, beetroot sprouts and bulbs were cultivated in hormone-free Murashige Cook solid culture medium from seeds. After that, they were cut for callus and cultivated in 4 culture mediums with growth hormones that were benzylaminopirin, 2,4-dichlorophenoxyacetic acid, picoram, and naphthalenoacetic acid. Benzylaminopirin was used by itself and combined in smaller concentrations with the other 3 hormones during 5 months. After this culture, callus were desegregated in 4 liquid culture mediums with the same growth hormone combinations during 2 months. Betalains were extracted from both callus and cells and quantified by colorimetry, and the results of these experiments were that benzylaminopirin and the combination of benzylaminopirin and naphthalenoacetic acid were the best for both callus and cells cultures, as well as they were the ones that presented most pigmentation. And this is because in 2008, George's book, he explained that some growth hormones affect metabolic routes and suspend secondary metabolite production, in this case, betalains. Picoram and 2,4-dichlorophenoxyacetic acid were 2 of those hormones with that effect in both callus and cells, and that's why they presented the least pigmentation. Also, pigments concentration rates from 0.1 milligrams per liter to 3.5 milligrams per liter from both callus and cells. In conclusion, the obtention of betalains from beta vulgaris out of an in vitro culture is possible depending on growth hormone combinations. Thank you very much for your attention.
Hello, my name is Gerardo Mejia, and I am going to talk about this project called Biological Traps to Recruit Plant Growth Promoting Bacteria, which is part of my PhD investigation. As we know, soil represents an important source of bacteria that associate with plant roots. Once in the root, some of these bacteria are able to promote plant growth by different mechanisms like nutrient solubilization, phytohormones production, nitrogen metabolism, and suppression of pathogenic organisms. These bacteria are called plant growth promoting rhizobacteria or PGPR. In this work, we established an scrutiny system in order to identify communities of PGPRs by subjecting tomato plants to different soil sources as inoculum in a hydroponic system. After 60 days of interaction and selection, plant phenotyping was used as an indicator of root and interactions with bacteria. Moreover, those plants with significant differences with respect to their control suggest having recruited a PGPR community in their root. Treatments with inoculum were proved with 50% to stimulate plant and bacteria interactions. Sterile controls of each inoculum were performed to know the effect of no biological components in the inoculum. Additionally, we used 50 and 100% of fertilized controls with no inoculum. As we can see, inoculum treatment performs better compared with their controls, and it reached the 100 control despite just having 50% of fertilizer. Finally, we used 16S ribosomal RNA and shotgun sequences to describe and understand the taxa and coding genes responsible for plant growth promotion. At the moment, we can highlight that that the abundance of some taxa correlate positively with some phenotypic characteristics like chlorophyll concentration, weight, and step diameter. As conclusions, we have that the correlation between the abundance of some taxa with plants phenotype suggests that they recruited a PGPR community in their roots, and that's why they obtain a better growth performance. So thank you so much.
Hello, I'm going to talk about population genomics of Quercus mcdougalli, an endemic oak in the Sierra Juárez, Oaxaca, Mexico. Oak trees are a species of the genus Quercus, which is widely distributed in the northern hemisphere. Mexico is considered a prime diversification center of this genus since it has the highest richness of oaks in the world, and also because approximately half of them are endemic. One of these is Quercus mcdougalli, which is exclusively distributed in the Sierra Juárez de Oaxaca and is classified as an endangered and a threatened species. We wondered about its genetic variation and structures, since this could affect the species survival. So we analyzed its genomic variation by sampling 79 individuals in 8 sampling sites located in the northern and southern zones of its distribution. We used data from massive sequencing and detected 8,186 single nucleotide polymorphisms, SNPs. We found moderate genetic diversity, as well as a low genetic differentiation and low inbreeding levels, which indicated that the species remain as a single population. However, the minimum expanding network, PCA and DAPC, showed a slight separation between the northern and the southern zone. Also, the admixture and fast structure analysis showed a genetic group with incipient structuring in the southern zone. The isolation by distance analysis showed a genetic variation gradient between these two zones. A closer look at locus level allowed us to identify several unique alleles at particular sample sites. Although the analysis of outliers SNPs based on FST did not show evidence of locally adapted genotypes, we identified the presence of variation at the nucleotide level associated with the differentiation gradient between zones. These results allow us to complement ongoing conservation plans and monitoring strategies for Quercus mcdougalli, particularly in the southern zone. Thank you for your attention.
Hi everyone, I'm Fernando from the Universidad del Papaloapan, and I'm going to talk about a research project that is called Isolation and Identification of Fungal Pathogens in Tropical Fruits and the Science of Synthetic Double-Stranded RNAs for Post-Harvest Protection via SIG strategy. As a short introduction, plant diseases are responsible for up to 30% of losses worldwide. The most of them are attributed to fungal pathogens. And to produce enough food, we need to develop new strategies for crop protections. In this work, we went to a look at markets and we recovered tropical fruits that were visibly infected by fungi. We isolated them in PDA medium and then by ITS sequenced and subsequently phylogenetic analysis, we molecularly identify them as Aspergillus tubiensis from pineapple and La Pseudoploida pseudobromea from mango. Both of them has reports as pathogens in crops. So the SIG method is based on the use of double-stranded RNA molecules that inhibit the development and growth of pathogens by silencing key genes of them. So in this work, we selected the TCL gene because it is involved in the biogenesis of small RNAs that regulate several molecular processes, including pathogenesis. For each fungi, we found 2 TCL sequences, so we choose a low similarity region between the TCL of the host and the fungal TCLs, and a 250 base pair fragment was selected between the death-like helicase and helicase C terminal domains for the designs of these double-strand RNAs. We design primes and we amplify them by PCR from genomic DNA, and then we add a complementary ends to fuse them in a single construct of 500 base pairs of length. Finally, a T7 promoter sequences was added to them for a further double-strand RNA synthesis reaction. The perspectives of this current work is the evaluation of these double-strand RNAs in bioassays that consist in the inhibition of spore germination and the inhibition of the radial growth of the fungi in PDM medium, as well as in vivo assays, to know their potential as a biofungicide. So this is the stage of this work so far. Thank you for your attention.
Hello, my name is Efraín Conejo Davila, and I'm going to present the project titled Molecular Effectors Involved in the Germination Process of the Mycetul P. caliculatus. Psittacanthus caliculatus is a parasitic mycetul that infects mesquite trees and absorbs nutrients from the host through a specialized structure called a hausterium. It is estimated that 80% of the human trees present an infection. It was hypothesized that once the seed is stuck, some metabolites from the host act as a cues to promote the formation of the connection between the parasite and the host. To complete the hausterium formation, the mycetul proceeds to degrade the bark tissue. During this process, it was believed that the cell degradation promotes the damp segregation that can result in stress response to the infection process. The objective is to determine the host metabolites involved in the mycetul germination and the host stress response to the interaction with fragmented DNA of the host and the parasite. We germinated mycetul seeds in different substrates. The result presents a higher germination into the host bark and in filter paper under like conditions. The results suggest a specific compound present in the host bark that promotes seed germination. During the germination process, we collect the volatile compounds emitted by the mycetul and identify as principal components the alpha-bisabolol, dihydroactinodiolide, and 2,6-dihydrobutylquinone. We identify and quantify enzymes involved in the bark degradation. In specific, cellulase and beta-1,4-glucosidase show the highest activities in stage 3 during the hausterium formation process. In addition, we investigate if the mycetul can synthesize hausterium-inducing factors. As a common phenomenon, there exists a high decrease in the hydrogen peroxide and total phenolic compounds concentration from stage 2 and 3 related to the infection process. Also, the stage 2 presents the biggest peroxidase enzymatic activity. Those results suggest the mycetul ability to degrade the host bark liberated the phenolic compounds derived from the lignin to synthesize hausterium-inducing factors. To elucidate the ability of the host to recognize the mycetul DNA, we expose the host leaves with a different concentration of mycetul DNA, self-DNA, and mechanical damage to contrast the metabolic early response to the stress, and finally we took samples immediately 1 hour and 2 hours after the exposition. The quantification of hydrogen peroxide of the sample show a similar response to the mechanical damage and self-DNA. Contrary, the mycetul DNA exhibit an increase after 2 hours to the more concentrated solution. The phenolic compounds content present an scavenging just after the exposition to the stress, and over the time we can see a small increase in the content regarding to control samples. We can conclude that the P. caliculatus mycetul germination is mediated by physicochemical factors and host metabolites that can be used to the production of hausterium-inducing factors, and the host presents an early oxidative stress to his self-DNA contrary to the DNA of the parasite.
Hello, today I talk about the effects exerted by metarisonal supply on plant growth. Metarisonal species are entomopathogenic fungus that can infect more than 200 species of insects And recently has been reported that some meta-region strains can interact with plants. The aim of this work was to evaluate the effect of three meta-region anisoplia strains on plant growth. For this, we used the strain 20, 25, and 28 in interaction with Arabidopsis thaliana under in vitro condition. Here we evaluate the direct and indirect interaction using a split system. The interaction was evaluated after 7 days post-inoculation. In both conditions of the interaction, we found that meta-region anisoplia strains can improve plant growth, particularly under split interaction. Also, the total chlorophyll in Arabidopsis plants were increased by the influence of the meta-region strains. As you see in the last slide, the best effect was found under split interaction. For the reason, we identified the volatiles produced in this interaction. We found that beta-caryophyllene, uracemene, and p-cemene were produced as ionic compounds. Additionally, we performed an experiment in which we evaluate the effect of these strains in Arabidopsis, tomato, and maize plants grow in pot soils. Also, we included the Trichoderma virens as a positive control. We found that three meta-region strains can improve Arabidopsis fresh weight after 14 days post-inoculation. The similar way, the interaction of these strains with tomato plants improve tomato fresh weight. Also, the fresh weight gained in maize plants was obtained with all strains included Trichoderma virens. All results show that these meta-region strains could be used in crop production with the advantage of the insect control and plant growth promotion. Thank you.
Hello everyone, I'm Citlali Fonseca and I'm a postdoc at UC Berkeley, and today I'm going to present to you some of the results I got in my previous postdoc at Instituto de Biotecnología UNAM in Mexico, and the title of my presentation is Metallothionein 1A regulates rhizobial infection and nodal organogenesis in Phaseolus vulgaris. Metallothioneins are metal ion binding proteins that have important roles in plants. For example, they are involved in the regulation of cell growth and proliferation, protection against heavy metal stress, and responses to pathogen attack. In a previous transcriptomic analysis in your lab, we found a metallothionein 1A was upregulated under nodulation conditions and downregulated under mycorrhization conditions. In addition, an in silico analysis and expression profile showed that this gene was most highly expressed in uninoculated and inoculated roots with rhizobia. So we hypothesized that this gene could have an important role in common bean and rhizobia symbiosis. We identified 6 metallothionein genes in common bean genome, and they were classified into 3 classes based on their homology with those metallothioneins in Arabidopsis thaliana. We analyzed the expression profile of MT1A at early and late stages of nodulation in common bean, and we observed that this gene was upregulated at 7 and 30 days post-inoculation, but it was downregulated at 21 days post-inoculation. Also, we observed promoter activity in cortical cells near to the form root hairs and in nodal primordia, as well as an infection zone on young nodules and periphery of the infection zone of mature nodules. After reverse genomics analysis, the downregulation of MT1A showed a reduction in infection events at 7 and 10 days post-inoculation with 50 and 75%. In addition, the total number of nodules was reduced at 14 and 21 days post-inoculation, and those nodules presented a reduction of 50% of nitrogen fixation capacity. Finally, an histological analysis showed that the number of infection cells and the size of the cells in the epidermical and parenchymal region was reduced in MT1A silent nodules. So we can conclude that MT1A gene is required for the proper development and progression of infection event during the early stages of nodulation in common being, and also has an important role in the nodule organogenesis in this legume. Thank you so much for having me, and see you next time.
Hi, I'm Gabriela. I'm a PhD student at Instituto de Biotecnología UNAM. I'm going to present my project entitled Role of the M6A reader protein ECT8 in the response to stress in Arabidopsis thaliana. Methylation of adenosine at the N6 position is the most common internal modification found in the eukaryotic mRNA. It's a conserved post-transcriptional regulation mechanism that affects the fate and function of the modified mRNA to regulating fundamental biological processes. Addition of M6A is a reversible and dynamic process where the protein complexes responsible for this addition, removal, and recognition have been described. Most of the reader proteins recognize M6A through the highly conserved domain known as GTH. This domain is found only in eukaryotes. In Arabidopsis, 13 proteins with this domain have been identified. Among these, previous work in our group showed accumulation of the transcript of the gene ECT8 under different conditions associated with low water availability or upon abscisic acid addition. Furthermore, in germination tests in the presence of ABA, we observed a partial insensitivity to this hormone when we used 3 different tDNA insertion and mutant lines for the gene, indicating a possible defect in its perception. Therefore, we suggest an important role for ECT8 in the response to situation of water deficit. However, the biological functions, the molecular partners, and the biochemical properties of the ECT8 protein remain unknown. Thus, it is of our interest to evaluate the molecular role and biological function of ECT8 under conditions of water limitation, identifying the interacting proteins and mRNAs during germination in the presence of ABA, and thus understand their contribution to the stress response. Finally, I want to thank all the members of this project and the institution involved.
Hi, I'm Soraya Esparza Reynoso, and I'm PhD student of Chemical and Biological Research at the Universidad Michoacana de San Nicolás de Hidalgo. I'm going to present my work entitled Trichoderma troviridae mated volatiles improve growth for Arabidopsis silence through modulation of sucrose transport and metabolism. I begin by mentioning that some Trichoderma species are able to colonize the plant rhizosphere and provide pathogen-resistant abiotic stress tolerance and improve plant growth and development. In the present work, we tested the effect of volatiles emitted by Trichoderma troviridae in Arabidopsis silence growth in media supplied with or without sucrose. The lack of sucrose in the media affected the normal plant development and also repressed the root branching and biomass production triggered by Trichoderma volatiles. To test whether volatiles modulate the sucrose allocation in plants, we evaluate the expression of sucrose, a sucrose protein importer that supports sucrose from landing. According to the expression pattern driven by sucrose promoter, plants grow in sucrose-deprived media and exposed to Trichoderma volatiles show an increase in expression at the root tips, unlike to the repression observed in plant growth with sucrose. This suggests that sucrose expression is modeled by volatiles, and we also determined that volatile 6-pentyl-2H piranto-1 and the locally applied sucrose replaces the sucrose expression at primary root tips, indicating that Trichoderma induces a reallocation of sucrose in the plant. Besides, We report the sucrose and glucose content in shoots and roots was increased in plants exposed to Trichoderma volatiles. And this positive correlate with the increasing expression of 3-isoform or sucrose photosynthesis in leaves. Finally, volatiles affected the expression of genes encoded with sugar transporter, especially the SWIT4, SWIT11, SWIT12, and SWIT16. To summarize, our research revealed that Trichoderma volatiles induce sucrose biosynthesis and improve its transport via sucrose and sweet carriers to trigger plant growth formation and sugar exudation, which ultimately attract Trichoderma to the rhizosphere. And that's the end. So thank you.
Hello. Agave species are highly tolerant to abiotic stress. Previously, we found that in Agave tequilana transcriptome, the most highly expressed genes are related to abiotic stress tolerance. Now, I will present phenotypes of several species of agaves facing abiotic stress. These are the collaborators of this work, which was carried out with the phenomic facility of the Colegio de Postgraduados in a greenhouse under monitored conditions. When you leave agave tequilana plants 3 months with no water, we can see that the plants lose turgor but are still alive. If you irrigate to field capacity, a recovery of turgor in rehydrated plants is seen. This is the visualization of chlorophyll fluorescence kinetics. The photosystem's 2 efficiency was affected by drought, but recovered by rehydration. However, while most of the photosynthesis parameters are lower in drought, the non-photochemical quenching rises. When we evaluated similar treatments on agave angustifolia, the turgor was also affected. You can see the small cells in drought recovered by rehydration. The plants in drought have less photosynthetic capability than rehydrataceous ones. But again, we found high levels of quenching parameters. As you can see, all photosynthesis parameters decrease in plants kept without water along the experiment, but the NPQ raised. In drought conditions, chloroplasts are aggregated probably as a protection mechanism. The carotenoids accumulate, but in thirstling, so does chlorophyll. The peroxidase transcript accumulation shows that only one peroxidase gene is induced by drought. When we evaluate drought effects on bulbils of angustifolia ecotype growing in coastal areas, the phenotyping shows that most of the parameters don't change by drought, except again NPQ, which is clearly elevated. In these plants, genes involved in stress protection are constitutively expressed. In control, but only one leaf isoform is induced by drought. Finally, we found conditions where this ecotype loses its chloroplast, as we can see in these empty cells. But the supplementation with nutrients recovered its photosynthetic capacity. We can discuss the conclusions or our phenotyping facilities in this email. Thank you.
Good morning. I am Luis Sarmiento from CIDIC Sinaloa, and the title of my work is Bioinformatic Analysis of the Xyloglucan endotransglucosylase hydrolysase gene family in Solanum lycopersicum and expression profile during the arbuscular mycorrhizal symbiosis. Xyloglucan are the most prevalent class of hemicellulose in the cell wall of higher plants. Xyloglucan endotransglucosylase hydrolysase XIH are a family of xyloglucan modifying enzymes responsible for the cleavage and rearrangements of xyloglucan backbones. Arbuscular mycorrhizal symbiosis is an association between soil fungi and the mossland plants that induce a systemic resistance against biotic and biotic stress. Recently, in our working group, a transcriptomic analysis of leaves from mycorrhizal tomato plants was performed and showed that genes involved in cell wall degradation and synthesis, including XTH, are differentially expressed in response to this symbiosis. However, little is known about this gene family in Solanum lycopersicum. The objective of this work was to characterize the XTX gene family from Solano lycopersicum genome by bioinformatic approaches and to evaluate the expression patterns of some genes members during the arbuscular mycorrhizal symbiosis. We identify 37 XTX genes from tomato genome that were clustered in a subfamily 1, 2, and subfamily 3. Also, all XTH proteins from tomato showed a conserved domain responsible for the catalytic activity on these proteins. Phylogenetic analysis of XTX proteins were clustered in an ancestral family in subfamily 1, 2, and subfamily 3, which is divided into two families named 3A and 3B. These results suggest that XTX proteins from tomato were clustered in a similar manner to other plant species. In silico gene expressions of XTX genes from tomato in different tomato organs exhibit low tissue specificity. To provide more information about this gene family, we analyze the relative expression of some XTX genes in leaf of mycorrhizal plants, where 3 genes were upregulated and 2 genes were downregulated in response to this symbiosis. Taken together, these results provide a comprehensive and systematic understanding of this gene family for future studies in tomato during different ground cycles, stress, and interaction with microorganisms. Thank you.
Hi, I'm Telma Parra, and I will talk about the study entitled Phaseolus vulgaris tetraspanin 8 plays a key role during mutualistic interactions. The arbuscular mycorrhizal and root nodule symbiosis require many mechanisms for their development, such as cell reorganization and accommodation of symbionts biovesicular trafficking. Though some proteins involving membrane fusions are important for these associations, tetraspanins are transmembrane proteins that organize into tetraspanin-enriched microdomains where they recruit specific proteins. Thereby, tetraspanins are involved in membrane fusion and in biogenesis of extracellular vesicles such as exosomes. In fact, TET-A promoter is active during nodule development, and TET-A increases its expressions upon rhizobia inoculation. Therefore, we wanted to address the role of TET-A in root nodule and arbuscular mycorrhizal symbiosis in detail. We found that TET-A promoter is active in arbuscule-containing cells, and we observed that TET-A-GFP fusion protein localized to the plasma membrane and some vesicles. Furthermore, with this construct, we generated Phaseolus vulgaris transgenic roots to determine the phenotype of TET-A gene overexpression on the rhizobial and mycorrhizal colonization. Compared with the control, TET-A overexpression in roots generated more nodules, and these were larger with higher biomassa, but nitrogenase activity per nodule dry weight was decreased. These images show that nodules overexpressing TET-A were larger and pinker than the control nodules. TET-A overexpression also increased phosphate transporter 4 expression and frequency of arbuscules. In addition, TET-A silencing reduced the number of root nodules, which were smaller and less efficient in nitrogen fixation than the control. And TET-A silencing nodules had little or no rhizobium colonization. Also, TET-A silencing decreased phosphate transport expression, generate fewer arbuscules as the control, and increase infection sites. Therefore, we propose that tetraspanins are new players regulating the mutualistic interactions and suggest that TET-A is necessary for rhizobia and mycorrhizal fungi colonization.
Hello, my name is Manuel Mendez Gomez. I will be talking to you about my work entitled Azospirillum brasiliense sp. 245 triggers cytokinin signaling in root tips and improves biomass accumulation in Arabidopsis through canonical cytokinin receptor. Okay, in this work, we analyzed the participation and the role of cytokinin signaling in the promotion of plant growth stimulated via Azospirillum brasiliense under 2 types of interaction, direct contact and volatile. Both interaction types increased root and shoot fresh weight, but had contrasting effect on primary root length, lateral root formation, and root hair development. The cell proliferation in root meristems analyzed with the cytokinin B1 goose reporter decreased over time with direct contact, but was augmented by plant exposure to volatiles. The expression of the response marker to cytokinin ARR5 was bigly increased in the shoot in the presence of azospirillum, while in the root an increase in expression of the reporter ARR5 and TCS-GFP was observed, mainly under condition of direct contact. Using mutant plants in the AHK receptors of cytokinin, we found that azospirillum was enabled to promote shoot growth of the double mutant and of the triple mutant, as demonstrated by the fresh weight of the shoot in the conditions used. The analysis in the development of the root system showed that azospirillum promotes the formation of lateral roots and the root hair in the single and the double mutants in a similar manner to the wild type, while the triple mutants was insensitive to the promotion of the plant growth stimulated by azospirillum. These results indicate the participation of the signaling pathways of cytokinin in the promotion of plant growth stimulated by azospirillum, regardless of the type of interaction. Nevertheless, additional studies are required to further define the precise role of cytokinin in the different mechanisms associated with the rhizobacteria-mediated phytostimulation. Thanks so much.
I would like to thank the organizers for the invitation to present my work today on the structure of plant mitochondrial complex 1 intermediate from etiolated mung beans. The mitochondrial electron transport chain is an essential bioenergetic process that's catalyzed by 5 large membrane protein complexes of the inner mitochondrial membrane, known as complex 1 through complex 5. And these complexes can come together to form higher order structures known as respiratory supercomplexes. All of the structures shown here are from mammalian mitochondria, which is a much more well-studied system. But in plants, the mitochondrial electron transport chain is still an essential biochemical process. And until very recently, we did not have a very good structural description of the plant complexes. Though we could see from these negative staining electron microscopy images that the plant complexes were very different, specifically in this gamma carbonic anhydrous domain. So my lab set out to fill in this gap of structural information using the model organism Vigna radiata, or mung bean, in which we were able to grow etiolated in the dark and isolate mitochondria from them, then extract the mitochondrial complexes from the membranes using detergents and run them on a sucrose gradient. Given that the mitochondria is very low abundance in plants and the sample is limiting, we took a partial purification approach in which we took the partially purified sample off the sucrose gradient and used it directly for cryo-EM grid preparation, and then used classification algorithms to isolate the individual complex particles and solve multiple high-resolution structures of the plant complexes simultaneously. Today, I'll be talking about complex 1 star. So we were able to solve a high resolution structure of this complex 1 intermediate and assign and build 31 subunits of complex 1. Although complex 1 star is missing the distal proton pumps of the full-length complex 1 shown in the top corner, it contains the plant-specific gamma carbonic anhydrase domain. We were able to assign the composition of the gamma carbonic anhydrase domain and show that one of the 3 possible active sites still has a catalytic zinc and could potentially undergo catalysis. And we also showed that the interface between the gamma carbonic anhydrase domain and the membrane arm is very large. The complex 1 star is a plant-specific assembly intermediate. We looked at the structure to see whether or not we could understand what plant-specific features would result in this different assembly pathway. And indeed, we could see quite a few differences in the accessory subunits at the interfaces between the different modules of complex 1 that might influence the assembly. I'm just showing a few examples here. Thank you very much. I hope you enjoyed this flash talk. I'd like to thank everybody in my lab, as well as my funding sources.
Hello, everybody. This is my PhD work. The cuticle and the cell wall share functions such as mechanical support, control of water loss, and protection during the attack of pathogens. In fruits, both the cuticle and cell wall have changed during the fruit of development. The cuticle can change in both thickness and composition, and the cell wall undergoes degradation of the polymer that composes it. This has been related to the reduction in firmness of the fruit. There are several enzymes that participate in the degradation of the cell wall polymers. For instance, rhamnogalacturonane lyase that degrades the polymer rhamnogalacturonane 1, which is component of pectins. Within our work team, the overexpression of rhamnogalacturonane lyase was carried out. Transgenic tomato had ripening out of phase and long shelf life relative to isogenic lime. We found an increase in gene expression in root and leaf tissues. In fruit, we observed an increased expression during the initial stage of fruit development. However, the expression of the gene decreased when endogenous genes begin its expression in the maturing stage. That is why we can observe a decrease in the expression of transgene, suggesting that co-silencing is occurring. Based on our finding, we decided to measure respiration rate and ethylene production. Indeed, we found in transgenic fruit that the climateric peak appeared 2 days later compared with isogenic lime, and with an ethylene production twice as high. After we observe in the ethylene pathway, we select important genes to ethylene biosynthesis and response. We found that SAM was expressed until 49D88. Surprisingly, the two gene ACS allow us to distinguish between two ethylene systems. In transgenic H2, we can observe that high expression of AC2 in 56D88 was expressed. Also, the ACO1 gene response of ethylene production was downregulated in all samples compared with transgenic lime. We decided to analyze other independent transgenic lime. Here, we observed that the gene ACO1 was also downregulated relative to isogenic lime. Here, the activation of ethylene biosystem 2 was distinguished by the expression of ACO2 after 49D88. Interestingly, we observed that ethylene response genes E4 was expressed as we expect, opposite to E8 that was downregulated in 56D88. For this reason, we are working with another genes related to the cell wall degradation during fruit ripening, such as pectate lyase, among others. Thank you so much.
Hello, my name is Juan Olivares, and I'm going to talk about our work titled Downregulation of Phaseolus vulgaris aquaporin PBPIP4 in person modulation. Aquaporins are membrane proteins that facilitate the movement of water and other solids through membranes, and they are implicated in several physiological processes. It has been proposed that hydrogen peroxide is transported from the aquaplasts into the cells by aquaporins, and this is important because hydrogen peroxide is needed for the polar growth of rod hairs and for the interaction of rhizobia with legumes that leads to the development of rod nodules where the nitrogen fixation take place. We are interested in studying the role of aquaporins in the Phaseolus vulgaris rhizobium interaction, and we found an aquaporin gene, PBPAP24, that is mainly expressed in root hairs and is induced by inoculation with rhizobium. By means of promoter activity assays, we found that the PBPAP24 promoter is active in the central vascular cylinder and in root hairs of inoculated roots, and it is highly active in the root nodule primordia and in young nodules. However, in mature nodules, the promoter activity is restricted to the vascular bundles. In order to gain insight into the role of PBPIP24 in the Phaseolus vulgaris rhizobium interaction, we generated transgenic roots that overexpress this gene or that are downregulated in this gene. The roots that overexpress the aquaporin gene form more nodules than the control, and they fix more nitrogen. On the contrary, the roots that are downregulated in this gene form less nodules than the control and fix less nitrogen. With this, we can conclude that the aquaporin PBPIP24 has a positive role in the Phaseolus vulgaris rhizobium symbiosis. Thank you.
Hello everyone, my name is Guilherme and I'm a PhD student at the Federal University of Santa Catarina in Brazil. And today I'll talk about a part of my PhD thesis, which is entitled Comparative Study of Polyamine Changes upon Desiccation of Eugenia piriformis and Eugenia astringens. This work was supervised by Professor Neuza Steiner. So Eugenia astringens and piriformis, they produce desiccation-sensitive seeds, which means that they're dispersed with high water contents and high metabolic activity, which hampers conservation by traditional methods. The thing is that desiccation actually happens in a spectrum. So we have species that are highly sensitive to desiccation, species that tolerate mild desiccation, and species that are highly desiccation tolerant. Polyamines are involved in several processes in seeds, including protection against abiotic stresses. So we believed that they might be related to desiccation tolerance. Therefore, our study aimed to verify the effects of desiccation on seed germination and polyamine content, and investigate the relationship between polyamines and desiccation tolerant in Eugenia species. For that, we collected seeds in Brazil, and we desiccated them until they reached 50% of germination, which we considered their water content threshold. And we verified the polyamine content in both fresh and desiccated threshold seeds. As a result, we first observed that putrescine increases more than 3 times in Eugenia piriformis when they're desiccated to the germination of 50%. And this species is more desiccation sensitive than Eugenia astringens. We also see that for spermidine, we have more spermidine content in Eugenia piriformis than we have in Eugenia astringens, but for both species, spermidine is the most present polyamine. We also observe a higher content in spermine in Eugenia piriformis than in Eugenia astringens, but their content do not change among fresh and desiccated seeds. We also see that the free total PAs that do not change, but we see that the ratio between putrescine and spermine will increase in Eugenia piriformis, again, a more sensitive to desiccation species. As conclusion, we see that polyamine content is species dependent, but spermidine was the highest polyamine for both species, and it might be related to desiccation sensitivity. Besides, the decrease in germination was correlated to an increase in potassium content and polyamine ratio for gene piriform seeds. Thank you so much for watching, and if you have any questions or any comments, feel free to contact me or my advisor through our emails. Thank you so much.